the time when we choose this path. We are all ready to take a leap into the unknown. We embarked on our journey since 1991. For these 30 years, we are down to earth and forge ahead with our initial intention of bringing better healthcare for all. During turbulent times, we adhere to innovation as always. Along the way, we stay close to our customers, going deep to the front line to achieve continuous breakthroughs. From a single product to the completed solution. From patient monitoring and life support to in vitro diagnostics and medical imaging and so on. From primary medical institutions to top-notch teaching hospitals. From one-lane focus to diversification. From a startup to a global company. "30 years on, we never stop innovating ourselves. Together with our global partners to face challenges and celebrate success. At the age of 30, we are ready to take on new challenges with our original heart and will. We will be always striving to develop and share technologies. Creating customer value with comprehensive solutions and broad portfolio.
At Mindray, we work tirelessly on improving our analyzers to help every laboratory fulfill their mission. And now, Mindray is proud to present the cute BS240 Clinical Chemistry Analyzer, a compact, utility, technology-enhanced, and efficient diagnostic tool for today's fast-paced labs, tailored to fit the limited space of small to mid-sized labs. Mindray BS240 boasts an almost unrivaled compact footprint in the 200 tests per hour throughput autochemistry analyzer market. BS240 offers a flexible solution to the arrangements of sample and reagent positions. The doctor can adjust the positions according to the test requirements of the day. BS240 features the unique smart sampling technology for HbA1c testing.
After more than 2 decades of development, Mindray Clinical Chemistry has established a comprehensive portfolio of solutions ranging from benchtop analyzers to chemistry CLIA integrated systems with original manufacturer reagents, calibrators, and controls, fulfilling a wide range of laboratory needs. Now, we launch our next generation standalone chemistry analyzer, BS430. Mindray's new flagship standalone chemistry analyzer strikes a perfect balance between size and throughput and will be a reliable solution for your expanding laboratory. BS430 has a large region onboard capacity and sample loading volume, which is suitable for a wide range of laboratories from 200 chemistry sets to 800 tests per day. Together with Mindray's reagents and calibrator, BS430 provides a system solution with traceability, which offers users not only accurate results, but also intelligent functions and easy maintenance. Equipped with a high-precision aspiration system, BS430 can maintain a high degree of efficiency. A sample clot detection function can prevent abnormal results at the source, and the waterfall cleaning design can guarantee a minimal carryover rate. Furthermore, BS430 has a concave grading system providing 12 wavelengths and ensuring accurate results via a stable and reliable photometric system. The intelligent system supports abnormal reaction curve detection, including substrate depletion of enzyme parameters. BS430 is able to identify samples with abnormal reaction curve caused by high enzyme concentration and trigger auto-dilution and auto-rerun. Moreover, BS430 can provide an enzyme linear extension function which delivers reliable results without dilution and rerun, reducing not only reagent consumption but also TAT time. BS430 also supports HbA1c onboard hemolysis, effectively reducing the cumbersome procedures of manual preparation of hemolysates and preventing biohazard risks and manual operation errors. In terms of maintenance, BS430 is equipped with a maintenance-free, solid direct heating system in its reaction compartment. As for its operation, an intuitive service wizard in the software helps operators carry out automatic maintenance step by step, simplifying maintenance procedures and increasing ease of operation. In this era of technological advancements, expanding laboratories with increasing samples are facing limited space and longer TAT. Their current chemistry systems may not be able to meet the growth in demand. In this situation, the powerful and compact BS430 is the right solution for you. For emergency tests, BS430 can wake up from sleep mode within 1 minute and generate a report within 30 minutes. Mindray is dedicated to providing comprehensive solutions for a range of laboratory needs. The BS430 features more than 60 different reagent parameters, all with the necessary calibrators and controls. BS430, a reliable solution for your expanding laboratory.
In today's climate of patient care,
You make it simple, you know. Yes, sir. Now, I have my PowerPoint. If I forward it to you, you can. Yes, please give me forward to it. So several things are tested here. Maybe next time.
Everybody is very active building the institute. Oh,
Yeah, yeah.
This is the biggest amongst all aims. This is the biggest.
After that, there are other aims also. So in this
Total 22 now.
22. Total 22.
Good morning. Good morning, sir.
Okay. Good morning, doctor. Good morning, sir. Good morning. Welcome, sir.
Is it? How can I open that?
Like that.
Sir, can you hear me, sir?
This is yours.
I can share that. When I say sharing. Barnali.
Good morning, sir.
Hi, good morning, Barnali. How are you?
Sir, doing good.
I'm in Guwahati.
Ah.
From here, I'll be talking. You know Dr. Subhash?
Hello, I'm.
Hello, Dr. Subhash.
I'm here in AIMS Guwahati. I'm heading the biochemistry department. So, sir has been very kind enough to visit here and give us a lecture. We are organizing a CME here.
Oh, congratulations.
You must involve Dr. Barnali. She's an excellent worker, speaker, very hardworking, young person.
Very good. Good to see you, ma'am. Good to meet
She is
you.
our future.
Thank you, sir.
You will need to share a screen.
When you say share screen, should come. Oh, okay, it's here. That's okay.
So you have to do this.
Double click on that, it will come.
You have to share this also. Your presentation is here.
Okay, we'll bring it up. You'll stop.
Sir, can you hear me, sir?
I can hear you.
Yeah. So, sir, we are just starting in 5 minutes. Extremely sorry for the slight delay. We are just waiting for a few people to join in.
We are late by approximately 10-15 minutes. The first speaker is informed. And May I request Team Mindray to please get all the delegates inside? May I please request everyone to be in the hall, please? Kaha hai sab?
Our speaker and mic to this one. No, it is there. I'm closing.
Doctors kaha hai?
Presentation is
Finish it.
okay, sir. So do you need any assistance here? I know
You are always there, I know.
I think I can tell someone to sit here. Yeah?
I'm quite familiar.
But it's you have to say, sir. I have already your presentation is already. You have to go. Yeah, click this one. Click this one, and then you go see that.
Good morning, ladies and gentlemen, distinguished guests, esteemed speakers, and participants. Apologies for the delay. There are some delegates still arriving. My team is at the airport and they will be arriving shortly. But time is time and we have to start. So otherwise there will be a delay the whole day. So let's start. Welcome to a momentous occasion, the inaugural Scientific Forum for CLIA and Chemistry 2023, hosted by Mindray in collaboration with the International Federation of Clinical Chemistry and Laboratory Medicine, IFCC, Consortium of Accredited Healthcare Organizations, CAHO, and the Association of Clinical Chemistry Lab Medicine Practitioners, ACCLMP. I am Kshama, the Marketing Manager IVD for Mindray, and I'm truly honored to be your host for this groundbreaking event. Today marks the beginning of a new era as we gather to explore the frontiers of science with a special focus on immunoassay and chemistry, all made possible by the gracious presence of eminent stalwarts of the IVD industry. We witness the power of collaboration in industry leaders, professionals, and experts from IFCC, CAHO, and ACCLMP and Mindray come together to explore the frontiers of science in immunoassay and chemistry. Before we proceed, let's extend our sincere gratitude to our esteemed collaborators, that's IFCC, CAHO, and ACCLMP. Their commitment to advancing scientific knowledge and fostering innovation has paved the way for this forum. To kickstart our forum, the lamp lighting ceremony is a time-honored tradition that signifies the pursuit of knowledge, the triumph of light over darkness. Today, we are privileged to have with us some truly remarkable individuals who have made significant contributions in their respective fields. So may I extend a warm invitation to our esteemed guests to come up on stage for the lamp lighting. May I request Dr. Aparna Jairam, Dr. Manisha Patwardhan, Dr. Vinita Kothari, Mr. Sudeep Mukherjee, Dr. Vipul Doshi to please come on stage and do the honors, please. With the lamp now lit, let us proceed with the rest of the program with renewed inspiration and a shared commitment to the pursuit of knowledge. Thank you, our honorable guests. Thank you very much. May I call on stage Mr. Sudeep Mukherjee, Deputy Director, Mindray India, to officially welcome our esteemed guests for this two-day academic feast.
So good morning, good afternoon, doctors, all participants, and the entire healthcare fraternity. I'm truly delighted and honored to stand before you today on the dais as we kick off this extraordinary mega event, SFCC, Scientific Forum for Chemistry and CLIA. On behalf of Mindray, I extend a very, very warm welcome to each and every one of you. Well, I'm Sudeep Mukherjee, Deputy Director of Mindray India, and I'm privileged to witness the incredible talent and dedication that our team exhibits every year. Mindray India feels proud to host this event in association with IFCC, CAHO, and ACCLMP. This is the first year that we are hosting this event in our country, and I convey my sincere gratitude to Dr. Venkatesh Thukpil from CAHO, Dr. Aparna Jairam from CAHO, Dr. Banali Das from IFCC, and Dr. Sushrita Sen from ACCLMP, under whose guidance and leadership this entire event has been organized. We are very much pleased to welcome and thank all the audience and our respected guests who are present here in this occasion to bless us. Mindray has always stood forward in the healthcare innovation. We are dedicated in advancing healthcare for humanity. Our commitment to quality innovation, patient-centric approach, and the user-centric approach has actually propelled us forward in this industry. We are now one of the global leaders in providing medical devices and solutions to the world. Mindray has its presence now more than 190 countries and with more than 300 products, 300 plus type of products. We are always committed in providing advanced technology, transfer them into innovation, and then make it available for the larger part of the world always. I'm proud to say that Mindray has acquired leadership positions in all their segments. That is, one is the patient monitoring and life sciences. We hold leadership position in that, in the ultrasound, as well as in IVD, where hematology we stand to be number 2, and chemistry we stands to be number 4 in the industry. Mindray India started its operation in 2007, and over the period of about 15, 16 years, we have excelled as a market leader and established our presence in the healthcare industry as a provider of medical devices and continuously serving the healthcare industry. Mindray is well known among the scientific community for their hematology products. As we have acquired number 2 position globally, at the same time, many of us are not aware that Mindray has been also recognized as a very, very strong player in chemistry and immunology in the world market. We offer the products which suits the every segment of the market and every pocket. by maintaining the quality to the extreme standards. We are not known only as a manufacturer and marketeers for the medical devices, but we are also known for imparting knowledge to the fraternity through various CMEs, seminars, and scientific events. One of them is SFCC, the Scientific Forum for CLIA and Chemistry, which is a true academic scientific event wherein omniscience pathologists from all over the nation participates to enlighten us with your knowledge. During this two-day mega event, various scientific studies are going to be discussed to aid everybody with profound knowledge of the eminent speakers. I'm truly excited to hear all the lectures from the eminent speakers here. I sincerely urge everyone to hold back for these 2 days feast, scientific feast, and gain as much as knowledge, which will further help everybody in day-to-day practice. Thank you once again for being part of this. And once again, I extend a very, very warm welcome to each one of you. Thank you so much.
Thank you, Mr. Sudeep Mukherjee. Now let's take a moment to preview the exciting agenda that lies ahead. We have meticulously curated sessions that delve into the latest developments in CLIA and chemistry. Our goal is to foster collaboration, share knowledge, and set the stage for future breakthroughs. We have a fantastic lineup of speakers, researchers, and experts in the field of immunoassay and chemistry. Our aim is to provide you a platform of exchange of ideas, engage in thought-provoking discussions, and explore opportunities for future collaborations. Without further ado, let us kickstart the forum and welcome our first speaker for the day. To do the honors, may I request Dr. P.K. Mohanty to chair the session of Dr. Venkatesh Thuppil, please.
Very good morning, rather good afternoon to all of you assembled here. It's my proud privilege to introduce such a stalwart of our country in quality journey, and his name itself speaks, is Dr. Thuppili Venkatesh, and who has pioneered the quality control journey in the country, and not only as a professor and teacher of biochemistry, but at the same time, still now he is going on with the organizing various quality control programs all over the country, mostly in South India, and as it is there, he was the Professor Emeritus of Biochemistry in St. John's Medical College, Bangalore, and as it is Quality Guru of Diagnostics, the Chief Architect of International Lead Assessors Training Program, and he has brought unleaded petrol to India during the year 2000 and has innovated several laboratory technology which are currently used across the globe, and he has delivered 8 special modules on behalf of IAS to the medical laboratory staff, and many of his students have started training centers in the India and abroad. Dr. Venkatesh has been instrumental developing a lead assessors course for NABL, ARSSC, PAO, and AMLA. He is founder and chairman of many national and international organizations. He has over 350 scientific papers and has contributed to many textbooks. Dr. Thuppili is the first Asian to receive the International Science, Truth and Honor Award in 2010, which is awarded 1 in 2 years, and is a recipient of Arjyavat Award, recipient of Avades Saran Memorial Award, Seth GS Medical College Award, Thakur Memorial Award, CAHO Lifetime Achievement Award, and Lifetime Achievement Award, to name few. So I welcome Dr. Thupili Venkatesh on stage and enlighten us regarding the present journey of quality in the scenario of CLIA and chemistry in medical laboratories.
Sir, 1 second, sir. We cannot hear you. 1 second. Okay. Yeah, sir. Can you hear us?
Yes.
Dr. Kapil, sir, we cannot hear you.
Oh, Dr. Thupili Venkatesh, please, you are welcome, and you can start your session.
Sir, we can't hear you. I think you are muted.
No, sir, we cannot hear you. Your voice is not coming. No, sir, we cannot hear you.
Okay, once again.
Yeah. Yes, sir, now we can hear you. Yeah.
Am I audible now?
Yes, sir. Thank you.
Okay. If I am audible, please, Barnali.
Yes, sir. Yes, sir.
Okay, thank you very much. Sorry for this technical hitch. Okay, first let me convey my greetings to all of you in the capacity as one of the patrons of CAHO. Wonderful program and wonderful arrangement, wonderful speakers. I am from Bangalore. Unfortunately, I am not in Bangalore. I was in Delhi yesterday. Today, I'm in All India Institute of Medical Sciences, Guwahati, from where I am addressing all of you. I am given a very, very interesting, challenging portion about which every one of you are aware. This is the pre-examination, post-examination errors. Now, I would like to tell you some difference between the non-conformance, non-conformity, error, and risk before I start. Anyway, are you able to see my presentation? Okay. Barnali, are you able to see the presentation projected?
Yes, sir. Yes, sir.
Now, laboratory errors. Why it happens? It is error is to human, and I would like to quickly take you through some few statements. The error, why this error happens in the laboratory. 1 second. Many mistakes in the examination process are called laboratory errors. In all activities, all procedures, processes, because a human, although these may be due to poor communication, action taken by others involved in the examination process, including the physicians. It is not only laboratory people, even the physicians, the nurses, phlebotomists are involved in this. We think the laboratory people, all these are beyond our control. It is not so. In fact, a non-compliance, non-conformity, error, they have different meanings. You see, what is a non-conformity? What is a non-conformance? Non-conformance is yet to happen. It has not happened. Something which we are supposed to do, we are not doing. That causes an error. And potential source of error. Something which has what we should have done, we have not done, it becomes a non-conformity. That also is a evidence of an error. And what is the error actually, the mistake, or is because what we are not supposed to do, we are doing. That results in confirmed risk error. Therefore, before the error happens, if we get an idea then it becomes a risk we can manage. This is, with this small background, I tell you, if you go to ISO 15189/2022, there are some 8 clauses. In the 7th clause, which is the process requirement, there are 3 processes or 5. Actually, there are 7.1 to 7.8. There are about 7 processes. In that, 7.2 is pre-examination processes. In that pre-examination processes, there could be error anywhere, which is avoidable, 100% preventable. But there are 7 places in the pre-examination process where error can happen. They are the laboratory with relation to laboratory information for patients and users, or maybe request for providing laboratory information. Error can happen at the level of primary sample collection and handling. Error can happen at sample transportation. Error can happen at sample receipt. Now, when you see what I have seen when many people present, this is a slide I see, but I want to read in between lines. Now look at missing samples. This is quite common. Why it happens is because of lack of training, pressure, laboratory work pressure, many things. Sometimes the examination request itself will not have sufficient information, which leads to some kind of error which is introduced. Wrong identification of patients, not sample, patient himself, wrong identification, for surgery. Instead of right eye, left eye was operated. Instead of subject A, subject B was operated. Similarly, in the laboratory, wrong identification of patients because of common name and gender, etc. Wrong identification of samples. Samples are okay, right from the, but wrong identification. Contamination with infusion routes. This is this happens in the inpatients when they are in the ward, when some infusion is going on. Hemolyzed samples, clotted samples, insufficient samples. These are common things we see, and this also causes an error. Inappropriate container. I tell you, there are shortcuts adopted for the cost cutting. They prepare their own container. They are not properly prepared. No proper inappropriate blend anticoagulant ratio. I have seen people, to save money, they draw sample from disposable syringe and a needle and transfer it into vacuum tube where have some amount of anticoagulants. But the volume is not maintained. Inappropriate, this ratio, blood and anticoagulant ratio can cause error. Inappropriate transport. I tell you, I had to transport my son's sample to US for one test. And I don't know how safe it is, how environmental conditions are maintained, and inappropriate storage. This again because of lack of calibration, lack of training, concern. When everything is driven by money, finance, some of these things will get compromised. And procedure outside the lab thinks this is not our role. We have no control over that. These are all some of the pre-examination errors. And there is a point of care testing now that 22870 standard is merged with our ISO 15189/2022. In this case, what is happening? Lab thinks, we have no control as non-laboratory staff carry out this examination at the bedside. It may be some nurse or maybe somebody, some staff paramedical doing this, but still there is a requirement as per the standard in Annex A, Annex 1, that there should be multidisciplinary committee to avoid this error, which is related to point of care testing. Now, this is a mandatory requirement. Accreditation following standard may not be mandatory, but once we go in for adopting the standard for the purpose of third-party recognition, I tell you, all of us know that we have to have a control over point of care testing when there is a point of care monitoring. Earlier, point of care testing was a wave test. It is no longer wave test. We have to participate in external quality assessment schemes also, even for point of care testing. And here, one of the major issues I have come across, I have seen, when the IVD manufacturer, when they bring some change in their system or in their kits or reagents, when it is communicated to the laboratory which is using point of care testing, which is distributed to various clinical settings, it doesn't percolate. It doesn't get really, followed. As a result, there is an error here. The various types of error, the environmental error. the environmental errors are miscalculation that results from change in the environmental condition. Therefore, when we talk about environmental, it comes as one of the resources. Out of 8 resources, environment is one of the most important resources. If it is neglected, if it is not monitored, if it is not controlled, if it is not recorded properly, it is not reviewed, then there is an error. There is procedural error. No, we know that all the procedures, laboratory, in whichever section, whether pathology or molecular biology or biochemistry, all the procedures need to be accredited, validated procedure. And these validated procedures should be verified by the respective sections of the persons who are using. Then this is another one of the most common error. The causes of error in laboratory is the procedural error. This procedural error happens because of modification, the procedure without validating it, without verifying it. The procedures are borrowed from somewhere which is not checked. The procedures authenticity is also not established, especially in the molecular diagnostic. Throughout the COVID period, I tell you, left and right, every laboratory, whether they were trained or not, or equipped or properly or not, or whether supervised, the COVID testing went on and on. You know, international bodies said the data from India is questionable. That's why they said they could come out with certain errors in some of the setup. Human error, it is unintentional. There's a human error is another which causes the error, and instrumental error where instruments are used because of the instrument condition, instrument maintenance, instrument calibration, instrument, they are calibrated for the purpose of calibration certificate we produce to the assessors during assessment, not to use that K factor during the calculation. That also causes an error. Now, how do we prevent pre-examination errors? There are a comprehensive plan to prevent pre-examination error. Means these are proactive and has 5 interrelated steps. One is developing clear written procedure. I always say you must have a documentation. That documentation is reliable. That documentation should be reviewed from time to time because the technology is changing very fast. Developing clear written procedures is one of the important tools to prevent pre-examination error. Enhancing healthcare professional training. It's not only laboratory people who are involved outside the laboratory, who are collecting samples, who are transporting samples, who are preserving samples, who are separating samples. These are the people. This kind of training is also very essential. The third important prevention tool is automating functions. Of course, the laboratories are going into robotics, artificial intelligence to reduce. I'll tell you the percentage of errors we come across in this pre-examination phase, both for support operations and for executive operations. Here, the IVD industry, like Mindray, is playing a very big role. They are a great support for all the laboratories to reduce the error. I tell you, if technology or the IVD is one side of the coin, the other side of the coin is experts in the laboratory, the knowledgeable persons with competence, and monitoring quality indicators. If it's properly done, then number of errors can be brought down. This quality indicator is a key performance indicator, and also the error which is going to happen can be prevented to a large extent. And also, apart from this, the fifth one is improving communication among healthcare professionals and fostering interdepartmental cooperation. There are a lot of references on this. Lack of time, I will not be going into the details. And this communication could be verbal communication sometimes is not heard properly or forgotten. Therefore, right type of communication. Today we are going in for communication error. Why did we change from 2012 to 2022? Version is one of the main reasons is nano information technology. Technology is also fast changing. Artificial intelligence is getting into this. And this is one of the prevention of pre-examination errors. When you look at pre-examination, inappropriate test requests, order entry errors, misidentification of patients, container inappropriation, sample collection and transport inadequate sample, anticoagulant ratio, whatever I told you, leads to somewhere up to 68.2%. This is published in many international journals, 46 to 68%. This is in accredited laboratories which are certified by third party. Forget about the other laboratories. I'm not commenting on that. Accountable laboratories. These are all, legally recognized, ethically following all the system. There, the error goes up to that. However, in the examination area, because of automation and because of the system, which are all highly validated, the error is only 7% to 13%. When you come to the post-examination phase, the error, there are only one important thing in post-examination phase, that is the reporting of results. Therefore, failure in reporting, errors, validation of an analyte, erroneous validation. You see, laboratory manufacturers or producers the patient result. That is the only thing which is supplied as a service to the users, clinicians, and therefore that has to be validated. Like your industry validates what they provide you, but it has to be verified by you. Similarly, what we generate, what we manufacture, what we produce, the service we provide need to be validated. That's where the IQC and EQ, all this comes into picture. That comes in the examination phase. However, in the post-examination phase, these are the two: failure in reporting and erroneous validation of analyte leads to somewhere between 18.5 to 47%. Look at the error in the pre-exam and post-exam. I don't know why the organizers wanted me to talk on pre-exam and post-exam only. Yes, because they know very well the percentage of error contribution in the examination phase is only 7% to 13%. This is coming down with full automation these days. The post-examination errors, with a limited time given to me, I am trying to consolidate. The common post-examination error include failure to report test results. Results are there, some reason are there, maybe justifications are there. Failure to report test results. Now, root cause has to be found out. Unless we do that, it cannot be corrected. Delay in reporting. I tell you, this has caused a lot of problem to the clinician treating the patient. I was waiting for some reports I'm yet to get, but I don't know what to do. Suffering goes on with the patient. Delay in reporting and incorrect calculation. This is another major issue, because today some laboratories, I'm told, they get about 20,000 samples a day. And imagine, with a limited staff, in spite of very high-end computer technology, 20,000 samples, documentation and verification is a big challenge. We always depend upon the technology, and technology can fail, misguide us. That is incorrect calculation. Critical results not reported are maybe delayed and results sent to the wrong patient. This is, one is, result is correct. Everything is fine, but it has gone to some other patient. The patient who is supposed to get it is not getting it. Causes of post-examination error in general. Such post-examination error, I'm generalizing it to all the departments is applicable. I may be from biochemistry background. I have forgotten my biochemistry. Forget about that. But this is general to all the sections. For example, entry. At entry level, are we checking? Is it possible to check? Even computer system, do we have a system, inbuilt system to check the entry? Like firmwares, are we using it? Manipulation. This is something which is unacceptable, which happens. I am not saying it's you are doing or I am doing it. We have come across this happening. Reporting of test data. This is where the post-examination error occurs. If results are written by hand or entered by a keyboard, then an entry error may occur. Errors in data transmission, including relying on hearing verbal information incorrectly, and this, unless it is repeated, is the cause of post-examination error. And how do you reduce post-exam error? Suggest an appropriate response. For example, suggesting further investigation, not necessarily a laboratory examination, or referral. Promote best practices. Now, today I was giving a talk an hour ago on good laboratory practices. I said it should be changed to good life practices, not laboratory practice. Life practice, wherever we are, it should become a habit. They promote best practices in day-to-day life. That helps to a large extent. Promote clinical action. Avoid misinterpretation. These are the ways to reduce post-examination errors. In this post-examination processes include the alignment of the raw sequence data using a reference human genome followed by a variant identification with corresponding clinical interpretation, clinical decision work. Externally, quality assurance program for the post-exam fields. have to be introduced. This is a mandatory requirement recommended. We are now we have married to CLSI from India. We get all their documents at the 1/10 the price and membership 1/10 the price, and there are documents pertaining to the errors in the laboratory errors. Maybe you can avail those facilities. Here is human building a safer health system. It is there 1999, way back. The 8th leading cause of death is 44,000 deaths per year. And here is human building a safer health system. The Washington DC National Academy Press in 2000. Please go through that. We get a lot of information. I have given a general, introduction to this whole workshop today. Thanks to Mindray for taking up this. I tell you, we are all responsible. We cannot shirk our responsibility. We have to be very, very cautious, meticulous. Then only we can, be proud of ourselves that we are part of healthcare system. This is my personal, last view. And I am in the evenings of life. Most of you in the mornings of life. If I am the setting sun, you are all the rising sun. You have a lot of time, greater opportunity to avoid or correct the mistakes we made, our generation. Friends, I wish all of you all the best. Thanks to our Jairam, Barnali, our other colleagues who have invited me at this age. And I wish I was there with you in my hometown, but unfortunately, I'm missing all of you. I wish you all the best. Thank you very much.
Thank you very much, sir. It's a nice, vivid account of almost minute by minute account of the flow of work in the laboratory. And of course, you have raised a lot of issues. And maybe I feel most of the participants should be having some of their queries in their mind. And I would request, sir, to enlighten if our participants having any query both on site as well as online. And I request the participants to raise any queries if you want to hear from sir about any account of it. Welcome, participants.
Good afternoon, Dr. Jayalakshmi.
So, are there any questions for Dr. Thuppil?
Can I ask one question, sir?
Please go ahead.
Thank you, sir. Thank you for this excellent talk. Your quality journey is so inspiring. So my question is regarding POCT, like you mentioned in the slide, because now POCT is POCT is a big thing, and we always see during our hospital quality journey that we follow for our analytical instruments, everything, verifications and everything, quality control, IQC, QA. But most of the lab, in POCT, we find a lot of challenges, from glucometer to the POCT instrument, in from ABG to everything used in the hospital. So, sir, how to how to follow the checklist and how to do
Okay.
insight.
Very appropriate question, Barnali. I really like this question. You see, there are two standards which laboratory must keep with them. One is the International Organization for ISO Technical Standard 22583. If I remember correctly, I don't know, my memory may not be right. Technical Standard, ISO Technical Standard 22583 provides guidance for non-laboratory supported services. Okay. POCT is also one of them. It's a non-laboratory supported service by others. ISO 15190. Please remember, again, it's a medical laboratory standard, which is for medical laboratory environment and safety. And ISO 22367, that's another standard, provides guidance on safety and risk aspects of POCT. We are talking about risk, error leads. Therefore, these three standards, ISO TS 22583, ISO 15190, ISO 22367, if these three standards are understood by POCT committee. The POCT committee is the committee wherein you have laboratory representative. We have representatives from the clinicians, nurses, management, because they share the resources, financial resources also. Therefore, service agreement between laboratory and all locations using laboratory supported POCT shall ensure that respective responsibility authorities are specified, documented, communicated within the organization. This is number one. This will solve most of the problem. Apart from this, these agreements shall have clinical approval, like your ethical approval, clinical approval, and where applicable, financial approval, because the finances are required. And also we should remember the service agreement shall be with POCT area and may be managed by a health professional grouping like medical advisory committee. This is another important requirement. And finally, I would like to tell you, quality assurance program is the backbone of POCT and training program. A person with appropriate training. Who is going to train? The trainer. The person with appropriate training and experience shall be appointed to manage training. That is very important. Manage training and competency assessment of personnel performing POCT. If that is done, Dr. Barnali, I tell you, the error will be very, very limited or minimal. And finally, the trainer shall, like you and me, we should develop, implement, maintain as appropriate both theoretical, practical training program for all POCT personnel, where we should involve the industry also.
Thank you, sir. Thank you.
That's your question.
Absolutely. Thank you, sir.
Any other questions from the audience, please, for Dr. Thuppil?
I'm extremely sorry. I apologize. I am missing all of you in physical presence. Pardon me for that.
So my question related to the POCT, glucometer user. How you train the people who use the glucometer at home and how they manage the quality for the glucometer?
Okay. I will give you an example. In the Cleveland Clinic in Abu Dhabi, they have 200 and plus glucometers. There is one dedicated officer put in charge of only glucometers. His job, he has a small room. Every day he gets some 10 glucometers, validates, verifies, sends it, and brings some people, train them. There should be a dedicated the point of test managing person. That person is the key person. This is requirement. Future, I tell you, the whole laboratory is going to become POCT. Next 10 years to come, most of the examinations conducted will be through POCT. Take it from me. All these big machines will become smaller and smaller to a matchbox size.
Good afternoon, sir.
Yes, good afternoon.
See, you are talking about the pre-analytical and post-analytical errors.
Pre-exam and post-exam. We don't use the word analytical now.
So, while we are monitoring quality, mostly our systems are for analytical quality, whether the IQC or IQA. So, while you are moving through your evenings, do you have an idea in mind how to monitor this pre-analytical and post-analytical errors?
Please repeat. George, I could not get it clearly.
See, most of our systems are just for analytical quality monitoring, whether it is an IQC or IQA. So this over the years, we did not find any system for monitoring the pre-analytical or post-analytical errors. So do you have, after as long as you are moving to your evenings, so can you have any suggestion for us to monitor those
my suggestion is only to understand this much clearer is to follow this 15189/2022. That gives you complete solution for a laboratory management to even look into the errors in any aspect of laboratory activity. Therefore, my strong suggestion is, before regulators come, let us regulate ourselves. Before somebody comes and asks, are you accredited? Are you going in? Let us go in for accreditation.
Thank you, Dr. Thuppil. I feel now there are no more questions from the audience too. So may I request Mr. Sudeep Mukherjee to please hand over the certificates to our chairperson and our honorable speaker. May I please request Dr. Mohanty to... Dr. Mohanty. Dr. Thuppil?
Yeah, I'm here.
Yes, sir. Sir, your certificate.
Okay, okay,
We'll
okay.
show it to you.
Thank you. Thank you. Thank you very much.
Thank you so much, sir, for gracing this event with your presence. We are extremely honored. So moving ahead for the day, may I request Dr. Manglesh Srivastava to chair the next two sessions. May I call Ms. Dr. Manisha Patwardhan on stage for our next talk, please.
Welcome, August gathering here. Welcome, Dr. Manisha Patwardhan. She is Head of Quality, AG Diagnostic Private Limited. She is MD Pathology and DPB in 1995, SAIT GS Medical College and KEM Hospital, Mumbai. Topic allotted to her is a Six Sigma. Definitely it is a tool for lab excellence. And she has got enough experience to tell us about this Six Sigma. She has a long experience in the field of quality and accreditation. She is a technical assessor as well with NABL and has pioneered the start of quality related teaching programs across the country. Dr. Patwardhan is adept at handling all aspects of lab management, in-depth technical knowledge, day-to-day streamlining operations, training, recruitments, and liaison with multiple agencies in her foot. She is a frequent and well-appreciated speaker at various seminars, conferences, and institutions at state as well as national level. Welcome, Dr. Manisha.
Thank you so much, sir. Hello, everyone. Preferably, yeah. Hello. So good afternoon, everyone. And it's a great pleasure to be here with all of you today. Thanks to Mindray. The topic I'm going to discuss today is Six Sigma, which is to be used as a tool for laboratory excellence. Now, in the past, when we thought about our commitment or pledge to quality or to do well unto the patient, I will refer to the Hippocrates Oath, which most of us have taken as doctors. As doctors, put on this mic, please. That is not comfortable. Hello? So as doctors, we have pledged to do the best according to our ability and judgment to keep patients away from harm and injustice. And for years together, actually, healthcare has depended upon the innate excellence of clinicians, physicians, doctors, their individual expertise, which cannot be really defined or measured, and their actions in good faith. So all this is what has driven quality in healthcare and excellence in healthcare for years together. Now coming to today, and laboratories in particular, I'll be speaking about the laboratories obviously. So today's laboratory looks something like this, with a lot of people involved. There are phlebotomists, there are technical staff, there are your lab managers who are looking continuously at your quality controls, your operations, your costs, your LIMS. So the working of the laboratory today is an integrated, it's a lot of many integrated processes involving personnel and information technology. Right from registration to reception, preparation of the samples, work allocations, trainings, technician allocations to departments, their shifts, then validations, method validations, IQ, PQs, approvals, report approvals, then the report going, reaching the patient, the TAT, accreditation requirements, statutory requirements, logistics and transport, biomedical waste management, and LIMS. So there is so much to do in today's lab. So we cannot really depend, on only my ability, only a physician's ability, only a pathologist's ability as such. We will have to look at certain methods, strategies, and tools for quality management. So when we talk about laboratory excellence, what are we talking about? We want the laboratory to operate with accuracy, with speed, and with efficiency in order to deliver safe, reproducible, and patient-centric healthcare service. So broadly, there could be two components here, isn't it? So there's an analytical component, analytical excellence, which ensures accurate and reliable results. And you have operational excellence. So there was just a question a few minutes ago that how do we look at processes, especially the pre-examination and the post-examination processes, where we cannot apply the so-called traditional statistical QC measures. So operational excellence is where we have to optimize the processes to achieve maximum efficiency and productivity. Now, what are the challenges that healthcare in general and labs in particular face or have faced for some time? Well-established protocols and benchmarking in healthcare came much, much later as compared to the rest of the industries, and it is still a work in progress. So healthcare has had to draw from other industries, look at other industries. What are the other industries doing for managing processes well? At the same time, the complexity of disease and medicine and human body makes industrial grade quality a distant dream for us. We cannot be compared to a precision tool manufacturing company or a fan making company at all. Despite that, I would say, in fact, amongst the healthcare sector, labs have been more proactive from very early on in using QC, because we've always been running controls right as far as I remember 30 years ago, also applying statistical QC. I have started off with actually plotting the LJ charts on a graph paper. So we've been applying statistical QC for a long time, histograms, scattergrams of different types. Then there is lean management, there is the Kaizens, there are cost-benefit analysis. The ISO standards help us to benchmark and follow quality. Training and retraining and training of staff, quality circles. So there are so many types of tools that we can use for quality management. Today's topic is Six Sigma. So we will discuss Six Sigma in detail. This is a concept which was introduced by the industry, General Electric and Motorola companies. It has been adopted by both the manufacturing and service industries, as well as now, of course, the healthcare institutions from hospitals to reference laboratories. It is a management strategy that seeks to improve the quality of process outputs by identifying and removing the causes of defects, errors, and minimizing variability in processes. So Sigma metrics allows comparison of different processes with each other across different institutions and different industries. So this is the interesting point of it, that the other ones are more subjective, the quality tools which I pointed out earlier, or then statistical QC. You cannot be on a coffee table discussion and say that the CV% of my cholesterol assay is so and so to other people. But when you talk in the terms of Sigma, it becomes a universal language because it is recognized across the globe and across the industries. And on a scale of 0 to 6, 3 is the minimum Sigma that is required for any business or manufacturing process. It also measures or can give you an idea about the variation in a process, because every process, you will never expect that every time that that process happens or is carried out, it will be exactly the same. So there will be variation which is inherent in these processes, and that creates an opportunity for errors to happen. They could be due to the common causes, the variations, or special causes. Now the goal of the Six Sigma strategy is to eliminate or reduce all variations in a process so that the outcome is as best as can be every time. Now, how can we apply it to the lab? So when you're measuring outcomes or defects, you inspect outcomes and count the defects, calculate the defects per million, and convert the DPM to a Sigma metric using the standard Sigma table. Now, this can be applied to operational processes. So this can be applied to all your pre-examination and post-examination processes. On the other hand, if you measure variation, like how we have always done traditionally, so the bias, the imprecision CV%, and the total error, then you can calculate Sigma from a simple formula, which I will show. And this can be applied to analytical processes. So this seems to be the beauty of the Six Sigma tool or the Six Sigma strategy, where you will be able to apply the same quality management strategy to both your operational processes as well as the analytical processes. So suppose we take, now Sir Dr. Thupilu Venkatesh just spoke about the pre-examination errors, and right on the top was misidentification of patient or patient registrations in the demographic error in the patient registration. So here is an example. Units or registrations are 2,74,257, and there were 93 defects in this process. Now I have said opportunities 3. So what are opportunities? Opportunities is what can go wrong in a demographic while doing a demographic registration. So either the patient name can go wrong, the date of birth can go wrong, the gender can go wrong, the mobile number can go wrong, the Aadhaar card number can go wrong, so on and so forth. So there will be so many opportunities for error to occur in a registration process. So with the standard Sigma calculations, you will have, So with the standard, so all these formulas, there's no need to be bogged down by these. These are very easily available. The calculators are very easily available on the internet, and you can just go to any one of these or add in a QI macros onto your Microsoft Excel. So all these calculations become very easy. So you have a defects per million opportunities, percent defects, percent yield, and a sigma value. Let us not go into what is short-term sigma and long-term sigma at the moment, because that will be confusing. Let us stick to the sigma value 5.19. So for this process, if there are 93 errors or defects of any of these kinds, then the process sigma is 5.19, which is a acceptable process sigma. So I've just explained the calculation. If a laboratory reports 1000 ED stat results in a month, which is the product, 10% or 100 results are not reported out within the specified period of time, so TAT is missed. Defects. The number of opportunities for defect is 3 because the delay can occur either in the pre-analytical, analytical, and post-analytical phases. So defects per opportunity, defects per million opportunities, use the sigma table, and you have a 3.3 sigma for this particular process. The number of opportunities is sometimes difficult to understand. This is the simplest example, that if you have ordered 100 shirts and 3 of them are defective, then 1 could be wrong size, another one could be of the wrong style, and the third one could be of the wrong color. So every process that you will have to define the process, what you are going to measure, what will be counted as the defect, and how many types of defects can occur. So say this registration errors I have finished, so let's go here. So citrate sample rejection, one of the important pre-examination processes or problems where we do not want underfilled or overfilled tubes. So here, for example, the sigma in our laboratory was more than 6 up to March. There seemed to be some issue in 3 months, and then it is back here. So this is the area at this time you need to intervene. You need to do certain corrective actions, and then it was back on track. We will discuss this later. So if I had a more than 6 sigma and I had a 4 sigma, then what do I really need to look at? So if the sigma is less than 3, I already said, most manufacturing service sectors everywhere, they consider a process which is operating at less than 3 sigma as unacceptable. You have to improve it, right? Here, in fact, it's a no-brainer because you will immediately find the most obvious problems in that process which can be corrected and the sigma improved. If the process is at 4 sigma, then the processes have to be improved. That is, each step has to be looked at very carefully. If you are at 4 sigma and you want to achieve 5 sigma, then you may have to look at the design of the process itself. If then you want to achieve 6 sigma, you will require very rigorous tools, a complete revamp of the design, and then work towards perfection. So while zero harm or zero error is a bold and worthy aspiration, the scientifically correct goal is a continual reduction. So you go from 3 sigma to 4 to 5 to 6 using the DMAIC process or strategy. So that is defining the problem, measuring the baseline performance, analyzing the data, then going to the root cause. For example, I showed you that in 3 months we had slipped on the citrate tube sigma count. So there we will have to see where the training has gone wrong, whether suddenly a new B2B or hospital has been added, where we have not given clear instructions. And then if we correct that, we can get the sigma correction. So improve and then control and ensure sustainability. So practicing the philosophy of continuous improvement, getting a little bit better every single day, you can always try to achieve 6 sigma. Now in analytical or examination phase, how can it help us? So evaluating manufacturers' performance claims, support in decision making when purchasing new analytical systems, ongoing assessment of method quality throughout the lifetime of an analytical system, And QC planning, selection of appropriate QC rules or procedures based on the sigma metric of the method. So the sigma metric calculation is a very simple formula. Sigma is equal to % total error allowable minus % bias upon % CV. An allowable total error is an expression of how much combined imprecision and inaccuracy can be tolerated in the test result without negatively impacting patient care. So again, very simple calculations. If your error total allowable error is 10, at L1 control your total precision or CV % is 1, and the bias that you have taken either from your peer group or from your equas is 3, then sigma is 10 minus 3 upon 1 is 7 upon 1 is equal to 7. So this is essentially a very simple calculation. Of course, like I said, it does depend upon where have you taken your bias from, whether you've taken it from a reference material or reference method, peer group mean, or proficiency testing, or any other source. Similarly, it will depend upon where have you taken your imprecision from, whether you've taken it from your historical imprecision, that is your 3 to 6 months QC data, or within run, between run imprecisions. It will also very heavily depend upon the total error allowable that you have chosen. So if you have chosen a minimum requirement for performance, then you will have a good sigma, 10 in cholesterol, creatinine 7, okay? For certain period, these are examples, so these are for a certain period in the laboratory over a certain number of controls, etc. And if you have chosen a desirable level of total error allowable, then the sigma is this and this for the same period and for the same level of control. So what does it mean? So it means that the laboratory will be able to define the quality that it wants to achieve, okay, can decide what is that the quality requirements for your lab, your routine diagnostic lab, your specialty lab, your clinical research lab, whatever that it might be, and then accordingly calculate the sigma. Now, what do you do if you have assays less than 3 sigma? So you will have to do a series of corrective actions. You will have to check a lot of things, your QC, reagent, calibrator stabilities, check for any outliers in the cumulative data, verify the manufacturer's claims, check the TEA, if the used TEA is appropriate, and the assay method or assay design, because some assays inherently will have a lower performance sigma. Now, if all your methods or analytes are showing less than 3 sigma, it might mean that the quality requirement that you are wanting to reach may not be practical. It might be too tight, so you can even loosen it depending upon the clinical outcomes. If you have an analyte less than 4, you can also, now that the new standard and the requirement for risk management is again something which we are now learning to incorporate into day-to-day working of our laboratories. Here is an example that if you have an analyte with sigma less than 4, you can use one of the commonly used Ishikawa's or fishbone analysis for investigations. So you can look at the same things which I said just now. So the differences in methods across industry, your calibration frequencies, what multi-rules are you applying, what error have you chosen, are your materials near expiry, is the storage not proper, whether there's good compliance to procedure, the theoretical and technical knowledge of your personnel. So all these causes which can lead to a lower sigma performance have to be looked at. Now, when your sigma is 3, 4 and you want to improve, you should also have a pointer towards what should you improve. Whether you want to improve whether you need to improve the accuracy or the imprecision. So for that, you can use a quality goal index, which is again a very simple formula, bias upon CV after reducing. And It will tell you the extent to which both bias and precision meet their respective quality goals. So if your QGI is less than 0.8, you have to work on the imprecision. If it's more than 1.2, you have to work on the accuracy. Now, what happens if there is a happy situation and the better end of the spectrum and your sigma is more than 6, right? So here the sigma metrics can give you a lot of cushion from day-to-day variations, lot-to-lot variations, personnel variations, etc. And you can choose a tighter quality requirement. You can design an individual quality control plan. You can choose the multi-rules to be applied and save on time and materials. Remember one thing, there's no such thing as 10, though I had myself put in a value of 10 because that is the value which was there. But it is basically more than 6 sigma. There's nothing great about having a 14 sigma or a 15 sigma. It's not like that. It's more than 6, less than 3. That is how you have to look at it. So if you have a very robust method which is performing at a good sigma, then you can use lesser number of QCs and you can use lesser number of multi-rules. And that will save your morning time and resource in the long run. So for example, if we had 3 or 4 multi-rules applied and about 4 to 5 outliers, because this particular assay was performing at more than 6 sigma, we reduced it to 1 rule and got only 2 outliers. So Westgard or other sources give you a good table of what is, if your sigma metric is 6 or 5, what rules should you apply and what should be the frequency. I had mentioned that we can use it for analyzer evaluation also. So either when you're going to go in for a new analyzer or the one which you already have, if you know how many of the assays on that analyzer are in a good sigma level and how many need more attention, okay? So that will also help you. One is, everybody is aware then that, okay, these assays might have slight problems and we need to look at them more carefully. And if you're buying a new analyzer, obviously you will try to go in for an analyzer which is going to give you most of the assay performance. Because it was lab excellence and I have been talking about processes and people have been asking about processes, you can then apply it to, so our purchase inventory person is applying it for, this is his terminology, goods return, the defective kits or materials that he finds. So in these many orders, he had 3 defective materials which he had to return, either because of the wrong lot, wrong quantity, or the wrong price, and the sigma level he derived is 5.3. So you can basically apply to whichever process you want. So this is called resolution. At a call center, we had, suppose we had 192,000 calls, 177 error. That was customer grievance. Customer was not happy with the call addressal. So either the calls were missed or there was inadequate information given. These are the opportunities which we have identified. And that particular sigma here was 4.8. So the point I'm trying to make is that there can be a huge number of candidate processes where you can apply the 6 sigma. Inappropriate test requests, delay in pickup or transport, incorrect fill levels, sample lost, I like the word misplaced, data transcription errors, notification of critical values, errors in billing, cytology specimen adequacies, so many. However, over the last 20 years, our journeys, it has started becoming that, we are more of statisticians or mathematicians rather than doctors. In fact, I took up medicine because I hated mathematics. So I do not like this, that all the while we are getting embroiled in these numbers. So what I would suggest is that you use these quantitative targets with caution and as applicable. They are our tools and not our masters. However, the eye does not see what the mind does not know. So many insights can be drawn from these exercises. So it's not as if even ours is a standalone lab. catering to about 1,500 to 2,000 patients on a daily basis. Like Dr. Venkatesh already said, everybody is overworked. Not that the purchase inventory person is going to actually sit and do the sigma metric every month and on and keep doing it and then keep doing it for each small process. No. But like I said, it does give you an insight. Firstly, they design the process. They start thinking about it. They get an insight from it, which really helps. So goal in the form of such targets have an important role, but should never displace the primary goal of patient care. So it is not just a sample or value, it is a patient. And I will still believe in my ability and judgment and expertise and experience to do no harm to the patient. Thank you for a patient hearing.
Excuse me. Thank you, Dr. Manisha Patwardhan. Because as you have already told that this is a very difficult issue for pathologists not being the mathematicians. Definitely you have explained the topic in a nice and meticulous manner. But still there may be some issues. So the topic is open for any discussion or questions or doubts, please.
Hello.
Hi, that was a very lucid presentation. Regarding the opportunities that you were talking in each type of defects, when we talk about the number of delayed reports or the number of repeat samples or anything in the pre-analytical phase, for all this, then the opportunities will be only 3, right?
No, that was an example. So what basically you will have to do when you decide to measure the sigma metric or apply the sigma metric to a process, you will have to jot down the process and you will have to put in the steps. So it could be anything. So it, because according to your process, you can define as many opportunities as you want.
Okay, so that
is so it is
subjective.
It is not subjective, as I would call it. If you actually break down the process in the proper modules, you will automatically reach those opportunities. So that will be individual for each of the different processes.
Thank you.
Any questions further?
Thanks, Dr. Manisha, for excellent presentation. Thank you.
As usual.
As usual.
She's
definitely.
master in Six Sigma and she's applied it in her laboratory. So, thank you, Dr. Manisha. May I call upon Mr. Sudeep Mukherjee? Yeah. Thank you, Mr. Sudeep Mukherjee. Thank you, Dr. Manisha Patwardhan. So moving ahead, we call our next speaker, who is now between our lunch and the session. But she'll make it very interactive and none of us will, the stomachs will growl. So I hope Dr. Aparna makes it possible. So I call Dr. Aparna Jairam on stage for the next talk. Dr. Manglesh, may I request you to chair the session, please?
welcome Dr. Aparna Jairam. She is MD Pathology, founder and director of Asavli Labs, first state-of-art diagnostic hub, which is also the first NABH certified path lab in Palghar district of Maharashtra. The topic allotted to her are the case studies in immunology with autoimmunity and infertility. She is an NABL assessor and honorable secretary of the CAHO Diagnostic Division and APPI Managing Committee member. She has also hosted the CAHO conferences at joint organizing as joint secretary in April 2019 in Mumbai, along with several national conferences in India throughout APPI. A lot we have to tell about her. That there is a long list. I invite Dr. Aparna Jairam for her topic, which can be very useful for our fellow present here. Thank you, ma'am.
So sorry, thank you so much for a very, very big introduction. I'm just amongst all of you. Only thing what I like is I like arranging events and doing more work after our lab work. So I think, thank you, Shama. Thank you, Mindray. Thank you, doctors Vijay Agarwal and Lalu Joseph. And my CAHO team here. Thank you, JJ. Thank you, Dr. Vinita. I can see, yes, thank you. And Dr. Barnali will be on digital thing. So thank you so much. So as usual, before starting this, I promise you that there will be only 4 cases. So nothing between lunch and like Shama said. You will have a lot of time to digest, ruminate, and then go back to have your lunch. And I thought Shama told me there is a best dressed award now. And that's why I'm looking so funny right now, but it's okay. I think I'll win that prize. The reason I am taking up this case studies before I begin with this is one short story so that you actually understand why this. I think all of us do this every day, and as my previous speaker, Dr. Tupil, talking after him and talking after Dr. Manisha, who spoke about Six Sigma, one thing which is common in both of them and what we are all dreaming and doing is pathology or diagnostics is not like a KFC or a pizza outlet where you can just talk about numbers. It is about patient, and finally, like she said, we care about patients, and that's why this. So when I passed out my MD way back in like 28 years back, so you can guess how old I am right now, I felt that, listen, I always wanted to be with my patients, and I felt, how do I do that? Because when those days in Grand Medical College, I was always between the files and the paraffin blocks. And that's when I took this leap and joined into the corporate sector. And I felt, yes, this is a place where I can talk to my clinicians. And without meeting or talking to my patients, I feel incomplete. And now in this new journey of CAHO and my own labs, I fight with my patients, I talk to them, and I'm so glad that I do consulting with my patients for infertility, which is my field of passion. So you may see some of the cases of autoimmunity and infertility. With that backdrop, so that you understand why this lady is doing this, I'll go ahead. So as when I talk to my friends in not in big cities, when I travel into smaller cities, say like a Riva or tier 3 city, they are like, अरे अपर्णा क्यों तूने लैब चालू किया, अभी टाइम है ही नहीं लैब चालू करने का. Everything is more about prices, which package of what price, and patients are also smart. They look at their apps and decide मुझे किधर जाना है, where should I go. So this is what it is full of. But apart from this, like people are saying, ये package कितने में, इसका rate क्या? I think same thing industry, diagnostic industry is also facing as we are growing every day. So how do we make a difference to us more than the patients? Because if we are not there, next 5 years, I have decided that this is my chart. I want to be present in maximum places where, most of the big labs may not have the direct processing centers. So when I'm doing that, I'm trying to create a team of people who will be as passionate or more passionate than me, who can dig into the records of the patient, both radiology, pathology, clinical history, bug the clinicians, get the history, call the patients and talk. But most of the times when we call the patients, I think some of them who have worked with me in different phases of life, I can see my dear friend Rajesh here. When we used to call the patient, okay, this is what it is there a history? Suppose there is a TSH those days coming of 100, and we used to say, TSH, आप कुछ गोली ले रहे हैं? Are you taking any medications? I think we used to get reply, even now, whatever you have got, you please give it. Are you not doing the test? I think most of us face it when we are asking. In spite of that, pursuing this journey, it's very exciting. So we saw one case where I have picked up just 4 cases highlighting clinical interpretation for all of us, where we see irregular menses, USG apparently not showing anything remarkable. Semen report apparent, I say apparently because most of the semen reports, I was lucky enough to have worked with one of a good andrologist, so they were not satisfactorily reported, but that's why I'm writing apparently. So on examination, this was a common complaint. The reason I'm saying examination is I do consulting for infertility PCOS cases in some cities like Nagpur, Jabalpur, or even in some of the places near Surat. So that's how the histories are given. So if you look at the investigations, she had carried these files. Obviously, what is important is AMH 0.59. These were the ranges which were mentioned onto the report. Testosterone, glyco-HB on the borderline, prolactin 26, so insulin random 6.8. So with this particular case, we looked at her repeat values of AMH. They were done twice before. Every time the AMH value was 0.7, somewhere 0.9. This particular report was 0.59. So she was labeled clinically as a case where she should be advised to do lifestyle changes, particularly polycystic ovarian case. But if you look at the hormonal reporting, her AMH, which is actually looking at the age, has a very, very low ovarian potential or fertility potential. Her values persistently over the last 4 or 3 reports which were done were very, very low compared to her age. So I wanted to bring this to the notice that everybody was focused only on though the sonography reports were not showing ovaries showing, the classical picture of polycystic ovaries where you see multiple eggs on the periphery, or even her FSH LH I have not mentioned because it was unremarkable. Otherwise you have the 1:2 ratio. What was important here was the AMH and the prolactin. I think prolactin, though in the reference range, does show variation. All of us have faced that in our labs. Patient coming one day, a patient coming next day from another lab, showing a diurnal, I'll not call it diurnal, but variations because there is pulsatile release of prolactin. This is the next case where, we saw, I'm just picking up some points of the case which are important. Where patient is not responding to treatment. I saw her in a place called Rewa, which is in Madhya Pradesh, where she was responding, not responding to the treatments, not able to conceive. USG showed bulky uterus. This is what we advised. The reason we advised to do tripool prolactin was because her prolactin in 2-3 places was different. It was once 18, second time 75. So we said, let's do a tripool. I think all of us know the importance of doing a tripool prolactin sample and her DHES. So I have mentioned uh, the tripool value of prolactin here, which was higher on the higher side. Insulin is also high. So I think most important in my setup or my previous experience, whenever we see a prolactin of a higher or little bit on the borderline, say anything about 20 in a female who is coming with a query history of infertility or AMH workup or a BOH workup or a query PCOS workup, we do mention, advice to do ideally a tripool sample of prolactin. And there are many factors. I have one more interesting case to show you about prolactin. So this case we labeled as PCOS. We did later on free testosterone, and that was also on the higher side. I don't want to go to the theory. Yeah. So the second thing which overlaps into the clinical practice when we are looking at nowadays younger females between the age group of 18 to 27, we do see a high incidence even in males for insulin resistance, and the great test for doing that is HOMA index, or all you have to do is insulin and glucose levels. That's a better indicator than many times. I had a presentation in Diabetic Association with Dr. Kovil in Mumbai. So there was a question to him that then why don't you do a glyco-HB? Why do you want to do HOMA index for an insulin resistant diabetes? So this is basically an early indicator even before glyco-HB as per the ADA guidelines. So now this particular parameter of HOMA index is picking up faster for diagnosing early insulin resistance. So this is a case where sugar fasting is 105, insulin is 6.5, and the cutoff is 1.9. So if you are closer to the cutoff, the faster changes. So you can, the only changes into this are lifestyle changes, and that gives lot of results. Basically in patients who are going to be pushed into that diabetic zone or insulin resistance zone, or patients who are really getting treated for infertility, because that is what I want to really stress upon, because that's what I love doing. So that is HOMA IR for you. Why not? This was a question which really excited me, because and I like the reply. He did state a paper from ADA where it says this is now an early indicator for early diabetes even before glyco-HB. All you need to do is a serum insulin and sugar. So nobody talks about secondary infertility when you are into private practice. Patients are moving, the files are bigger and thicker, more and more complications. But out of my experience, which I have done in the last 6 years, talking to the patients and doing consultings along with the good, leading gynacs, it was always thought that the secondary infertility is more female cause than the male cause. Primary, still we all have opened our eyes and we are doing still equal attention to male and female. But out of this, it is equally shared between male and female. And we have seen mostly, this is obviously a case where we are seeing, if you see, 3 important causes in secondary infertility. One is male cause, that is sperm DNA fragmentation. So I have got that case. And the second is autoimmune. And the third and the most commonest, I think, is diabetes, because that also type 1 autoimmune diabetes. These are the leading 3 conditions for secondary infertility, apart from what we call as lupus, cardiolipins, because they all can be clubbed together into bad obstetric history or BOH. So this is a case of autoimmune condition where we saw that, if you can see it on your screen, that the TSH is on the border, ANA-IF shows weekly positive pattern. This was the antithyroid antibodies, when this is only after discussion with the clinicians. is what we get. ATABs are higher, prolactin again on the border. This is just a chart where we are talking about thyroid with auto antibodies or antibodies, particularly for female patients who are, you will see this very classically into secondary infertility, where you see TSH on the border and ATABs and ANAs positive if done by immunofluorescence, most important, the gold standard method. So that was a case of autoimmune where we could actually do corrections. The last case before, yeah, I have 7 minutes. So I can take up this case. A 33-year-old female, one healthy child, 5 years, now unable to conceive. Again, I'm covering secondary infertility. No history of abortions. You can see semen analysis okay, AMH 0.9. 3 months back, AMH 1.78. I wanted to point out this is Dr. Ritu Johri from a place in Gondia, which is not remote but pretty remote. If you want to travel to her clinic, it's like 3 hours from Nagpur. She was completely haggard. She said, Aparna, I have heard that AMH is like a fixed deposit. You are supposed to do it once in a year. All of us, or when we actually started AMH way back in 2014, I remember we used to present it as, even today, it is a first marker. Better than E2, you can do it once in a year. It will not change, but there are many factors which can change AMH. So I wanted to get this case to your notice because in our busy schedule, most of the times, I think as we become busy, we age, we are very impatient to read the finer lines of the reports of the pack inserts. Though all of us know OC pills can cause severe fluctuations in AMH. The second thing what we have seen is endometriosis. So if a case is seen with a high borderline CA125, which is a marker also for ovarian cancer, but early endometriosis, we do see a change in AMH. So that should not be judged on you as a good lab or a bad lab. Okay, it is a clinical condition. So I feel here footnotes under the reports are going to be very, very important, that what are the factors which can change apart from the clinical pack inserts which are available. These are our own day-to-day learnings which can help us and the patients at large. So that's why I wanted to get this case. So that's a theory. How does this work? I'll go to the male infertility. Yeah, I'm sorry. So this is a case which happened just last week in our lab. A very high profile clinician. We had a patient coming to our lab. This was her test. Okay. As a protocol, we did call the patient. We said, "Aapko tripool sample karna hai." So we were told that whatever it is, you give us the report. So we did give the report. We cross-checked it on 2 analyzers, Abbott and Roche, and we released this report of prolactin. To my surprise, sometimes you get a gut feel, isn't it? Something is going to be, you get a premonition kind of a thing. So we did speak and nothing happened, and the patient was like not willing to reveal anything. After 6 weeks, I mean, this is a story which we have just discovered with the psychiatrist. That's why I'm telling you. So we, prolactin levels were 85. So we were asked that why so much of variation. So we asked whether the patient was on treatment. Nothing, no history is revealed to us, except that the gynecologist told us that yes, there is this Dr. Kirtik, we know this doctor, who was treating this patient also. He is a psychiatrist. Obviously, medical records are not easily disclosed to all of us. So on reading, what I'm trying to tell you is on reading, we found out that this particular drug does cause a fluctuation and very, very clearly mentioned in the levels of prolactin, and it can cause fluctuations in the level of glyco-HB. So these are 2 parameters which can change and it can change to any level. Like you can get a glyco-HB of 13, and if the patient starts this treatment and within 8 days, if the peak of that particular drug happens into your body, which is called as hepatic clearance, and he does the glyco-HB in some another lab or for your lab, the level of his original level and this level can show a huge variation. So unless you have a clear indication about what the patient is taking, I don't think anybody will tell us that I'm taking an antidepressant, or I'm going to see a psychiatrist, or possibly I've taken gabapentin. I think it's not, at least in my practice of 24 years, I haven't got this. Only on digging up the records is what we get. So now safely, what we are doing, obviously we are trying to talk, but we are putting these notes. Please, if this is what it is, your levels can fluctuate. Advise to do a tripole sample, advise to communicate to us like we do in histopath, isn't it? We write below, we are giving you blocks, 3 blocks, 8 slides. Please confirm you have received, because tomorrow you are the one who are going to be facing legal or I don't know, clinical queries. So this was a very good case which I felt. So not all high prolactins, you know, or whatever we see are not PCOS. They can be drug induced, they can be psychological, they can be pulsatile releases, they can be just early what do you call, markers of PCOS even, or early indicators of insulin resistance. So that is what I wanted to get, and this is my last slide. So here I want to highlight on sperm DNA fragmentation, because we saw this case again with this old report of 2018. Findings are pretty good. We asked him to repeat the sample. This is a wet mount under phase contrast, and on this we saw 2 interesting findings. One is a live video which I'm not able to put it here, but we did see agglutination kind of a picture, so we advised to do anti-sperm antibodies. To our surprise, we do it with serum also, and I'm sure some of you must be doing with semen also. The more precise ones are semen anti-sperm antibodies, because obviously it's a live sample, and you can, if you have the right technique and the right kit, you can diagnose it. And the most important thing was the sperm DNA fragmentation. We are doing it by both flow cytometry, and we are also doing by indirect immunofluorescence. So here, this particular case, though all the parameters were normal, we saw anti-sperm antibodies and sperm DNA fragmentation index very, very high. On treatment now, we don't know, we are following up, but the repeat sperm DNA fragmentation, initially the report was 33%. We repeated the sample, and now the patient is on antioxidants and lifestyle change. The reason I mentioned pilot, when I was making these slides in the flight, I was asked by my daughter, why are you writing pilot? The reason I wrote pilot, because on the history, I'm not generalizing, we got the history of regular alcohol intake. So we advised him to stop alcohol. Obviously, how much will happen, we don't know. Antioxidants, and we did consult the gynac for anti-sperm antibodies. So with that, time is up. So I think I would like to thank Mindray and CAHO for giving me these opportunities. Thank you.
Thank you very much, madam. Any questions?
Hello. Yeah, hi, Dr. Aparna. It was a very good presentation. And I have a case study of one patient. On 19th of November, the prolactin level was 64.91 ng per ml. And 2 days back, it was reported, means on 17th of November, it was reported 388.86 ng per ml. It was a huge difference. So was it a lab error, number one? Because it was the report of 388.86 was reported from my lab.
Yeah.
I'm founder of Times Path Lab Private Limited, Gurgaon. And we ran the sample on Abbott CI 8200.
Yeah.
Both the levels of control were pass. And one thing which I would like to tell the eminent audience, that Abbott is the analyzer which does not have biotin interference. So I don't know how much biotin interference would have caused to bring this report too much normalized, too much on lower side, 64.9 on the after 1 day, means after 17th, or the second was on 19th. So the 19th was from Lal Path Lab. And I did not find the right answer whether my report was wrong, because after this much variation, gynae do not want to support. They want to change the lab. Okay? So I just wanted to know, can the prolactin B fall in such a great extent in 1 day or 2 days time? Number one. Number two, what could be the better way? Can the pool sampling would be the answer of it?
Thank you so much for being so honest. First of all, you have said it's your lab. It's very painful to be on that side. So I'll give you my answers, which I have been digging over years. Before that, one example. I have a very famous doctor, most of them from Mumbai, Maharashtra know her. Her name is Dr. Sangeeta Pikde. She is a very eminent speaker, and she had a patient with me, same problem. I did prolactin, I got a value of 48 or something around 50. As a protocol, I said, doctor, this is what has happened. So I'll repeat the sample. I'll go to the patient's house, because that's our protocol. If she's not willing to come, we want to do the tripooled, because we do see. The first answer, how do I convince the clinician? It's very tough job. So I said, she said, Aparna, leave it. I'm expecting this value. So we released it with a caution that advice to do this. To my good luck or bad luck, I'll call it good luck, because the next 2 days later, patient changed Sangeeta Pikde and went to my previous organization, that's SRL, and did a lab, because that's adjacent to them. And unke yahan pe 288 aaya, almost 3 to 4 times than my value. So I had, obviously, I'm lucky because I had my sample as per NABL protocols. I was keeping it. My SOP says 7 days, so I had it with me. So I challenged, I also do it on Abbott and Roche. So I said, here is the sample, take it wherever you want, okay? So patient went to the third lab. Trust me, all of us are facing, that's why this topic was chosen. In fact, the time is very limited. Otherwise, wonderful cases are there, but yeah. So third lab, again reported 47, 45 something. That's the lab of my friend, Dr. Jariwala. So I said, look, I am right. So the question was, why, who is right, who is wrong? Now we don't want to know. Then the clinician changes the side. Tell me why this has happened. Okay, now the first thing that should have come to me, but that didn't come. You are wrong. So only on digging up what we saw, there are 2 such cases. One case I presented here. I changed the dates because the patient was on antidepressant. That's what we saw. So here, what happened, the moment I gave a value of 48, the patient went to doctor, most of them from Mumbai knows Dr. Tushar Bankar. He is an DM endocrinologist. उसने 48 देख के immediately gabergolin चालू किया. If you read pharmacokinetics, so we as a pathologist, imagine, or microbiologists or biochemists have to not forget our first and second MBBS. What it says, and then I digged up the data. I got a German person who is doing work with Abbott. He shared the paper with me, where once you start gabergolin, that patient was on gabergolin the day I gave the report, and uska bioavailability is, the peak happens in 8 hours. Can you imagine? None of the clinicians know that. 8 hours, the value can come down. So the patient ने क्या किया, gabergolin start करके लिया ही नहीं. Patient went to MRI brain किया, pituitary adenoma आया नहीं है. Patient kept the prescription with her and did not take that medication. 3 days later, there was a pulsatile rise of prolactin. that explained Tushar Bangar's diagnosis, the DM endocrinologist. So what I'm trying to tell you is here both of us are right. It's the prolactin, which is a notorious hormone, which has behaved, and that is how it behaves. If the patient's endometrium, so here we all will have to sit and look at the radiology reports, the MRI reports, the patient's prescription. I know your machines are good. Who is right, who is wrong, we cannot comment on Lal Path Lab. I'm sure they also are following, everybody knows. So here, only thing what can be done, what I did, I set the sample, I said, this is your, like you give back the dress and say, you fit it to anybody you want. I said, boss, take your sample, run it wherever you want. I am not wrong. When that tallied, the patient said, abhi mein kya karu? Okay. So I think coming back to your thing, both of you are right. I'll try to share that paper with you where it shows prolactin can rise as much as 400 to 500 units in a day.
That will be great.
I will share that with you. And that's the reason I deleted all the slides in the flight when I understood the time. And I said, at least let me highlight on one hormone. Because insulin also behaves very, very massively I mean, you will feel so humiliated at the end of the day that somebody throws the paper on you and says, no more business to you for private practitioners. And it's unfair because you ask yourself, I am accredited, I am running controls, I am having barcodes, my Six Sigma is good, my everything is good, then why? So I think all of us as a community should come together on such platforms and form a group like we have in autoimmune, because that is something which can protect each other. I'll try and share that paper, and I'm gladful to Sangeeta pick it.
Without any medication,
it is, yes.
Can the
yes.
prolactin be can rise or fall?
Yes, yes, yes. But if you tell like this, nobody is going to believe you, because nobody believed me. So first thing is like transparency, what you did. Okay.
As
now
I have read in Google, like the serum half-life of prolactin is 4 hours or 3 hours. So this serum half-life.
Yes, yes.
So it may go down.
And
by
आपको अगर हिस्ट्री लेना है ना, तो आप अगर ऐसे पूछोगे ना, कुछ ले रहे हो क्या? हम कुछ नहीं ले रहे जी. वो गैबरगोलिन कुछ नहीं होता है उनके लिए. I had a case in Kerala where TSH, again everybody will think TSH क्या है? TSH, we did a camp. Everybody does a camp. हमारे पास 100 के ऊपर आया. हमने पेशेंट को हिस्ट्री पूछा, मेरे को वेट लॉस है, थकान है, वेट गेन है, थकान है, तो ऐसे करके रिपोर्ट इश्यू किया. अभी एक महीने के बाद she goes to DDRC, which is one of the biggest labs, and then throws the report on my staff and says, I am 0.8. तुम्हारा report गलत है. So I said, why don't you say that to them? I'm not against them. I am saying, why? So nothing high is not liked by anybody. If somebody says you are fat, then you will say that lady does need some correction, right? Nobody likes criticism. So people who got high values will throw it on you. So मैंने बोला, देखो भैया, एक महीने तक हम लोग सैंपल रखते नहीं है, कोई भी नहीं रखता है, unless you are having that big setup. Okay, I am right. You go and what have you done? Share your clinician's number with me. If need arises, I will. And refunding money is not. That's another thing which nowadays all of us are facing. Okay, that's not going to happen because I have my data. Everything is right. Let me talk to your clinician. On talking, the patient is taking homeopathy. Eltroxin नहीं ले रहा है, तो क्या ले रहा है अपने को पता नहीं है. Homeopathy can be steroids. You don't know. So we said, take this report. That's how lawyers will come into the picture, because the second thing which is thrown on us, आदमी केस करना रहे. Okay, okay. What can you do? Because you have your things. So I understand the pain points. The reason of getting this forum in maybe we all should contribute into our clinico-pathological cases, and that's how we will become more alert, more vigilant, and more important into this society of diagnostics, which is just becoming numbers. I really feel that. But I'll take your number and मैं आपके साथ share करती हूँ.
Thank you so much.
Thank you.
One more thing, ये pool sampling का तो इसमें कुछ benefit नहीं हो सकता, right? Because same day में
आपके केस में कोई benefit
कोई
नहीं है.
benefit नहीं. Thank you so much. Thank you.
Thank you. Thank you, Shama, for giving me this topic.
Thank you, Dr. Aparna, for vividly discussing this topic, which is a very puzzling to our clinicians as well as to the pathologists. Thank you very much.
Thank you, sir. Thank you, madam. May I call on stage Dr. Manglesh Srivastava to hand over the certificate to Dr. Aparna? May I call upon Mr. Vikash to May I call upon Mr. Vikash to hand over the certificate to our honorable chairperson for 2 sessions? Thank you, sir. Thank you so much. Thank you. Thank you, Dr. Manglesh Srivastava. Thank you, Mr. Vikash Kumar. So it's time to have lunch, but looking at the time constraint, is it okay we have half an hour lunch break and then we come back, because there is an online speaker, Dr. Barnali, waiting for us. So we can have half an hour lunch and then come back to our venue. Thank you so much for your understanding.
Thank you very much, online participants. We will resume in half an hour. Thank you very much.
The time when we choose this path, we are all ready to take a leap into the unknown. We embarked on our journey since 1991. For these 30 years, we are down to earth and forge ahead with our initial intention of bringing better healthcare for all. During turbulent times, we adhere to innovation as always. Along the way, we stay close to our customers, going deep to the front line to achieve continuous breakthroughs. From a single product to the completed solution. From patient monitoring and life support to in vitro diagnostics and medical imaging and so on. From primary medical institutions to top-notch teaching hospitals. From one-lane focus to diversification. From a startup to a global company. "30 years on, we never stop innovating ourselves. Together with our global partners to face challenges and celebrate success. At the age of 30, we are ready to take on new challenges with our original heart and will. We will be always striving to develop and share technologies. creating customer value with comprehensive solutions and broad portfolio. With the aim of providing better care for more people and making healthcare within reach. Our mission lies not only in the past, but more in the future.
India, a land of many faces. It's the bustling markets of Delhi, the thriving streets of Mumbai, and high-tech powerhouses of Bengaluru. It's feeling the beat of different pulses and the harmony of different sounds. It's where trust is placed in a critical moment with our life support. It's where reliable data are transformed into discovery with our IVD solutions. It's where intelligent inventions save lives with our imaging technology. And where healthcare is advancing with more possibilities. It's the trust in your partner that makes history, whether big or small. As your trusted partner, together we will reach the summit and watch the sky glow. Together we will build a healthier Bharat. Mindray, healthcare within reach.
Mindray Serum Automation Line Series, including the SAL 8000 and the SAL 6000 so far, are high-functioning integrated systems connecting seamlessly the clinical chemistry analyzers with the chemiluminescence immunology analyzers. By delivering fast, reliable results of both clinical chemistry and immunology tests on one platform, the systems are designed to optimize your laboratory workflow and achieve maximum efficiency. The SAL 8000 is a high-volume system connecting the BS 2000M, one of the fastest chemistry analyzers in the world, with a throughput of up to 2,000 photometric tests per hour and 2,200 tests per hour with ISE, and the CL2000I, a high-sensitive chemiluminescence immunoassay analyzer at 240 tests per hour with 36 reagent positions. The SAL 6000 is comprised of the CL2000I and the BS 800M clinical chemistry analyzer, which runs 800 photometric tests per hour and 1,200 tests per hour with ISE. With seamless integration, Mindray SAL series achieve much more than the two standalone analyzers combined. Save labor and space. The systems simplify laboratory work process by conducting both clinical chemistry and immunology tests on one platform. Only one tube of blood sample is needed for both tests without additional aliquot. Instead of two operators, one is enough to operate the system, using a single computer interface, which helps laboratory to save labor and space. All advanced technologies in one. Mindray SAL Integrated Systems Series have benefited from the most advanced technologies in clinical chemistry and chemiluminescence immunology. The clinical chemistry unit is equipped with a concave grading system and the reliable indirect ISE module. It can also detect sample clot and reagent bubble to ensure accurate testing. The chemiluminescence platform utilizes the non-touch vortex mixing, 4-step magnetic separation, and high-sensitivity ALP plus AMPPD system to enhance its performance, and uses bottom piercing and auto-switch technologies to reduce substrate waste and contamination. Uncompromised performance. The integrated systems connect two analyzers without compromising their performance. The systems offer a large capacity for 300 samples and support non-stop continuous loading of samples. An STAT button allows to push the normal racks back and load the STAT rack immediately. The uniquely designed rack delivery mechanism with a manipulator assembly can distribute sample racks efficiently to provide quick response, especially for STAT and rerun tests. The systems can also distribute samples dynamically and intelligently between chemistry platform and immunoassay platform to improve the throughput. Minimize carryover. The SAL 6000 and SAL 8000 have special carryover prevention to guarantee the results. For example, there is an intelligent rack distribution principle for highly sensitive tests such as HIV and HBsAg. Enhanced probe washing is also adopted to avoid sample and reagent cross-contamination. Broad test menu. So far, Mindray provides more than 100 parameters for chemistry and immunology, with a lot more to come in the near future. The result accuracy is guaranteed by Mindray's quality and traceability system. Mindray SAL Integrated System Series optimize laboratory workflow substantially by combining all benefits in one platform with minimum labor cost. The flexible on-site integration between clinical chemistry and immunology analyzers can cater to customers with diversified needs and offers them an accessible solution to laboratory automation.
After more than 2 decades of development, Mindray Clinical Chemistry has established a comprehensive portfolio of solutions ranging from benchtop analyzers to chemistry CLIA integrated systems with original manufacturer reagents, calibrators, and controls fulfilling a wide range of laboratory needs. Now, we launch our next generation standalone chemistry analyzer, BS430, Mindray's new flagship standalone chemistry analyzer, strikes a perfect balance between size and throughput and will be a reliable solution for your expanding laboratory. BS430 has a large region onboard capacity and sample loading volume, which is suitable for a wide range of laboratories from 200 chemistry sets to 800 tests per day. Together with Mindray's reagents and calibrator, BS430 provides a system solution with traceability, which offers users not only accurate results, but also intelligent functions and easy maintenance. Equipped with a high-precision aspiration system, BS430 can maintain a high degree of efficiency. A sample clot detection function can prevent abnormal results at the source, and the waterfall cleaning design can guarantee a minimal carryover rate. Furthermore, BS430 has a concave grading system providing 12 wavelengths and ensuring accurate results via a stable and reliable photometric system. The intelligent system supports abnormal reaction curve detection, including substrate depletion of enzyme parameters. BS430 is able to identify samples with abnormal reaction curve caused by high enzyme concentration and trigger auto-dilution and auto-rerun. Moreover, BS430 can provide an enzyme linear extension function which delivers reliable results without dilution and rerun, reducing not only reagent consumption but also TAT time. BS430 also supports HbA1c onboard hemolysis, effectively reducing the cumbersome procedures of manual preparation of hemolysates and preventing biohazard risks and manual operation errors. In terms of maintenance, BS430 is equipped with a maintenance-free, solid direct heating system in its reaction compartment. As for its operation, an intuitive service wizard in the software helps operators carry out automatic maintenance step by step, simplifying maintenance procedures and increasing ease of operation. In this era of technological advancements, expanding laboratories with increasing samples are facing limited space and longer TAT. Their current chemistry systems may not be able to meet the growth in demand. In this situation, the powerful and compact BS430 is the right solution for you. For emergency tests, BS430 can wake up from sleep mode within 1 minute and generate a report within 30 minutes. Mindray is dedicated to providing comprehensive solutions for a range of laboratory needs. The BS430 features more than 60 different reagent parameters, all with the necessary calibrators and controls. BS430, a reliable solution for your expanding laboratory.
In today's climate of patient care, laboratories around the world are facing a growing demand for higher quality of care, lower operating costs, and faster turnaround time. The Mindray BS600M chemistry analyzer is intended to help laboratories achieve their goals. BS600M is a compact chemistry testing system that can deliver maximized throughput in a minimal footprint of 1.2 square meters. The robotic sample handler helps to maximize the sample loading capacity. Continuous sample loading by racks also contributes to higher efficiency and increases walk-away time. Stat samples can be run quickly in around 1 minute. This means that results can be reported promptly to allow doctors to make critical decisions for patients. Onboard hemolysis with whole blood samples for HbA1c tests can simplify the laboratory workflow. Online pre-treatment without any manual intervention also reduces biohazardous risks and human errors. Caliber and control values are input by QR code scanning intelligently, which helps to improve work efficiency and eliminate human mistakes. Besides its powerful functions for maximum working efficiency, BS600M also provides powerful performance with reduced costs. The BS600M chemistry analyzer can provide excellent quality of results with a substantially lower reaction volume to a minimum of 70 microliters, which ensures reduced cost of reagents for laboratories. The integrated ISE module offers reliable sodium, potassium, and chloride results, which can be reported together with photometric chemistry tests. The ISE electrodes have a 9-month stability period, which is sufficiently long to help reduce the replacement frequency and cut down the cost of electrolyte tests. BS600M with Mindray original reagents can lower the inaccuracy and imprecision, which contributes to achieving world-class and excellent sigma metric performance. Systems with higher sigma level can help laboratories offer high level of quality at reduced cost and enhance clinician confidence and satisfaction. BS600M is designed to incorporate more innovative technologies and be more user-friendly, making the system be powerful yet efficient. Therefore, it enables laboratories improve working efficiency and save the cost. Moreover, it helps reduce false results risks and enhance the quality of patient care.
Welcome back after the good lunch. And moving ahead, we have very good talks again. I hope it's it's giving thought for the brain. So moving ahead, I hope none of you fall asleep, since we have very, very, very good speakers ahead. And I hope, and they will not let you fall asleep. Okay, so we start with the chairperson. So we start with the next session, the talk by Dr. Prajwal Amali. येतील चालू नाही गेलं ना ते बाहेरच राहणार. May I request Dr. Prajwal Amali go through the agenda. Amit, go. May I request Dr. Aparna to chair Dr. Prajwal's session, please? You can sit here. You can sit here. AC is not working.
So, please come inside, those who are having lunch, on behalf of Shama. Hi, Dr. Prajwal Amali. Welcome. He's from AIMS, New Delhi, consultant and head biochemistry at Agilus Labs, Fortis Hospital. Topic is, yes, uncommon interferences in biochemistry, an interesting topic. He comes with a rich experience with various institutes like NIMHANS, ILBS, currently, as I said, consultant and head in Agilus Labs. Welcome, Dr. Prajwal. Okay, has a publication on diagnostic markers in ovarian cancers. Thank you. Prajwal, pleasure to have you with your lecture.
बेल तो पहले ही बजा दिया, 2 बार बजा
नहीं, अब वो अपने नहीं है।
इसी में है क्या?
कुछ कम तो नहीं किया
सेंटर में।
Good afternoon, everyone. Thanks for giving opportunity for Mindray. So I will be talking about 2 uncommon interferences in biochemistry, which we come across mainly in hospital settings. So there was a 68-year-old female who was regular on health checkups for last 4 years. Everything is unremarkable. She came to OPD with jaundice and she has been admitted for further workup. This was the LFT report. If you see, it is more of a obstructive jaundice picture with increased total bilirubin and increased direct bilirubin. And the protein was very low because of low albumin. And we went further tests. There was a surprise, cholesterol being 1002, and last year health checkup and all, cholesterol was unremarkable, normal. Triglycerides were also just borderline. So, and this was the electrolytes picture in this patient, sodium being 123 and chloride being 93. So I immediately called up the doctor and informed not to do sodium correction. Why? Sorry? ICTRES won't affect electrolytes much. And after I asked to stop sodium correction, and I immediately advised to do ABG on the spot. Yes. So when ABG sodium was checked, it was 140, and this is the case of pseudo-hyponatremia. So I'll be going through how exactly this pseudo-hyponatremia occurs. To understand that, we have to understand what exactly the components we measure during electrolytes measurement and what exactly the calculation happens later. So if you take a serum, there will be serum water content, what we call as HWC, and serum solid content, SSC, which will be around 7% and 93%, which is the constant. When there is increased serum solid content, issue status. What we see on the reports is serum sodium, but what we measure is serum water sodium. We measure only sodium in this component. We calculate and further extrapolate and report it as serum sodium, what we see in daily reports. So what is the relationship? Serum water sodium is serum sodium divided by serum water content, or you can multiply the other way around. So when you call it as serum pseudo-hyponatremia, when sodium serum water is normal, but what you see in the report that sodium serum is low, then you call it as pseudo-hyponatremia. So to understand that, there are 3 factors which commonly affects why it's generally seen only in indirect ISC, but not in direct ISC. There are 3 factors. Most important factor being dilution effect. To understand that, I will show a small calculation, how exactly the calculation happens behind the instrument or in the instrument. So this is a serum sodium for what you see in normal 140 report, sodium serum would be close to 151. What you see is sodium serum water, it will be 140. So I am taking base sodium as 151. And I am taking three scenarios, solid content, one being 7%, 14%, and 21%. So based on fraction, we'll have the serum water content. And this is the amount of serum that the equipment would aspirate. Generally, it will be 10 microliters. And how much would be the serum sample water? It would be water content, that is 0.93 to 0.01. Same applies for all the columns. And you add dilution. Most of the dilutions will be from 22 to 34 range, depending upon the manufacturer. I am taking 30 as the dilution factor. So in the instrument, to the 0.01 liter serum, you will be adding 0.3 liter diluent. So by this, the total sample water will be this, that is serum water plus diluent, total volume. So what will be the dilution factor? In first case, the dilution factor is 33.2. In second case, it was 35.8. In third case, it is 38.9. So now the electrode would measure the electrolyte. So it comes like this, because based on serum water content, what you get the sodium values. So you multiply by 151. Now you measure total sodium. Then you come to the calculation. System does now. It will divide using the dilution factor. In first case, it divides, it is a normal solid content, so it will use a 33.2 factor, so value will come to 151. But in second case and third case, the instrument is prefixed for 7% only. It won't use the different dilution factor. It will use the same first dilution factor in all the calculations. Same 33 point. The 151 became 140 in second case and 128.9 in third case. The calculation doesn't stop here. What we report is sodium serum, not serum water. So there is one more calculation, that factor 0.93. This is also constant in three scenarios, because these do dilution factor and 0.93 is prefixed in the system. So in first case, where the solid content was normal 7%, you get normal sodium 140, whereas 14% solid content, the sodium would be reflecting 130, whereas in third case, where the 21% of solid content, what you measure will be 120. This shows pseudo-hyponatremia because of dilution factor effect happens in indirect ISC. It is not seen in direct ISC because there is no dilution at all in direct ISC. There are other two factors, hyperviscosity effect, majorly seen in multiple myeloma patients where aspiration itself, so there will be low aspiration of serum, so dilution will be more, but you are using dilution consistent factor, so again the calculation will go wrong. So you will have abnormal value again. And electrolyte acceleration effect, which you don't see nowadays because it is only seen in flame emission spectrometry. So coming to summary, when you should look for pseudo-hyponatremia. One, when the protein is more than 10 grams. Second, when TG is more than 3000. And when cholesterol is more than 1000. And how to identify? There is discrepancy between lab report if lab is using indirect ISC and ABG. And there is high osmolar gap between measured and calculated. And there is discrepancy between measured LDL and calculated LDL, because I'll coming to that why it will happen. So three mechanisms I already explained. One is dilution effect. The second one is hyperviscosity effect. The third one is electrolyte exclusion effect. And there is one more thing in this case, why the cholesterol is 1000. So this was a case of bile duct carcinoma, obstructive bile duct carcinoma. In case of obstructive bile duct carcinoma, because of the blockage of flow of bile, there is an abnormal lipoprotein metabolism, which gives rise to abnormal lipoprotein called LPX, sometimes LPY also. This is falsely giving high value of cholesterol. Some in few equipments, depending on the method, it will even give high value of direct LDL also sometimes. It's very important to identify LPX. It could be identified only with electrophoresis, lipoprotein electrophoresis, which is rarely available. Best way is if you are seeing generally cholesterol more than 1000 without any genetic this thing, then it's most probably because of LPX. The obstruction should be relieved immediately because it is highly atherogenic. It is noted that within 28 days it could cause a plaque rupture and cause a MI. There is something pseudo-normonatremia, which is associated with hyperglycemia. It doesn't generally cause pseudo-hyponatremia, where there also you have to stop sodium correction. So which laboratory technique, I went and saw how many people use direct ISC and indirect ISC. There was a very good systematic review where people have checked what technique is being used in a hospital and everything. They took 361 articles and they had only information in 177 articles, where 126 were indirect, 25 were direct, and 19 were both, and 2 were flum. And remaining 50% article didn't have information. Why I'm highlighting is, we all will be publishing. I want you to highlight that you should kindly mention the method. Otherwise, clinician will be discussing with one method paper and you will be releasing the report in the other method, so that it can be confusion. And surprisingly, all these 361 articles were related to hyponatremia only. Only 177 articles had the methodology. The other half of the articles didn't have the methodology. So coming to second case, sorry for the pic. This was the WhatsApp pic sent by my nephrologist. That was the report released by me, and this was the ABG report. Potassium. Done within 10 to 15 minutes difference. So I repeated the test with fresh serum samples. Still the serum sample potassium was 6.48, but ABG value was still giving 3.5 even with the repeat sample. So we went and looked. It's a case of pseudo-hyperkalemia. Patient had platelets of 30 lakhs. So potassium was leaking. By the time sample reaches and you centrifuge, potassium leaked into serum and it was giving high value, whereas in ABG you don't centrifuge and it was immediate. So the value was coming normal. So when you will label, before labeling pseudo-hyperkalemia, you have to rule out pre-analytical errors. Without ruling out pre-analytical errors, you can't put into pseudo-hyperkalemia, like prolonged tourniquet time, squeezing, heat contamination, delayed transport, or excess centrifuge, and hemolysis. So when to look for pseudo-hyperkalemia? Most common causes are thrombocytosis and leukocytosis. Thrombocytosis, about 20 lakhs of platelets, you can see this. And in leukocytosis, above 50,000, you will be seeing this. And there are two rare genetic disorders. One is familial pseudo-hyperkalemia, as the name suggests. And the other one is dehydrated hereditary stomatocytosis, where the leaky potassium happens from the membrane. Solution, one option was, in my case, we did a POCT there itself, and the potassium we asked to continuously follow up, because until the platelet comes down, I can't give the potassium value. Or you can use heparin samples, lithium heparin specific. It may help, which will have shorter centrifuge time. You bring it immediately, centrifuge only for 3 to 5 minutes, and report it on plasma, saying difference with the serum. There is a difference in reference range for serum and plasma. Just put a note on that, and you can do still if you have a POCT facility. Thank you.
So, Dr. Prajwal, thank you so much. I just want to ask if you can throw some light on what happened when you gave this report of pseudo-hyperkalemia. Clinician sent you that ABG report on. So, how much time it takes for the clinician? Because that is also very, very critical. So, how did you handle this?
So actually what happened with this case, I released the report and I was walking along with my hematopathologist. He also got the call, I also got the call from the same nephrologist saying he has released higher platelet count, I have released potassium difference. That's how I got to know the platelet, because I am with him for the breakfast. Okay. So we could able to solve very fast. As soon as I heard 30 lakhs platelet, I knew the answer. I sent a Barricard tube to get the sample. It was showing only 3.
So thanks to the breakfast.
Yeah.
And one more question is about the sodium.
Yes.
Now, if there was no lipid profile with that case, and just like say an electrolyte sample comes to me without a lipid profile, and I get this, then
it would generally we see for HIL indices, lipemia would have picked.
He
would have picked.
You know, I mean, it can be missed.
It can be, because triglycerides never directly proportional to turbidity.
Yeah.
It may be missed definitely.
I think it's a super, I think the moral is, yes, luckily you were with the clinician, and most of us should also know high platelets can cause this. If
Yes.
ignorance is bliss, then you would have missed it. Yeah. So fantastic. I think we should, get this spread more on our platform also. We will try to, ask you to come on our webinars also, so
Sure.
that most of people who are attending, it would really be beneficial for people who are functioning in hospital-based labs, particularly because it's both bilateral. It's on fire also, but there is beneficiary, like you can immediately get histories and other
Yes.
reports also. Thank you so much. Thank you. Any other questions?
So no more questions, it seems. May I request Dr. Aparna to hand over the certificate to Dr. Prajwal, please? मैं वही देखना है डॉक्टर बरनाली क्या बोलती है है क्या इट्स ओके फॉर हर वेट
Yeah, I am fine. Please carry on the schedule.
Yeah, ma'am, 1 second, ma'am. We just want to get your audio in the main hall. 1 second, madam. Doctor का audio डालना. Doctor बनाना बस. Switch off.
Shama is
Yeah, yeah.
Yes, yes.
Can you hear me? Can you hear me?
Yes.
Yes, ma'am, we can hear you.
Okay. So I can wait also if the schedule talk.
No,
Is it okay?
We are waiting for you and Aparna here.
Okay, okay, thank you. So I'll just share my slide. Is visible?
Yes, yes.
Thank you. Thank you very much, Mindray, for organizing SFCC. And on behalf of IFCC Executive Committee member of Scientific Division, I congratulate Mindray for organizing this academic treat and being a diagnostic division member of CAHO and also member of ACCLMP. It's my privilege to have this talk and in front of this great audience. And I'm really sorry that I can't, because there is some institutional ethics committee things are going on. So because of that, I have to attend that. So today my talk is on proper implementation of IQ, OQ, PQ, the tools to ensure the quality of results. So for us diagnosticians, every blood sample tells us a story. Since morning we are hearing, we heard Dr. Thupil Venkatesh sir, then Dr. Manisha, and then Dr. Aparna, and now Dr. Prajwal. So we have seen how different, how quality control tools, as well as how different case studies, how being alert, and how implementation of the protocols and harmonizing with the clinicians, how it is important. So let's see, this is my acknowledgement to all the diagnosticians present here, biochemist, pathologist, microbiologist present here, to acknowledge all of our efforts. Maybe we are seeing slides under microscope, or many of us are doing tests in autoanalyzer and interpreting this RT-PCR. So we are not behind curtain, and also the stakeholders like IVD stakeholders like Mindray. So everybody, we have seen during COVID time how all stakeholders played a very significant role. So this is my painting to acknowledge all of our efforts. So the genesis of my today's talk is this TSH report. So this, like Dr. Aparna mentioned during her talk about AMAs, so many times, like this TSH is a very simple test we are doing routinely. And many times we see patient goes to 3 different labs during the short span, or maybe same day they send 2 samples to 2 different labs, and seeing also morning and evening the difference in values. So like one lab reported 0.02 microinternational unit per ml, another reported 0.67, and we reported 0.1. So then when 3 reports same time, the patient goes to the endocrinologist, then endocrinologist is thinking what lab result to believe and what which is a good lab. And so this is us, our lab system. If we see this, we are coming with burden, patients are coming with burden of disease. Then we implement our intelligence, discipline, and creativity. And then patients are going with reports. And sometimes we get call, like the previous talk, he mentioned that when he was having the breakfast, he got the call, and luckily the hematologist was with him. So we get the call from clinicians, and then Barnali, what creatinine value you reported, what or many times maybe they want to do, like just now I received a call that they want to do IPTH in tip, the needle tip. So how to report that? See, IPT in needle, saline. So sometimes we need to guide also the clinicians or radiologists that you have to do saline wash for PTH, and then send the saline wash PTH sample for cystic fluid. So these are our role. So sometimes they may be happy, our clinicians, or sometimes our patients are complaining, oh, why there are 3 different TSH report. So because of that, it is our role. This is our today's lab system. And if you can realize this can be you or me, being an ABL assessor or CAP inspector. So we go for sample audit for College of American Pathologists 1 day, or for NABL 2 days. And based on our observation, we say, oh, this lab seems to be very good. There is no non-compliance, or there are so many non-compliances or deficiencies. So this is our today's lab system. So this is the genesis of like, why do you think that there are 3 different reports at the same time? Do you think that sample transport or sample integrity is issue? Because being an assessor, many times I have seen all the transport media for sample transport, not seen yet the elephant, but hopefully I'll see that too. But so do you think the sample integrity is an issue, or do you think the analytical variation between instrument to instrument is real challenge? So, so for that, I will take the reference as thyroid hormone standardization, because I was part of IFCC Committee for Standardization of Thyroid Function Test, and also being in the scientific division, we are trying to standardize and harmonize many of the immunoassays and biochemistry assays. So are we nearly there yet for thyroid hormone standardization? So it's for any like biochemistry or immunoassay testing. So can IQ or QPQ really help us? So I can see Dr. Tina Liu from Mindray. So last time when we met, I think in Rome, so we discussed that at like, you know, at length, like how all the IVD, because we see a lot of variation between instrument to instrument in the harmonization and standardization. So how we can harmonize the test in biochemistry and immunoassay platform. So these are some of our papers, published papers in European Thyroid Journal, Clinica Chimica Acta, and Clinical Chemistry on thyroid hormone, like how to harmonize and standardize. So now these are based, some are based on reference intervals, some are based on like recalibrations and harmonizing, or based on the APTM, or purpose stream mean, how we can establish after recalibration. So these are some of the papers. And now the outline of my today's talk is understanding IQ, OQ, PQ for IVD devices, then installation qualification, what are the tangible and intangible aspects related to installations, then requirement of the IQ, and then operational qualification and performance qualification. I remember when I started my journey in lab, so initially we were only bothered about the performance qualification in lab. About IQ, about OQ, we used to think whoever the manufacturer or whoever the IBD person supplying us the instrument or kit, so they will do the IQ, OQ, PQ. So why I should bother? I should only be bothered about the part of PQ or part of the performance qualification. But that's not correct. So now, from IQ, from installation qualification to operational qualification and performance qualification, the lab, as a lab personnel, we need to validate that, verify that, and we need to sign that. So let's see how to do that. So IQ, OQ, PQ protocols are methods for demonstrating that equipment being used or installed will offer a high degree of quality assurance. The goal is the process validation to deliver a stable IVD that offers consistent performance. Variation should be minimal and predictable, and the process validation plan will provide the framework. Like we can start, if it is a new lab, we can start from design qualification, then we can shift to the installation qualification. When we talk about installation qualification, it should answer, is everything installed correctly? Operational qualification, is everything operating correctly? Are operating limits understood? Then performance qualification, did it produce the correct result? Is the process stable and capable? So this is that full chart where from initially when we do this process, we form the validation team, then we create our validation master plan, then we create our protocols, perform the design qualification, then we perform IQ, OQ, PQ, and prepare the final report, and then we validate it. So IQ is, it verifies the instrument being qualified and have been installed and configured according to manufacturer's specification, and it is documented collection of activities necessary to establish that an instrument is delivered and designed and as designed and specified, and it is properly installed in manufacturer specified environment. So what should be the checklist? Then for checklist, the location of installation and necessary floor space, documentation, any and all computer control instrumentation, gathering all manuals and certifications, and then properly unpacking and cross-checking the instrument, examining instrument and components for damage, ensuring correct power supply, documenting firmware versions and serial numbers, environmental and operating conditions, checking software system installation and accessibility, recording calibration and validation dates of tools used for IQ, and verifying connection and communication with the peripheral units. So IQ verifies that the instrument being qualified as well as subsystem and ancillary systems have been installed and configured according to the manufacturer specification or installation checklist. The FDA's IQ definition offers a useful statement of the overall goal, documenting that the system has the necessary prerequisite condition to function as expected. So this comes along with the current good manufacturing practice CGMP requirements. So if we want that checklist, so components and equipment, software and standard operating procedure, then calibration and testing, then installation, software and system testings, records, electrical, records and verifying the electrical things in the lab, then signals and alarms, revalidation, facility condition, and documentation. So now IQ, there should be an SOP checklist or some other documented process that defines the standard installation procedure for each type of system or deliverable being installed. So these requirements may come from design specifications, system specifications, manufacturer's recommendation, and data sheet. So IQ verifies and documents key aspects of the installation meet approved requirements. So this, if we see the checklist, so this is one checklist from our lab. So these are the checklists like operator manuals, environmental parameters, instrument delivered, undamaged, transport locking, all connection correctly installed, instrument positions according to installation manual, instrument boot process successfully, and then specification met or not, mechanical adjustment, auxiliary components position or not. So these are different components where we should, when we are signing IQ report, we should see that that are installed properly or not. Or if there is a deviation, what corrective action, what preventing action the IVD has taken, and if the deviation resolved satisfactorily or not. Now coming to the OQ. OQ's purpose is to determine that the equipment performance is consistent with the user requirement specification within the manufacturer specified operating ranges. So after we have installed IQ, then comes the next step is OQ. This means identifying and inspecting equipment features that can impact final quality. So during OQ, all items in the test plan are tested and their performance is thoroughly documented. So OQ is associated with equipment performance to ensure that the function of the machine, measuring devices, utilities, and manufacturer areas perform as intended throughout all anticipated operating ranges in the selected environment. So actually, after completion of OQ, we permit a formal release of the performance qualification process. So what should be the components of OQ? Internal hardware calibration, software verifications, maintenance verifications, parameter calibrations, internal quality control, precision of analysis, verification of all the above with respective documents, documentations of the above step. So this is like the operational qualifications or OQ. These are the like calibrated successfully or not, then OQ quality control, then accuracy check successfully or not. So this is the validation process. And then this is the checklist and where we sign the OQ results acceptable or not. So the final steps where we as a diagnostician play a very, very significant role. The final step of qualifying equipment is the PQ. So in this phase, the qualification and validation team verifies and documents that the equipment is working with the reproducible results within a specified working range in laboratory setting. So the team must create a detailed test plan based on the process description. So we call sometimes as a process performance qualification or performance qualification. So the manufacturing conditions such as equipment limits, operating parameters, component inputs. So this all should be mentioned in PQ. So like in manufacturing condition, equipment limits, operating parameters, component inputs, and then calibrations, a thorough list of data that should be recorded or analyzed during test, calibration, and validation. So like the protocols can be precision, linearity, sensitivity, accuracy, method comparison, carryover study, reference interval. So I will give some of the process we have done in our lab. So yes, our lab life is like this philosophical lines of Albert Einstein. Life is like riding bicycle. To keep your balance, you must keep moving. So in our lab life, we have these two wheels of the bicycle. One is the wheel of quality. Second is the wheel of cost. Wheel of quality depends on accuracy, precision, reference interval, and wheel of cost depends on man, machine, and material, 3 magic M's. So we need who will do the balance between these two wheels to have smooth ride in our lab life. So this balance will be done by harmonization and standardization and proper IQ and OQ and PQ. So if we do proper IQ and OQ and PQ, so in that the variation and between methods, it actually we can harmonize our protocols quite a bit. So verification and validation play a very significant role, as we know ISO 15189 2022, and also the CAP checklist mentions that verification and validations is very important. So how we should verify any, so when it comes to we start any new test or any new instrument, so the verification protocols should consist minimally this like analytical accuracy, run samples by both new method and comparative or gold standard methods to evaluate the bias, then verify the precision, then checking the reference interval according to CLSI EP28A3C. So we check the reference interval and verify, I'm in CLSI standardization committee. So there are many difference in opinion, like I have seen, like also I'm part of ICMR reference interval advisory board. So when we talk about direct reference interval and we compare that with the indirect reference interval, so there are between our diagnosticians, still there are many variation in opinions how to establish the reference interval. Should we go for direct method or should we go for indirect method? So because of that, when we are doing verifications, it's very important to verify the reference interval. Now verify the LOB and LOD, limit of detection and limit of blanks, by running 20 blanks or zero calibrator or low patient samples, we can verify that. Then linearity verification. I remember during my CAP inspection, I have done everything, but I forgot, like I thought we are taking linearity surveys in CAP. So why we need to do before releasing any patient samples, why we need to do linearity verification. But I got deficiency because of that, linearity verification. Range that can be reported without dilution or pre-treatment of samples. So as we call that as analytical measurement range or AMR. So AMR is without dilution or pre-treatment of the samples, and CRR or clinically reportable range or extended measuring interval according to CLSI EP 34. So now the term CRR is, we call it as an extended measuring interval or EMI. So range that can be reported with dilution or pre-treatment of samples. When we talk about immunoassay, or like Dr. Aparna told about us, different fertility assays, LH, FSH. So we see, like, you know, the IVF specialist many times gives us a call that, Barnali, why you are not reporting the exact value? Why you are giving more than so-and-so in while reporting? Or sometimes with the tumor markers, you will get call from an oncologist, why you are not reporting the exact value, why you are reporting more than so-and-so. So I need exact value because I need to monitor the patient. So many times it's very difficult for us diagnosticians to give the reportable value because of this extended measuring interval or EMI. Why? Because instrument manufacturer, IVD manufacturer, like Mindray, so they do their validation, and in that they mention that you can dilute the sample till that range. Beyond that, we are not supposed to dilute. So because of that, we can't dilute any samples till infinity. So whatever the manufacturer mentions, whatever the limits manufacturer mentions, we can verify that, and we should have our CRR or EMI chart displayed in lab so that no technical staff, even at night, release any report which is beyond extended measuring interval or CRR. So this is how much time I have. Okay, so this is how we actually adopt the reference limits. So these are manufacturer suggested. I'm audible, no?
You have, no, I think time is done, Barnali.
Time is done, so then I will not, I have already said this. So this is, I'm just like, you know, how we establish the reference interval. So we, in 20 sample, we do verification, we can do, but suppose we want to establish, then we can do in the CLSI say 120 if it is following Gaussian distribution, but if it is not following Gaussian distribution, then 200 samples. So these are some of the like how we should check the accuracy. So this is our accuracy check. Then the how we should do the precision, like 5 replicates for 5 days, 25 replicates in all. So yes, data, and data. I can't make bricks without clay. So like in biochemistry lab or immunoassay lab, so when we need to establish our IQ, QPQ, when we need to establish our verification protocol, we have to actually go a lot of statistical analysis, and we have to give importance to our data. Thank you.
Thank you, Dr. Barnali. So, I mean, I think very well said. We should have had more time for you, unfortunately.
No,
Any
that's
questions
absolutely
for Dr. Barnali? Just a second. Huh?
Hi, Dr. Barnali. This is Dr. Dhanlakshmi from...
Yes, ma'am.
On the operational qualification of the instrument, what is your thought on including the environmental aspects of using the new instrument in our laboratory?
Yes, ma'am. We should actually, even in when we talk about the installation qualification, like actually because I missed the design qualification slides because of the time constraint. So when we are doing the design qualification, from that step, we should actually take care of the environmental aspects. This mainly comes in design qualification, but when and part of the installation qualification, but when we are releasing the for the performance qualifications, so we should have in our checklist the environmental aspects. But mainly we do it in the design qualification steps and IQ steps.
Okay, anybody else? Yeah.
Hello, ma'am. Good afternoon.
Good afternoon.
I'm Dr. Manoj from Ghaziabad, Uttar Pradesh. My question related to POCT analyzer.
Yes.
POCT analyzers. The manufacturers not provide OQPQ IQ for the POCT analyzers. So what to do in this case?
So like suppose glucometer. So many of the tests, like we talked that wave test and non-wave test. So many of the like for glucometer, if we ask for IQ, OQPQ, so that actually they don't provide. But the POCTs like for like high sensitive troponin I or high sensitive troponin T. So for that or troponin T or troponin I, for that, we need to have the proper IQ, OQ, because that needs IQ, OQ. So if they are not providing, then this is our role, like, let them provide the IQ, OQ, and we can do the PQ on our own.
Thank you. Thank you.
Okay, I think, yeah.
Great presentation, Dr. Barnari. So regarding the dilution of recommended by the manufacturer, in certain parameter, we have to extend the dilution beyond the manufacturer recommendations. Then what would be the protocol and how to validate that dilution?
So we can actually better to not give in written report the dilution, because I know a couple of the legal case where the lab has reported beyond the limit of the manufacturer. Then nobody can because they will ask another expert opinion, nobody can support that lab. So because of that, many times when we get the call, and mainly for the reference lab, because the second reference lab may be giving the diluted exact value for the tumor marker. So it becomes very difficult. So then you can verbally inform that this is the value we have got, but because of the manufacturer recommendation, we can't go beyond that. So better to stick to extended measuring interval or CRR in our report. And verbally, we can inform that this is the report. And if you ask, then we can do the dilution. So this protocol, we actually, well, in my practice, I follow. But written report is better not to give, because I have seen many 2-3 cases where in written report, the dilution given beyond CRR or beyond EMI. So then there is difficulty to give the expert opinion. I hope I'm correct.
Barnali, yeah,
Yes.
I think very well said. We do face this problem in particularly in commonest thing like TSH.
Yeah.
Oh, you give more than 100, tell me how much it is. Okay, so I think I'm completely convinced. It's better to stick to.
Actually, for TSH, it's not much. There is no, if I give 2000 or if I give more than 100, so that endocrinologist will do the same treatment.
Exactly.
Yes, but tumor marker, I have faced the many reference lab many times say we can't follow this because of the tumor marker, or you have mentioned during IVF specialist. Many times they will not accept that if you are not giving the exact value.
So it's better, I think what very, very well said. It's better to give the printed report with those instructions.
Extended, yes.
And you can say, okay, verbally, if you're going out of your way for the clinician, because legal angle is also very, very important and very important point for point of care. Because that is what is coming up and that's what is the future. In case they are not giving us, I think the onus is on us to get the PQs done well. Thank you, Barnali. Superb presentation, and we miss you here.
Thank you. I'm missing. I'm seeing so many familiar space, Dr. JJ and Vinny. So I'm really missing.
So thank you. Thank you so much.
Thank you. Thank you.
So I think Chama will show you the certificate
Yeah.
online.
Thank you. Thank you, Dr. Barnali. It was a
Thank
pleasure hearing your talk. Dr. Barnali, I hope you can see your certificate.
Yes, yes. Thank you so much. Thank you.
So may I call on stage Dr. Aparna Jairam?
Thank you.
So moving ahead for the day, may I call upon stage Dr. P.K. Mohanty to chair the session for Dr. Jayalakshmi Jayarajan. So our next speaker for the day is Dr. Jayalakshmi.
So, for the afternoon session, let's welcome Dr. Jayalashmi, eminent microbiologist, and all her this thing. She is a research committee member of Dr. MGR Medical University, as well as I know her as NABL assessor because she assessed my lab.
I saw so many familiar faces and I've seen them all online.
As well as so many other accreditation bodies like JCI, she is also
Thank you very much,
surveyor,
sir.
and also she also a member of the CAHO and program director, CPSC, then CAHO member research committee and all other things, all these things. You please
Yes, sir.
read this. And I won't waste much time. I request her to go ahead with the presentation because this is a very crucial matter. Although we talk a lot about this quality control issue, when it comes to a changeover from 2012 to 22, it's a real challenge for us. So she will enlighten us on this topic. Yes, welcome, madam.
Thank you very much, sir. First slide, please. First of all, big thank you to Mindray and Shama, and also to the CAHO. Very happy because Dr. Aparna pulled me into this. So thank you to be part of your, amongst you all today. It's a post-lunch session, and I'm going to talk to you something which is 1.5 years old, and probably a lot of labs have started to implement them in their own labs as well. But let's look at what we will want to do in our own labs when we are in the journey of these implementation. So I'm going to just take to the key points only. And so I'll be looking at just quickly into the ISO standard, just to understand how this ISO 15189-2022 comes in and what are the key changes in the standard and how do we implement them. So the ISO 15189, ISO would say it's an independent, non-governmental international organization since 1947. However, currently with 169 members, it shares knowledge and develops voluntary consensus-based, market-relevant international standards that supports innovation and provides solution to global challenges. So with changing techniques, technology, the way we look at things in the laboratories, a lot of things change. However, ISO has multiple verticals, looks at multiple things for standardization. We're going to look at the ISO 15189, which is a globally approved standard for medical laboratory practice. So the ISO 15189 is formally recognized by an authorized national accreditation body. In our country, it would be NABL, that a medical laboratory is capable of doing a particular activity as per the standard. That's what it would say. So the revision happens every 5 years, with the first one in 2003 and the next one in 2007 and 2012. So we didn't have it in 2017 because it is based on consensus, and that time it was it did not go through an approval for the next edition. Therefore, there is a wait, and then came with the 4th edition, which is the ISO 15189-2022. So there's a 10 years gap from the previous edition to this one. But there's a huge, huge change in some of them, and there are not much of changes to what we see, but how we see is going to be changing. So let's look at the ISO 15189-2022 standards. This is based on the evolving medical laboratory markets. Most of it is now international, and a lot of tests are done in one laboratory with multiple sample collecting points across the country or sometimes overseas as well, and done in one single facility. Or there could be a lab which is doing an exclusive, very specialized test in one particular lab. So it has to be relevant and applicable in both these situations as well. So these are technology of AI. We are moving into the point of care testings, and there are a lot of tests like what Dr. Tupil sir was saying. In 10 years, you're going to get all of these tests in a POCT mode. So we have to upgrade ourselves to the way we look at these tests and the standards that we need to follow for ourselves in our laboratories as well as for the accrediting bodies to accredit as well. So the objective of this particular document is very, very focused to look at the welfare of a patient. So patient is the center of this document, and satisfaction of the laboratory users who are the clinicians or the consultants who refer samples to the test, to these laboratories. So they have to produce, or there should be a confidence in releasing out a quality and a competent test report from these medical laboratories. So that's the objective of this particular standard itself. Now, I mean, this whole standard in the document itself. Now, what does the document contain? Okay, it contains requirements on how these medical laboratories should plan and implement actions to address basically the risks and look for opportunities for improvement. Why is it speaking about the risks when it is saying something else in the previous one, where it is looking at the patient as well as the user or clinical lab users, as well as looking at the quality and competence? Because at every level, you're going to look at them and look at the risk involved to either the patient, the sample, the healthcare professional, or the environment. So a lot of focus is given to risk in this particular standard. So it might have been used 8 to 12 times in the previous edition, but it has been used about 86 times in this current edition. The word risk and implementing risk is an important thing. Now, what is the benefit? Why should you do this? What does this 2022 offer, right? It increases the effectiveness of the management system. Okay, it's going to be applicable not just for the laboratory, or it's also applicable to the entire organization where the lab is a part of. And it has to be talking to each other at multiple levels, so they have to be uniformed. So it kind of brings about that clarity in the document. This decreasing the probability of an invalid test. Therefore, there is a safety to the patient and a good satisfactory customer at the end. Lab safety is a priority here, reducing potential harms to the patient in terms of reducing errors, to the lab personnel who's working in the facility, considering the biorisk that they would have, the public in general, and the environment as well. So all its means, it kind of looks at the safety. Does it apply only to the laboratories? No. The same standard applies to other healthcare services as well. It includes for diagnostic imaging, respiratory therapy, blood bank, and transfusion divisions, as well as the physiological sciences. So the same document in its existing form would be applicable to all of these, which means there may not be a mention of lab everywhere to say so, because it's going to be applicable for a diagnostic other facility as well. This kind of helps. Having this in a similar form in another system would also help in assisting in exchange of informations and harmonizations of methods and procedures across. So you could have them similar so that you can compare them and brings about more clarity in understanding a patient more than just a test. So what are the key changes? I'm sure this has been told multiple times. Almost every day there is a session at a point of time from December onwards more. And then every day there was a webinar to say so. And everybody knows that this is going to be in alignment with the ISO IEC 17025-2017 document, where the management requirements usually appears in the end of the document. And here also you would go in a line. In its structure, it applies exactly like the 17025-2017 document. That's the alignment it takes. Also, it includes the requirements of a point of care testing, which was previously in ISO 22870-2016 edition. With this, that becomes redundant or cannot be used any further. However, with people applying for POCT alone for its accreditation, it would be still valid till this document gets completely implementable or completely mandated to be implemented from the December 2025 onwards, right? Till then, you can go with this POCT ISO 22870 document if there is a laboratory which is going for accreditation for its POCT testing. And it also increased, they kind of has an increased emphasis on the risk management. So that's a change. So these are the key 3 changes that it would have in its structure, in addition of a new testing modality, which is going to become the future, as well as the emphasis being on risk, because everything is based on risk in this particular document. Now, this document also aligns, like I told you, with risk assessment. It takes up the ISO 22367 document. For the lab safety, ISO 15190 document. Sampling and transportation, ISO 20658 document, as well as the, of course, the format of this is going to be based on ISO 17025 2017 document. The content, if you look at the document as such, okay, there is a lot of clarity that brings into the content in the way it's been written, and there are certain verbs used where it says, when it says shall, it indicates it's a definite requirement. If it says should, it's a recommendation. If it says may, probably it's in a permission. When it says can, it's a possibility or a capability that can be considered. However, if you would see the document, many times you would see this shall statement is followed by a phrase called as appropriate, which means there is a flexibility in the document. It does not say that for everybody it should be shall only. Like I mentioned to you, when it says shall, it should be a requirement for sure. However, this shall does not mean that it should always be so. So there should be, as appropriate to the situation for the testing that is required, it may be applicable. I've just added one of the clause as an example, 6.6.3, where it would come at the end. Okay, so that it's been added to those. So if you would look at some shall, it will also have these as appropriate, which means there is a flexibility of its implementation. So it's more challenging for an assessor than an SSC now, because the SSC has all the liberty to do however they want to their laboratory, and an assessor should understand what the lab is talking, how are they doing it. So what is that why are they bringing this context, and why are they having this document, etc. So, these are the 8 clauses or sections of the ISO 15189:2022. The previous one had how many clauses? 5? Yes, where the 1, 2, 3 were the same, which is the scope, the formative, the normative references and terms and definitions were the same. I don't know if you can read. Oh yeah, you can read here. So the scope, the normative references and the terms and definitions are all the same. Okay. However, what we did have in the 2012 where, the quality management systems requirement and technical requirement. Now, what do we have here? You have a general requirement, structural and governance requirement, resource requirements, process requirement, and the management systems requirement. So the quality management systems requirement is now in the end, like I told you, the 17025 document, it is aligned to. And here you would going to see that in this, instead of a quality management system, it's now the quality systems requirement that you see. Now, are these completely the same? The scope is the same? No, we've added point of care testing. So therefore, there is an addition to this. Okay, is the normative references same? Almost yes, it's the same. ISO IEC 17,000 to 2022, and of course the 17025 2017. But if you look at the terms and definition, I'm so sorry about this. This I don't think anybody can read. I can't see it on my screen though. This one has 15 new terminologies added. Some of them are modified terms. Some of them are very new terms. For example, the bias, so the bias and the measurement bias is not there earlier. The clinical decision limits not earlier, okay? Or the commutability was not there, or the complaint, the consultant is not there. The external quality assessment is not there in the terminologies there earlier. In vitro diagnostic medical devices, not there in the previous system. So you didn't have quite a few of them which are included. The previous one had 27 terms and definition. This one has 32. Some of them removed, some of them added, some of them modified where the content looks more clearer. Okay. So quickly looking into, of course, the general requirements, let's go slide wise. So here in the general requirements, it looks at impartiality, okay, which is the same, however, it emphasizes more. Confidentiality, where it applies to both the internal and the external personals involved. Then requirements regarding patients. Here it looks at the patient's well-being, safety and rights as the most important thing, and they are the primary considerations that it looks at. So that's the general requirement. If you look at the structural and governance requirement, it looks at the legal identity. So whom should I hold responsible? Some things go wrong. The lab director, who is a key of the laboratory itself, whose responsibilities definitely required. The lab activities, including the POCT added into that. The structure and authority, giving the organogram completely in saying who's responsible for what. Objectives and policies clearly defined, does not clear, says that you need to surely have a quality policy there. And the risk assessment all of these are based on the risk assessment and risk management included in the system, where you need to establish, meaning you should look at the risk evolved at every level, and you see that it gets implemented. And once it is implemented, it becomes a responsibility of everyone who has instituted that to maintain it as well. So these three things are important, and the responsibility of seeing this happening is the lab director or somebody the person can nominate to or delegate to get into the responsibility. So evaluation and modifications based on its performance is done by the lab director. So the focus here initiates with risk. Definitely a very, very important move in this particular document. The resource requirement, it looks at every type of resource in terms of personnel, people working within and out of the laboratories. Facilities, the previous one would look at it as an accommodation. Here it specifically looks at facilities and environmental condition. The previous document would look at as lab equipment. Here it would specifically say only an equipment. Calibration and meteorological traceability, reagents and consumable, all kinds of service agreements that the laboratory does with the other personals that they would be involved in, and externally provided products and services, including the referral labs that you would have. So all of these are addressed in the resource requirement. The process requirement is on the performance of the test itself, looking at the pre-examination process, where the test request form, the TRF we've been mentioning, has been mentioned as an examination request now. And you can also give an oral request. You need to document the way you will capture the oral request, because you need to know how this is being done. The examination process, including the verification and validation of the test that's been introduced. Post-examination processes, including the storage and handling of samples. It doesn't mention it as retention. It mentions as a post examination handling of samples, which means how do you handle them, how long you hold them, what do you do to them, where do you store them, how do you discard them as per the biomedical waste rules. Control of data and information management. So more is discussed about this, because this is how it's happening in most places. There are still laboratories doing manually, but many have a hybrid mode where you have a manual and LMI, you have the LIS system there, or you would have to include the importance of a cyber security, the way you will handle your data, how the confidentiality is maintained at multiple levels, what are the contingency plan whenever there is a when wrong, okay, and how do you capture the downtime, and how do you do the continuity. So all of these are being discussed in more details. Of course, also is the complaints and looking at multiple levels of capturing process and addressing the complaint, because here again the focus is on a patient. If the patient is complaining something, that's taken really seriously. So the last is, of course, looking at the emergency preparedness planning in all its processes need to be looked at. The last one is the management system requirement, which used to be the quality management system, which now is under the general requirements. Then the management system documentation, including documenting all those which are internal and external. External includes maybe a regulatory requirement, the legal law, or everything that you would have from an external body that is applicable for the laboratory need to be documented. It also clearly gives the control of records, all types of records, and how long do you retain this record, how do you manage these records, and what do you do with these records later. And how do you create these records also are described in detail. Okay, action to address risks and opportunities for improvement as a continual quality improvement process, looking at quality indicators and seeing how you can improve everyday functioning, everyday activity as a continuous process of improvement in the facility. And then non-conformities and corrective actions as a requirement, where you don't look at it as a preventive now, where you're addressing all of them as corrective, right? So what are the other supporting documents that the lab need to look at? Just the ISO 15189-2022 does not suffice. You need to reference them to ISO 9001, which is a quality management system document, which is some other document where you take all your inputs from to create. However, the 17025 is taken from that, which looks at testing and calibration laboratories. The difference is that here you're looking at the medical laboratories. 22367 is for the risk management. 15190 is the laboratory safety. The 3501 is 5001 is the biorisk management. 20658 kind of looks at the sample collection and transport. 20914 measurement uncertainty, which is a focus in this particular document. 19011 internal audit of management systems. 17043, where it is important to look at the proficiency testing and EQA, because it is no more called as interlab comparison. It is specifically mentioned as a external quality assurance program only. And for the internal quality control, the IQC, you can define how many times, how you would do based on a risk that you would do in your own laboratories. And you can, so every assessor in the future is going to ask you about how did you derive this? How are you maintaining it, and how would you want to take it forward? And 22583 is about the POCT operator, and 27001 is looking at the information security or the cyber security. Quickly looking at the summary of changes that we see. The priority is for the technical requirements on how you perform the test, and the management requirement definitely appearing at the end of the document only. POCT is a requirement. Emphasis on the risk management. However, there is a flexibility given into how do you choose and implement them, and what method you choose, and how do you document. So it's in your free. So you can choose whichever method you want to do that. So because the most commonly done is, you're trying to take, risk assessment using the severity, the occurrence, and the detectability score. You could do it in whatever style works well for you, and then capture it the best way and continue to perform. Okay, then requirement has been now, some of them combined, some separated, some of them elaborated, some of them highlighted further. So if you look at the previous document to the current document, there is not much of a change in some of them, just that they are clubbed together and put elsewhere. That's it. Okay, but you need to know where this one is coming. Okay, it provides comprehensive guide for controlling the documents in terms of how to create them, how to amend them, and how to retain these records. By the way, no more it's called as revised reports. I mean, it's going to be called only as an amended report, even in our reports that we will be amending in the future. It's less prescriptive. It doesn't say this is how you should do it. It gives you a freedom and flexibility to do however you want, looking at your process and outcome and not looking at your procedures alone, right? So the quality manual is no longer a requirement or obligatory. You could have if you would want to, but it's not a must that you have. Requirement for a designated quality manager has also been eliminated from this. LIMS, I told you, the cybersecurity, the offsite management in case you're storing it in a cloud, the contingency and preparedness should be there. The most important is a strong emphasis on validity pertained to clinical decision making. This is something that has been emphasized, enhancing that wonderful connection between the lab as well as the clinician who is sending the samples to us. And that's what we've been talking so much through the day on how it is important that you interact with your clinicians to understand why a lab tests are performing or behaving a particular way. Okay, now that in 2022 December it was released, for any document to be implemented completely, the time given is 3 years, which means 2025
we
onwards.
Have got 2 minutes more.
Yes, done.
Okay, thank you.
So we only have I have 2 minutes and we have 2 years for us to implement this 2022 document. And for the implementation, it's very important that the key persons understands and orients themselves completely to the standard. They list the specific requirement of the lab because very unique for themselves, wherein you do a gap analysis, identify which are the areas which you need to address in the current existing practice of your 2012 to that of the required 2022 standards, need to have a transition plan in place and define timelines as well. Train all lab personnel on the standard, assign roles and responsibilities for implementation. It has to be innovative so that it gets implemented better. And it has to be very proactive to mitigate implementation challenges, because any implementation would take a lot of people finding it very difficult and very hard to understand and implement. So you need to have some innovative ways to do it, and because that is given easily in the document itself. So reassess and audit the facility to see your gaps and then readdress them. So that's how you need to do your implementation. That's how we have started our journey of 2022 in our labs. So the take home is that the ISO 1518922 is risk-based, very specifically patient-centered. It includes POCT, provides flexibility for implementation. You could do it the way you like to do. Okay, encourages continuous improvement of your test processes and contributes to the betterment of the healthcare system. Thank you very much.
Thank you, madam. Now it's time to take few questions from the audience.
Ma'am, on question, if there is any update on the concurrent document the NABL prepares, when it will come?
What is that, sir?
NABL 112?
It is getting prepared. It would be released soon, sir. There is one document getting prepared, yes. Inputs have been sought from a lot of people. Experts are working and it is going to come soon.
Thank you.
Excellent talk, Dr. JJ. Very simplified and very nice, very elaborate. Thank you.
So may I request Dr.
Thank you, Doctor.
The certificate to Dr. Jayalakshmi. A sincere request to all our esteemed speakers. Your cooperation in adhering to the allotted time is greatly appreciated and contributes to the success of SFCC 2023. We are confident that with your expertise and conciseness, we will have an engaging and well-paced event that benefits all participants. Thank you for understanding. So, moving ahead, we have swapped the talk of Dr. Rajesh Bendre with Dr. Wahid Ali. So may I request Dr. Wahid Ali to be our next speaker, please? And extremely sorry, Dr. Rajesh, and so courteous about him that he's good to swap the lecture. Thank you.
Thank you, Mindray and Shama, and moreover, Mr. Manoj, who is coming to our lab. I am working as a professor in the Department of Pathology, as a unit head of clinical chemistry section. And choosing this topic Actually, since long, I am working in the field of clinical biochemistry, as well as my area of interest is lipid biochemistry. Moreover, most of the laboratory we are doing lipid profile as a traditional test. So should we go ahead still with traditional lipid profile, because I am working in KGMU and cardiologist many time asked me a question, Dr. Wahid, our lipid profile report is quite normal, quite okay, triglyceride is okay, total cholesterol is okay, HDL is in the good range, but even if the subject is suffering with a recent MI or cardiovascular event. So what is the thing behind whether the traditional marker we should think on that, or we should look for some other newer markers that is upcoming. So considering these things, I have chosen this topic, that whether the good cholesterol is still good or it is something else behind the scene, behind the curtain. We are okay that our good cholesterol or HDL is beyond 50, beyond 65. I am thinking that our HDL level is okay. I am prone to with cardiovascular event. I am protected. I may not have the recent cardiovascular event in future. Looking that, we have prepared this slide that the burden of cardiovascular disease looks like that. We are overburdened, and the cardiovascular disease is the leading cause of death globally, and approximately 17.7 million are suffering every day globally with the cardiovascular event. And in that, high density lipoprotein serve as a protective response, whereas low density lipoprotein cholesterol as a risk factors for coronary artery disease. This is the leading cause of death globally. If we see the data from 2000 to 2012, we could see here that the increasing rate of incidence of stroke, ischemic heart disease, and respiratory disease along with diabetes, they are increasing day by day. And the risk factors that attributed about more than 50% of total risk is due to the lipid factors, or lipids and lipoproteins. And we know that there are some risk factors, they are modifiable risk factors, and some are non-modifiable risk factors. We can easily modify by active lifestyle intervention, dietary changes, and further things like dyslipidemia, raised LDL cholesterol, triacylglycerol disturbances, low high density lipoprotein cholesterol, smoking, hypertension, diabetes, obesity, PCOD, etc. Whereas non-modifiable like genetic risk factor, age, sex, family history, and other things. We can define dyslipidemia that there is a change in the cholesterol level, in the triacylglycerol level, as well as there is a change in ApoB content of the lipoprotein, LDL, and LPA, that is recent marker, that is upcoming marker, and concentration of the HDL like ApoA1, that is HDL associated proteins, that is ApoA1, and these disorders may be primary or to secondary with the disease. This is the evolution of atherosclerotic plaque. I am not going in detail due to constraints of time. There is a relationship, direct relationship between HDL cholesterol and incidence of CHD, and the level of HDL more than 75 associated with longevity or receipt of longer healthy life. Thus, the goal is that the primary reduction in HDL cholesterol are in common patient with premature CHD. Low HDL levels are seen in patient with first myocardial infarction in about 19%, whereas some study shows that the 52% patient with CHD with normal LDL and low HDL cholesterol. Various studies shows that the like CARE studies and some Birmingham heart study shows that myocardial infarction increases by 25% for every 5 milligram per deciliter decrement in HDL below median values. Data showed here that the risk of myocardial infarction increases by 25% every 5 milligram percent decreased in the HDL level. And NCEP ATP third panel shows that HDL level below 40 constitute an independent risk factor for coronary artery disease. Just to show here how the structure of HDL particle looks like, that is ApoA1, ApoA2, cholesterol ester and triacylglycerol rich particle. This is the denser particle, smaller in size, and the production of HDL cholesterol by de novo and by the reverse cholesterol transport. So HDL as antithrogenic mechanism by two mechanisms, that is the reverse cholesterol transport from peripheral tissues to again the liver, and cholesterol ester transfer protein that transfer cholesterol from VLDL to IDL, LDL and cholesterol ester transfer protein. This is the mechanism how the reverse cholesterol transport system happens, and cholesterol ester transfer protein play a major role in the production of reverse cholesterol transport and VLDL. Now here the scene is that whether at the level of 50 or 60 or 75 milligram per deciliter is still protected or not, where our functional HDL that protect us from the incidence of MI or cardiovascular event, whether it is still considered as a good or something else is going on. Then the functionality aspect of the HDL should be assessed. Of the HDL theory or studies in the HDL particle started since 1951 and going on day by day. And there is a myth that HDL cholesterol levels means lower cardiovascular disease risk. It means that if your HDL level is beyond above normal limit, it means we are protected, whether it is correct or not. The answer is that the HDL cholesterol levels is only a part of the HDL story, because we have to look into the particle, we have to look into the content. The particle matters whether the functionality aspect of reverse cholesterol transport system is going on or not, properly it is done or not. Then it does not give an indication if the HDL particle is functioning properly or not. Here an study has been taken, that is the HDL assumption called into question by multiple studies from 2012 to 2017, that there is a U-shaped curve of HDL concentration. Means at below 40 there is increased hazard ratio or mortality, whereas when the HDL concentration is beyond 70 or 80 milligram per deciliter, there is also increased hazard ratio. It means ki if your HDL is below normal or above normal, it means ki you are at equal risk, you are at similar risk. Where there are various trials, they are raising for HDL cholesterol did not reduces the risk of cardiovascular event, like having very low or very high HDL levels indicate to increase the mortality, and raising HDL cholesterol levels with different drugs like niacin and other factors, they fail to reduce the risk of cardiovascular event. Moreover, the story is not only for HDL, it is as applicable for LDL as well as triacylglycerol as well. That here the study was conducted by Sachdeva et al. In 2009 and many other studies, that most people who develop CHD, they may have normal LDL cholesterol. And we can see here the incidence of CHD with the persons they are suffering with CHD, that is at the level of 130, below 130, and at the level of 70 milligram per deciliter, the incidence of myocardial infarction is still high. And 23% had LDL cholesterol beyond 70 milligram per deciliter. So what does it mean? Whether normal LDL or low LDL is okay, or we have to look into the matter, whether behind the LDL there is some other fraction like a small dense LDL that is more atherogenic to develop atheromatous plaque. Same is here, like that triglyceride level patient done on the analysis of 1,36,955 hospitalized patients, and showed here that the 60% patients with the myocardial infarction have shown the triglyceride less than 140 milligram per deciliter. And the scene is also looks like that. If 52% had HDL cholesterol more than 40 milligram per deciliter. So what is the mechanism? What is the theory behind that? At the level of normal LDL, above normal HDL level, below normal triacylglycerol, the incidence of CHD and cardiovascular disease is still very common. So behind that, one theory has been given that the small dense LDL particle, LDL has some subfraction, isoforms of the particle, that is a small dense LDL and large band LDL. The small dense LDL behind the total LDL, that is the major player in the risk assessment and risk development and leading to the cardiovascular event. And the reason is behind that the lower affinity to hepatic LDL receptor, a stronger affinity with the vessel wall, and more rapid oxidation, accelerated oxidation. Your LDL is normal, but that LDL is become oxidized, that is more atherogenic, more serious event is there. We can see here, and we can very well compare that at both the level, one patient is healthy patient, another is CAT patient. Here the level is 140 in both the cases, and you can see here in healthy men the small dense LDL cholesterol level is only 20 milligram per deciliter, whereas in CAT patient the small dense LDL level is about 90 milligram per deciliter, means equal at the equal level of LDL cholesterol. One person is with cardiovascular event and another person is healthy. It means there is a small dense LDL that is the major player in the development of cardiovascular event. Here we can see that normal size, normal number, no risk. Large number of LDL with normal size, intermediate risk, and small dense LDL but a small number, low risk. Large number of small dense LDL at a high risk. So we should think whether our total LDL is responsible, or in behind the LDL, how the concentration of the small dense LDL they are behaving. What is the level? What is the concentration of small dense LDL in between the total LDL? Now the theory again comes to the HDL, no more as a good cholesterol. Why? Because there is a recent theories that shows that Birmingham studies of incidence of coronary heart disease shows that 44% of the event occurred in men with HDL cholesterol levels of at 40 to 40 milligram per deciliter, whereas 43% event at 50 milligram per deciliter. It means that a significant number of CHD event in patient with normal LDL and normal HDL cholesterol. So therefore, we have to search, we have to look for the novel markers, for the recent markers that is upcoming. So quantity does not reflect the quality. Irrespective of quantity, what is the quantity of our LDL or what is the quantity of our HDL? It is the functionality, whether the particle that is as well functional or not, whether the reverse cholesterol transport theory is still working or not. So HDL function can be assessed through the number of HDL particle, various size of HDL particle, integrity of HDL particle, as well as some inflammatory markers, that is myeloperoxidases and oxidized low density lipoprotein cholesterol, that has a major role in assessing the HDL functionality. There is pro-inflammatory theory, that is that ApoA1 convert into HDL into pro-inflammatory, that is become non-functional. Macrophage retain increased cholesterol, and by reverse cholesterol transport is reduced, and myeloperoxidases modifies the tyrosine amino acid residues and that become dysfunctional HDL has increased hydroperoxides and make it anti-inflammatory to pro-inflammatory. I have to skip few of the slides due to paucity of time. The heterogeneity of HDL in component renders these molecules very susceptible to alteration in various aspects like cholesterol transfer protein, SR beta 1 scavenger receptor beta 1 protein, ABCA1, PON1, paraoxinase, LCAT, ApoA1 and ApoA2, and free cholesterol, cholesterol ester, hydroperoxide, phosphatidylcholine, membrane phospholipids, and other factors that are associated with modification of or attenuation of HDL particle. So here we can see that the physiological HDL become anti-oxidative, anti-inflammatory, anti-apoptotic, and anti-thrombotic, and after become it is dysfunctional or it become attenuated, become dysfunctional, that become pro-oxidative, pro-inflammatory, pro-apoptotic, and pro-thrombotic activity. There are various conditions where dysfunctional HDL could develop, like in cardiovascular diseases, in metabolic diseases, in chronic inflammatory conditions, in kidney disorders, and in various other conditions where HDL functionality become disturbed. And in coronary artery disease, why the reduced HDL or functionality, dysfunctional HDL is associated with reduced PON and HDL activity, reduced inhibition of monocytes, reduced inhibition of oxidation of LDL, and various other factors that are responsible for HDL functionality. And here we could see here. Now, considering these factors, we have chosen two markers like PON and SR beta 1 as a pilot study in our King George's Medical University, collaborated with cardiology department, and we have recruited few of the angiographically proven cases of coronary artery disease and study will show how the result we have find. So PON1, it is an HDL bound aryl esterase enzyme that is antioxidant particle that is associated with the HDL particle, and it able to hydrolyze oxidized or altered lipid. It protect HDL from lipid peroxidation, and decreased PON1 activity has been proposed because dysfunctional HDL, it is premature atherosclerosis, and production of advanced glycation in product is also in hyperglycemic individuals as seen in type 2 diabetic patients. Whereas scavenger receptor beta 1, it is important enzymes reverse for cholesterol transport, and it is mediated for cholesterol facilitated bidirectional cellular cholesterol efflux, and hepatic overproduction of SR beta 1 reduces HDL level and lowers chance of atherosclerosis. These are the research tools how whether we could do the PON1 activity. We would simply in the serum we could analyze the PON1 by ELISA method, that is that test kit is available, and we do by our experience on testing on PON1 and SR beta 1 in coronary artery disease patients. This is just a pilot study, and the aim of the study was to determine the serum level of two novel HDL associated markers like PON1 and SRB1 in coronary artery disease patient and in control healthy individuals. And the study design was case control. It is of 1 year, sample size 92 subjects, suspected of CAD, 69 were angiographically proven CAD, and 23 were age-sex match control, healthy control. Serum PON1 and SR beta 1 along with other biochemical marker were done. And patients were further subdivided into 3 categories, like category 1 means have single vessel blockade, category 2 with double vessel blockade. Yes, sir, I have to conclude. Yeah. And 3 multiple vessel blockade were there. Like this, the study was planned, and we find here that the age, sex was significantly different, and we could see here that BMI and all these things were not very much significant. And here we could see that the PON1 and SR beta 1 was significantly difference was observed, whereas we could see here that there is significant difference was found in HDL level, but there is no significant differences was noted in control in comparison to cases for LDL as well as for VLDL, and triacylglycerol, there is no significant differences. Means lipid profile were still okay. And when we compared with the category wise with the control, we could find that the control with category 1, category 2 and category 3, there is significant differences was noted. There is no much significant difference with the habit and like alcohol use, family history, obesity, hypertension, etc. There was no much significant difference was found. And this is the sensitivity and specificity, how the case versus control we could find here that the cutoff value for PON1 and SR beta 1, and it is found significant specific, 100% specific and 75 sensitive as a biomarker. This is the relationship, dot plot relationship with the control PON1 and category PON1, category PON2 and category 3. This is the ROC curve. And conclusion that serum level of HDL, PON1 and SR beta 1 are significantly lower in CAD patient. It was inversely related to increasing severity of vessel blockade. Serum PON1 SR beta 1 could be used non-invasive tool for identification of vessel blockade and risk assessment of CAD. And further assessment of these markers could correct the functionality of HDL aspect. Limitation of the study, smaller sample size, single center study, and recommendation larger sample size to be taken for further validation. Angiographically proven cases in control that will increase the sensitivity and authenticity of the study. Summary of the test ordered till date, like lipid test routinely ordered 90% HDL, LDL, TG, half of the respondent only LPA, ApoB and HSCRP. LPA, ApoB were found to be more common in more lipid-centric physician. Lipid parameter most predictive that non-HDL ApoB believed to be same. However, ApoB is ordered less often. Take home message for this pilot study was lipoprotein particle. Small dense is more prone to increase atherogenic genesis. Small dense LDL is common in CAD patient even with low or normal LDL. Oxidized LDL is more susceptible for plaque rupture and native LDL. Thus, for those subjects who are at increased risk, must evaluate for LPA, ApoB, ApoA1 and OxLDL. Serum PON1 and SR beta 1 could be used as a non-invasive tools. Further assessment of these markers could tell the in-depth aspect of the functionality aspect of the HDL particle. And with this, I would thank the organizers and thank the chairperson for giving me some extra time. Thank you, sir.
Okay, thank you very much. So we will just go ahead like that. We'll not have the audience session. One or two questions we can have if anyone.
Very good talk on lipid profile. Mostly, I don't know how many people can follow this in future in the routine laboratories.
Yes, sir.
Difficult.
Because yes,
so this is not a question, a comment. Since time immemorial, we say good cholesterol and bad cholesterol. Is there any cholesterol like that?
Yes.
You have only cholesterol.
Yes, sir.
So the cholesterol attached to HDL we call good and the other is. So we use it. Sometimes we use cholesterol as HDL, sometimes HDL as cholesterol like that. This use is going on since a long time. Only a comment.
Thank you.
Okay.
Thank you. Actually, most of the things that we see here in the day-to-day life, our report goes to the clinician. Clinician asks, your lipid profile is normal, but the patient at the age of 20, 25, 30 with the recent MI, what is the scene behind that? Scene behind that, the thing is theories has been changing. No history of tobacco, no history of smoking, nothing, and all of a sudden there is MI. It means that there is some genetic factor that is responsible, and our study population should go for screening for further high risk individuals for these. If these biomarkers is available, we could do, and who could afford, most of the important thing is that who could afford, they could evaluate for this novel marker that is upcoming.
Okay, thank you. As you said, there is no good cholesterol, no bad cholesterol. Cholesterol. And moreover, that's what I teach my students also.
Yes, sir.
Don't say good or bad.
Yes.
It has got a very different connotation for saying a good cholesterol, bad cholesterol. And moreover, as we said, it has already time since more than half a century we are going on with the lipid profile. It's a high time that we should move ahead with newer parameters and markers. Thank you, sir.
Thank you.
Any question?
Yeah, my question is, we are getting a couple of prescriptions for testing small dense LDL.
Yes.
So what's the method for testing the SEM and who is doing it?
There is a dual precipitation method. First time it was given by Hirano et al. In 2004. It was a Japanese scientist. It was a manual method for dual precipitation by sodium heparin citrate method. Nowadays it has been developed by Randox, and they are making the kit for the small dense LDL as well as PON1 and SR beta 1. So, and they are fully automated biochemistry analyzer, they are compatible. And the cost per test, I think more over 500 to 600 rupees per patient.
Thank you.
Thank you very much, Dr. Wahid Ali. May I request Dr. Mohanty to hand over the certificate to Dr. Mohanty? One more question? So may I request Dr. Mohanty to hand over the certificate to Dr. Wahid Ali? Thank you, Dr. Mohanty. May I request Dr. Ramesh Kumar to chair the next session, and may I request Dr. Rajesh Bendre to please come up on stage.
Thank you, Shama and Mindray team. I have great pleasure in introducing my ex-colleague and a close friend, Dr. Rajesh V. Bendre. He's a MD Path, DNB, and DPB. He's presently working as the Chief Pathologist and National Technical Head at Apollo Diagnostics, Hyderabad. He's a postgraduate from Grand Medical College, Mumbai, has technical work experience of more than 25 years in a standalone and a hospital-based and a reference labs handling clinical chemistry, hematopathology, and molecular pathology. Academic teaching experience for MBBS and MD or DNB path postgraduate students for more than 10 years. 5 international publications and participated through posters, oral presentations in many national and international conferences. His special interest in autoimmunity, flow cytometry, hormonal and drugs assays by HPLC, LCMS, and trained in management of component blood bank and apheresis services. He's accomplished NABL CAP assessor as per ISO 15189 of 2012 and ISO 15189 of 2022. Trained in laboratory management through IIM Calcutta and IIM Indore. Acquired skills in development and implementation of laboratory information system. So Dr. Rajesh is here to speak on tumor markers. Rajesh, floor is yours. Thank you.
So very good afternoon, everyone. And at the outset, I thank Mindray to providing me this opportunity to share some of the thoughts on this topic given to me, which is serum tumor markers as per current clinical practice. Now, this is a very huge topic, and I hope I would be able to do justice to this topic. So starting right away, so taking some introductory and setting some background to what I want to present. So when we talk about serum tumor markers or biomarkers, what are they? So they are basically substances which either the tumors produce or they could be various host reaction and proteins of the host reactions that we are looking at. In either which ways, we want to get some biological characteristics of the tumor using these biological markers. We may measure them either quantitatively or we measure them qualitatively. In terms of our understanding as to what is the nature of these substances. They could be antigens, which are secreted by the tumor and released into the periphery. They could be hormones secreted by the tumor cells. They could be enzymes, or they could be tissue-specific proteins. And in today's era, which is the last part by which I would like to conclude, is the nuclear proteins, which is the next future that we would be talking about as various signature molecular proteins and liquid biopsy. And that is where I would like to end. In either which ways, whatever are the characteristics of this particular substance, we would like it to be as an ideal biomarker with reference to that particular tumor. So does an ideal tumor marker exist in today's world? And what is it that we would like to have when we use the word as an ideal tumor marker? So we would like this tumor marker to be highly specific. So when we say high specificity, we would like to have, in terms of numbers, it should have a specificity to as high as 95%. It should be highly sensitive. So that means it should be at least 90% sensitive. It should have a very established medical clinical decision-making cutoff. And that cutoff which a clinician can use either to differentiate between a normal or a healthy versus a abnormal or that is a cancer patient. And the same marker he would also like to use later on as a follow-up or during monitoring. And last but not the least, which is purely our domain as laboratorians, that we would like to have the methodologies for these various markers, and all those methodologies we would like to have it very nicely standardized and harmonized. So that is what we would define, or we would wish to have something like an ideal tumor marker. So do we have one, and are we there? Let's discuss on that very, very quickly. So there is a whole gamut of list of tumor markers that we know today, and they have evolved over a period of last, say, 2 decades. And that's a whole list which we all know, which often always are even specific to certain cancers. In fact, there are various recommendations as to how they should be used as well. So not all of them can be used for screening, diagnosis, and monitoring. So each of them has its own utility and a purpose to be used in a clinical setting. But is it really followed as per the recommendations and guidelines? Again, a big question mark. The big example here I would like to quote, in my experience being abused or misused, is CA 15.3. CA 15.3, the recommendation is purely as a monitoring marker and for CA breast, but we often see this being used for diagnostic purposes. And here is where all the disconnect starts, and a lot of queries that we need to handle, both from a patient side and from the clinician side. So that is one message, that whatever list of tumor markers that we talk about, there are standard guidelines and recommendations as to how we should be using it in terms of diagnosis or monitoring, and we need to ideally completely follow those guidelines. So having set that background, now let me run through individual tumor markers that we know today and talk about certain advantages and limitations of each of them in this short time which is given to me. So taking the first one, which is more or less a more time-tested one, which we all know, it's CA125. So serum CA125 is almost, we corroborate it with a marker for CA ovary. And yes, definitely true, we know that CA ovary, when we talk about, and if you look at the WHO classification for CA ovary, it is humongous, it is huge, it's like a Pandora's box. And out of the so many lists of tumors that is possible in ovaries, the one which is really related to CA125 is only one histological type, and that is serous surface epithelial tumors of ovary. So that means that you may have a patient with a huge ovarian mass, but the CA125 report can come perfectly normal. It just means that this patient is probably having an ovarian tumor which is of non-serous type. So that's one very important point, that when we say that CA125 is related to CA ovary, it is with the reference that serous surface epithelial tumors of the ovary are the commonest variety. And this is the marker which is raised in this particular type of tumor. Having said that, it is not, though it is something specific for this particular histological type of CA ovary, it is still not a specific marker for CA ovary. And the reason for that is, if you go back to the embryology as to what this protein is, this is a protein which comes out from mesothelium. And all the cells which are derived from, which are mesothelial derivatives, they are the ones which can secrete CA125. So when I say this, now you can go back to your histology anatomy and you can understand that the ovarian capsule, the peritoneum, the pleural surfaces, they all have mesothelial linings. So any conditions which irritates the mesothelial lining here will cause a rise in CA125 apart from the ovarian tumor. So that makes CA125 less specific because of this very biological nature of the protein. So if you look at the literature, that's what it comes out to be, that's what it comes out to be, that in the screening when you use, it has got about 83% of the patients can be detected for epithelial ovarian tumors, and those are the sensitivity specificities, and the number which gives you the maximum sensitivity and specificity is 65 and above. If you recollect, our reference range is more or less 35. So we are talking about 2 times the upper normal range, a number is likely to be most significant in favor of a possible ovarian tumor. A further very nice guideline which ACOG and SOGC has given up in terms of interpretation of this single number that we get as a serum CA125 is this. You should take the patient's age into consideration. If the patient is premenopausal and in the reproductive age group, any number that is about 200 is to be considered significant. But if a person is postmenopausal, any number more than 35 is to be considered significant. Very, very clearly defined by ACOG and SOGC guidelines. And the reason that they are talking like this is the same background the comments which I made earlier, that there are more possibilities of other benign conditions, infective conditions, which can also give rise to rise in CA125. Now, can we make this marker more stronger and more specific? So the answer to that is yes. We can make this more specific and more stronger in terms of identifying CA ovary, and that is if I complement it by using other biomarker. And one of the other biomarker which has been used is what is called as HE4, which is human epididymis protein 4, which is being documented to be overexpressed in majority of the ovarian tumors, especially epithelial ovarian tumors again, and that is how it is being used in conjunction as a complementary marker with CA125, and you have a software algorithm which would statistically give rise to a risk calculation in favor of malignancy and not in favor of malignancy, which is what is referred to as risk of ovarian malignancy algorithm, or ROMA. The cutoffs which are recommended, as you can see here, again we have to take into consideration whether it is premenopausal or postmenopausal, and the cutoffs as 13 or 28 as a combination of 2 biomarkers, CA125 and HE4. There have been another similar study also where more number of biomarkers were used in combination with CA125, and that was called as OVA1. And there are comparative studies between ROMA and OVA also available in our literature, where it has proven that ROMA seems to be a better statistical model rather than OVA1. So this is one way by which CA125 specificity can be improved by using complementing markers, especially HE4 and ROMA. Okay, so that was some points to highlight specifically on CA125. I shift to the other very commonly used marker, serum marker, which is PSA. Now PSA, again, like CA125, yes, it is an enzyme which is secreted by the prostatic epithelium and prostatic epithelium only, and thus it becomes very, very specific to prostate. The normal function hypothesized of this enzyme that it facilitates in liquefaction of semen. This particular enzyme, which is secreted by the prostatic epithelium, circulates in our serum combined with certain proteins. And that is how, like our thyroid, the concept has evolved as total PSA and free PSA. So the part of enzyme which is not protein bound is the free PSA, and that's the next slide which I am going to show of a combination of total PSA, free PSA, and we talk about a ratio between the two, which makes it more specific in identifying a possibility of a CA prostate instead of a standalone total PSA alone. As it is an enzyme, one must realize and always keep in mind that any infection in prostate any infarction in prostate, any benign enlargement of prostate will also cause rise in this enzyme. And very, very important point, this is our pre-test limitation. Any manipulations in terms of per rectal examination or any procedures, we should not be doing the test immediately for PSA, which will give rise to erroneous or false increments in PSA. This is worthwhile to remember. If there is a biopsy which is done, the patient needs to wait for at least 6 weeks before a test of PSA should be prescribed or should be done. I don't think most of the clinicians or even our own laboratory people are aware of this particular pre-test requirement before we collect a sample for total PSA. So these are some very important points that we need to bear in mind. Another thing which I want to highlight is this table. Generically, we always say as a reference range for total PSA as 0 to 4 nanogram per ml, by and large. But if you look at the literature, as age advances, your PSA levels also increases. So especially in elderly patients, if you are doing the PSA for the first time, this is something which we must also keep in mind, the age and the total PSA level that we are looking at. Okay, as I said, we can make the specificity of this marker better by combining it by using with free PSA, and we take a cutoff of 25% as a ratio between free PSA to total PSA, and that's what the literature says, that if you have this ratio which is less than 15%, it is the message to the clinician. If when you look at this particular ratio, the grid or the recommendation, it goes like this, that it may warrant a biopsy and the risk of possibility of a CA prostate is in the range of 28 to 56%. If the ratio is 15 to 25%, the risk is 12 to 19%. And if the risk is about ratio is about 25%, that you may avoid a biopsy, just follow up by a DRE, and the risk it becomes as low as 8%. So that's what the cutoff of 25% is what is recommended as a ratio between free PSA to total PSA as a guideline to interpret the ratio. The same total PSA can also be used as a monitoring tool in a diagnosed case and after post-surgery. And here the cutoff is what I wanted to highlight. The cutoff what we must be looking at is 0.2 nanogram per ml. Is 0.2 nanogram per ml. So if you are having a patient of total PSA as a follow-up, so that is where we should be careful. If you have a number of total PSA in a follow-up patient post radical prostatectomy, if it is going 0.2, this should be our laboratory alert to the clinician that this warrants further investigation. It warrants probably a recurrence or probably a resurgence of a metastatic lesion somewhere. Okay, going further to the next serum tumor marker, which is alpha-fetoprotein. So in alpha-fetoprotein, the points which I wish to highlight, number one is the units. There are various kits available. Some kits give us in terms of IU per ml, some kits give us in terms of NG per ml. So it would be best and a good laboratory practice that if we put the conversion factor in our report format, so even if the patient follows up from different labs A to B, the clinician is able to convert and correlate it appropriately. That is one very important point. Alpha-fetoprotein is a very important marker, especially when it comes to liver diseases and when it comes to hepatocellular carcinoma. But please remember that AFP can also rise in non-liver conditions, and mainly it is the germ cell tumors where it plays an important role as a tumor marker. In the previous lecture, Dr. Barnali was mentioning about clinical reportable ranges, and we just spoke about a point where do we have to report values beyond the AMR or beyond the dilution protocol given by our manufacturers. This is a classical example, and I will relate to that discussion which Dr. Barnali made. Now, clinician for this particular marker will definitely not agree and will make you give you the endpoint titer. And the reason for this is the last sentence, that this is a marker which correlates with the tumor burden. And that's the reason the exact number is very, very critical for the clinician to know. So this is one tumor marker where clinician will never agree and he will always want us to give us the final number. So yes, I do agree with what Dr. Barnari advised us, that in terms of medical legality, we cannot go as beyond the kit insert or manufacturer insert, but we need to comply by what the clinical need also is. So what I would follow in my practice, which I do follow, is I would give the first report as above whatever maximum dilution. In the remark, I will say further manual dilution done as per clinician's request and give whatever the number is. So I comply by both the requirements, medico-legally as well as the clinician's need. AFP again, and when you talk about hepatocellular carcinomas, again there is a catch. There are almost 1/3 of the hepatocellular carcinoma tumors which do not secrete AFP. So what do we have for them as serum tumor markers? So we have certain other biomarkers here, and the other biomarkers that we have are these two. Now AFP, if you look at from an electrophoretic point of view, it has got 3 subfractions, and what is considered is you take L3 fraction is the one which is especially increased in these other hepatocellular carcinomas or non-AFP secreting hepatocellular carcinomas, and a ratio between the total AFP and L3, if it is more than 10%, this is one way of identifying them. Another marker is DCP or PUCA2, which is also called as your protein induced by vitamin K absence or an antagonist 2. So this is another novel marker. ELISA kits of research only use are available, and that can be also used in such cases which are non-AFP secreting hepatocellular carcinomas. So the next tumor marker is CEA. In CEA, one must consider that the reference ranges for patients who are smokers, patients who are having inflammatory bowel disease, chronic lung diseases, the cutoff then is considered an elevated cutoff of 10. If all these predisposing factors are not there, then a cutoff of 3 can be considered for CEA. So that's a very, very important point which should be there as a part of our report formats. CEA is a very, very important classical marker for definitely a colorectal carcinoma, both for diagnosis and also from prognostic point of view. When we talk about it in terms of post-chemotherapy in a colorectal carcinoma, the cutoff considered is 6 nanogram per ml. So I think what I'm trying to do, I'm going a bit fast, but what I tried to compile here is these cutoffs. I think as a laboratorian, we need to know these cutoffs when we are reporting our tumor markers, and as we correlate with the history from the clinicians or the patients, this is what we need to correlate and discuss with the clinician accordingly. This is what would give a value add, and that is what I suppose the clinician is also expecting. CA 19.9, now this is an extremely notorious and a tricky marker, I would say, and the reason being that the nature of this marker is that it is a mucin, and because that it is a heterologous mucin, the assays are poorly standardized. A very, very notorious assay, Vidas, Roche, Abbott, huge variability you will see in this CA 19.9. A patient who is having a rise in CA 19.9, and because it is a mucin, it also causes interferences in lot of our other immunoassays and other tests. So that is one thing which we must keep in mind. You can have abnormal peaks causing in protein electrophoresis because CA 19.9 is raised, and there are published cases on these. So these are certain issues with this particular marker, but yes, this is the only marker that we have in order to diagnose CA pancreas, and that is one of the important markers for CA pancreas for us to pick up. Okay, another very commonly used marker is beta HCG, and here I want to highlight, like CA 19.9, though it is one of our commonest marker, the molecule beta HCG circulating in our serum is extremely, extremely complex. And if you look at what all circulates as a form of beta HCG, are so many different forms. And our kit that we use, does it target all of them? I don't know. I don't have an answer. And if we really compare, I think there will be a huge variability between all our standard kits that we use in terms of what are they targeting as a molecule, as an immunoassay. And this itself causes an inherent variability between our results across the lab and across the testing platforms. So very important point that one must keep in mind. And for this, obviously, scientifically, we will never have an answer to this. But what the message I think we should give as laboratorian to the clinician or to the patient is follow one lab. So that is the best way that the clinician does not get misled or the patient also does not get misled. So that is one point. And the same thing is corroborated. I have pulled out a CAP participant summary here. The same CAP sample, which is run across the world in different labs, done by Siemens is 702. The same sample done by Vitros is 1844. So this itself tells the fact that they are not standardized, and you have so much of variability from 700 to 1800 from a same CAP sample tested in a survey. So this is one point first upfront that I would like to highlight. Going ahead, when we report in terms of beta HCG as a tumor marker, we all know this is a tumor marker very, very important in gestational trophoblastic diseases. And this is one disease where it's a single chemotherapy methotrexate which makes wonders and works wonders. But a very critical point is this, that what the clinician is looking at in a follow-up beta HCG, once a methotrexate is given, is this, a 1 log reduction within 18 days after the first treatment. This is what they are looking at. And if this is happening, means the patient is responding. If this is not happening, means either they have to step up the dosage or maybe they'll have to add certain other chemotherapeutic agent. So I think this is a key that we should be looking at when we are reporting it. Another problem that I have also faced many a times is unpredictable transient rises in beta HCG when the patients are on follow-up. And this is especially known in patients with molar pregnancy or patients with choriocarcinomas. As choriocarcinomas, as we all know, histologically has got extensive areas of necrosis and hemorrhage. So tumor necrosis as a phenomena is common in choriocarcinoma, which can give rise to this transient rise in beta HCG. And this can cause a lot of interpretative challenges and problems when in a follow-up patient. So there is published reports on transient rise in beta HCG follow-up in such cases, so wanted to highlight that point. Chromogranin A is another very important novel marker which has become a very important marker now for almost all neuroendocrine, suspected neuroendocrine tumor in the body, ranging from carcinoid to your small cell carcinoma lungs to pheochromocytomas. So for any neuroendocrine tumors, this has become a very important novel marker. So here, again, the cutoffs, if I have to highlight, the reference range, what we would consider is 60. The reference range here, what we would consider is 100. But when we are talking about a neuroendocrine lesion, usually it is 2 times the upper normal range, that is about 200, is what we must be considering as significant. The reason why it is almost 2 times the upper normal range, the answer to that is this. That this marker, again, there is a prerequisite that we must be looking at. Patients who are on H2 blockers, our simple antacids which people take without even non-prescription antacids that everyone takes, that can interfere and cause false erroneous rise in chromogranin A. So patients should be off any antacids before they are giving the sample. A very, very important pre-test requirement that we must be looking at. Thyroglobulin and calcitonin, again important markers for our thyroid carcinomas, medullary and follicular carcinoma thyroids. So here what I want to highlight is about thyroglobulin. Thyroglobulin, one notorious point is some technical challenges here. And technical challenges is that there are interferences with this assay, especially if there is a concurrent presence of anti-TG antibodies. And if there is so, we should be careful about it, we should be aware about it, and accordingly report with a rider if that is positive along with the whatever value that we are getting for TG. Okay, so having run through some important specific individual tumor markers and some salient points on them, coming to this. Why do you want to have such huge battery of different single markers? Can I have only one universal single serum biomarker? And the answer to that also is yes. There have been a marker which came into existence as FDP-DR70, and this was extensively studied, especially in stomach and colorectal carcinomas, and it showed very, very promising results. And then it was also extrapolated for all other cancers, where you can see there is some variable sensitivity and specificity, the way it behaved. So finally, FDA gave approval to this particular marker as a universal cancer marker, but FDA gave it as an approval for monitoring purposes only, and that too for CA colon only. So this is what was the story was of this marker, but people misused it, and there were a lot of litigation cases, and currently this marker again is withdrawn by the company because of the litigation cases. And the reason is not because scientifically this marker is wrong. The reason is it has been misused for diagnosis and screening purposes, which it was not supposed to be done. It was supposed to be done only for monitoring and in cases of CA colon, but unfortunately it was used for something else. Since it is a FDP, one should also take into consideration other causes, other non-malignant causes, which can give rise to rise in FDP before we interpret this particular marker. So that also is a limitation with this particular marker. So what is the next new future? So the current stage as what we are, and as the world is moving towards molecular diagnostics today. And that has already happened in the field of oncology. And we are already talking about various signature markers and a liquid biopsy using a peripheral blood sample. So when I talk about serum tumor markers, apart from our clinical immunoassays and biochemistry, we are moving from this to molecular genetics and cancer genetics using a peripheral blood sample. So the example for this and why it is happening is because NGS as a technology, which was always available only in research labs, today it is no longer a research lab technology. It is easily available in most of the reference labs today. And now that it is easily available, the sensitivity of these NGS has also improved significantly in the past decades. The challenge when I talk about using molecular markers in oncology is a very small amount of DNA, which is what is circulating in the peripheral blood, like a needle in a haystack, is what I want to pick up and I want to identify. So the current NGS technologies have improved so much that they have this capability of identifying very, very small and very, very low quantity of abnormal DNAs within a peripheral blood sample. And hence, that central dogma, which we all learn as DNA gives rise to messenger RNA gives rise to protein. So our whole focus right now was here, the final protein. We were targeting this. So now we are taking a deeper dive inside, and we are now looking at messenger RNA and DNA to give the same information. So some of the examples which you already know, classical example is chronic myeloid leukemia, BCR-ABL transcripts and real-time PCR for both diagnosis as well as follow-up of a CML patient is already existing today, right? So your RT-PCR transcripts which we identify on an RT-PCR in a peripheral blood for CML is nothing else but a molecular biomarker which is present in a peripheral blood. Same way we have HER2 for CA breast, EGFR, BRAF, ALK and ROS, which is already available, and we are using it for CA breast and lung cancer.
Dr. Rajesh, you have only 1 minute left.
Yes, last slide. So this is what is already existing. These markers, as we are identifying, we are having targeted therapy, and this has evolved to personalized medicines and theranostic, and this is going to evolve still further. What is still the future which beholds us? The still future which beholds us is this part, which again maybe in another 5 years down the line will happen. A general screening marker for cancer genomics for general population, having known this entire knowledge that we would have of this cancer genomics and genetics. So today we are talking about targeting hotspots, cancer hotspots alone. Now we are talking about screening the entire DNA of a person and predicting a possibility of a cancer in near future. So that would be the next step. And there the key role would be played by this analysis, which is methylated DNA markers, which is going to be the next future. So those were my thoughts on serum tumor markers. Thank you for a patient listening.
Thank you, Dr. Rajesh, for a nice presentation of tumor markers. Anyhow, future of cancer testing will be NGS. Let us have 1 or 2 questions from the floor. Any questions? If
Any
there are
questions?
no, yeah, no, I hope there are no questions, I think. Everything is clear, I think. Thank you. Thank you.
Thank you.
May I request Dr. Ramesh to please hand over the certificate to. May I request Anil to hand over the certificate to Dr. Ramesh Kumar? So I think we will have running tea coffee so that we won't waste more of time, because it's already late, running late, and the next speakers and chairpersons are eagerly waiting when my turn will come. So without further ado, may I request the next speaker, Dr. Dhanalakshmi, to please come on stage. And may I request Dr. Ujjwala Maheshwari to chair the session, please.
Welcome, Dr. Dhanalakshmi. Dr. Dhanalakshmi is Senior Consultant and Head Biochemistry, Apollo Hospital, Chennai. Our topic today is interferences in immunoassays. I'm happy to announce Dr. Dhanalakshmi is MD Biochemistry from Madras Medical College, Chennai, and she has 20 years of rich experience in heading biochemistry laboratory. She has publication in both national and international journal. Her area of interest is interpretation of serum proteins electrophoresis and immunotyping, validation, and test equipment. Over to you, ma'am.
Thank you, ma'am. Thank you for the introduction. Namaste all, and a very good evening. Thank you, Mindray India team, for having given me this opportunity to be here to share my thoughts on interferences in immunoassay. Not everything is perfect in life, and it is very much true in the medical diagnostic field, in clinical testing laboratories. And in spite of the efforts taken by the developers and the manufacturers of the reagents and the vigilance of the laboratory staff who perform these tests, there are not all tests on occasions subjected to many interferences that can lead to a false high report or a false low value. And immunoassays are no exceptions. Immunoassays, we all know, basically is done with the principle of antigen-antibody reactions, and they are the method of choice for all of us for estimating the hormones, tumor markers, therapeutic drugs, and microbial serology, etc., because most of it is available in the fully automated platforms, because of their very short turnaround times, relatively high specificity and sensitivity towards a panel of large heterogeneous molecules. But we have to keep in mind that these immunoassays are subjected to a lot of interferences which may not be recognized because they are very difficult to be recognized. Just to a quick recap on the types of immunoassays so that we'll be able to understand the mechanism of these interference acting on these immunoassays to produce false results. Immunoassays are broadly classified into competitive and non-competitive. Competitive are the limited reagent methods, and non-competitive are the reagent excess methods. Non-competitive, we also call it as a sandwich method or a immunometric method. In a competitive immunoassay, the capture antibody is anchored to the solid surface. This is the pointer. Okay. So as you can see, the capture antibodies are anchored to the solid surface. And then the green ones are the analyte of interest present in the patient sample. And then a limited quantity of the labeled analyte is also added along with the blood sample. So these both analytes compete for binding with the capture antibodies that are anchored to the solid surface. So after a washing step which removes the unbound fractions of these analytes, the signal that is present on the bound antibody is measured, and it is indirectly proportional to the concentration of the analyte present in the sample. And with the non-competitive amino assay, again there is a capture antibody coated on to the solid surface. The analyte of interest is added, and another antibody which is called as a signal antibody to the analyte of interest is added, such that this analyte forms a sandwich between the capture antibody and the signal antibody. Then again, after removing the unbound fractions by washing away the supernatant, the bound fraction, the signal is detected, and the amount of signal is here directly proportional to the concentration of the analyte. So these are the two basic principles of the immunoassays that we use. Now, there are many other classifications like homogeneous and heterogeneous. We call it as homogeneous when there is no washing step available. It is just based on a change in the reaction that happens when an immune complex is formed. For example, a nephelometric assay or an immunoturbidimetry assay which employs antigen-antibody reactions. A heterogeneous assay is where we employ a washing step to remove the unbound fractions and then measure the bound fractions. And again, based on the type of signals used, they can be classified as chemiluminescent assays, turbidimetric, nephelometric, and radioimmunoassays. Now, what are the interferences? Interferences may be defined as the effect of a substance present in the sample that alters the correct value of the result and might be the possible reasons for the false reports that we get in an immunoassay procedure. Now, what are the types of interferences? These interferences can be exogenous and endogenous. In the morning, during Dr. Aparna's presentation, there was a discussion on the lot of factors that were affecting the levels of prolactin. So those were the exogenous reasons or causes that could affect the levels of hormones. I am going to more focus on the endogenous interferences that are present in the sample that could lead to a false positive or a negative result. So these are again endogenous interferences are classified as analyte dependent and analyte independent ones. Under the analyte dependent ones, the first one we take is the cross-reacting substances. When these cross-reacting substances are present in the sample, then we will have interferences. How is it? The very good aspect of immunoassay is that the antibodies used in these assays are very specific to the analyte, but at the same time, when the structural differences are small between the molecules, then there is a substantial cross-reactivity. For example, the cross-reaction is substantial between the cortisol levels and prednisolone. When the patient is put on prednisolone and a request for cortisol assay comes, then the cortisol levels are overestimated. The prednisolone gives rise to a positive interference. That means it elevates the cortisol levels spuriously. Similarly, when we estimate the immunosuppressant drug cyclosporin A, most of its metabolites, again cross-react, have a very close structural resemblance with cyclosporin A that these immunoassays are unable to differentiate. The antibody is unable to differentiate, so that the exact drug is getting overestimated by the presence of their metabolites. Then this is also true for steroid hormones like testosterone and estradiol because of their structurally related compounds being present in the serum or blood sample. The second one is the heterophil antibodies. Now, what are these heterophil antibodies? Heterophil antibodies are naturally occurring polyspecific low affinity antibodies, which when faced with a particular antigen becomes a high affinity antibody. The heterophil antibody is almost around 25% of the elderly population is said to have an increased amount of this heterophil antibodies in their blood. And these antibodies, when faced with an animal antigen, becomes an anti-animal antibody, to be very specific, when we are exposed to mice, which is ubiquitous in environment, we develop the anti-mouse antibodies, and this is particularly a problem because most of the immunoassays use anti-murin antibodies in their assays. So how does the heterophil antibodies bring about interferences? They can either cause a false positive value or a false negative value by forming either a false sandwich and prevents the antigen of interest to form the sandwich, leading to false positive results, and again a false negative result when it individually attaches to both the capture antibody as well as the signal antibody, because of which a sandwich is not formed at all. So this is the basis by which a heterophil antibody can interfere in an immunoassay. The next one is the human anti-animal antibodies, which I already discussed. The very important one is the HAMA, that is human anti-mice antibody, because mice is ubiquitous in nature. Again, these, especially the immunometric assays, are more sensitive to the interferences because of these antibodies more than the competitive assays. Again, they would lead to a false sandwich formation in the immunometric assays, leading to false negative results or false positive results. Both way it is possible. It is not that these anti-animal antibodies will always produce false positive results. They can produce a false negative result for another immunoassay. So their mode of interference varies between assays. Now, high-dose hook effect. When an antigen of interest is present in large quantities, we observe this effect called high-dose hook effect, wherein the result is produced as extremely low value of the analyte. The analyte may be in lakhs or thousands or lakhs, but when we get the results from the assay, it would be in, it could be just normal or even below normal. This is because the excess antigen that is present in the sample, because the antibodies are available on both from the signal aspect as well as from the anchor aspect, it individually goes and binds to both of this so that a sandwich is prevented. So what we get is a very low value for the analyte, which is actually not true. And then comes the analyte independent endogenous interferences. We all are aware of the biotin interferences that can happen in the assays. And anti-ruthenium antibodies. These ruthenium is the signal which is used in the electrochemiluminescent assays. And when an anti-ruthenium antibody is present, it leads to false negative values. Then use of highly specific monoclonal antibodies. We were very happy that the immunoassays have become more specific because they have moved from using polyclonal antibodies to monoclonal antibodies. But here again, we have a limitation. Because of the use of this highly specific monoclonal antibodies, each of these antibody is targeted towards a particular epitope of the molecule of interest. Say, for example, let's take any hormone, a thyroid hormone or a prolactin or anything. So when this epitope is modified because of a benign mutation, which can have a normal functioning of the molecule, but it becomes different in its immunoreaction. So these antibodies are not able to identify the epitopes which is modified. As a result, it gives us a false negative results. Then the disease status per se can give us a lot of interferences. when the free fatty acid levels are high, as in diabetic ketoacidosis, it displaces the thyroxine from its binding proteins, as a result of which we have a spurious elevation of free T4 levels when we do the assay. Similarly, in cases of renal failures, the PTH metabolites accumulate and then cross-react with the PTH sandwich assays and gives us a spurious false reports of PTH levels. Then the pre-analytical factors. Again, we were discussing about this in the morning session. The notorious hormone is prolactin, which is subjected to so much of diurnal variations. It changes with stress, with inadequate sleep, or any slight deviation in the regular activity. It give rise to a lot of changes in the value of prolactin. Also, the prolactin peaks during the sleep, and it tends to come down when the person is in the waking up phase and remains elevated for almost up to 2 hours after waking up. So sometimes when the sample is taken early morning, as soon as the patient is up from the bed, we can have a small spurious elevation in the prolactin value. Or in a very stressful conditions, if these samples are given for prolactin levels, again there will be a marginal elevation which might cross the reference intervals for prolactin. And so just to summarize the properties of these interferences, they are unique to an individual. Interfering antibody concentration can change over time within the same individual. Low affinity polyspecific antibodies can be present in high concentrations, or high affinity antibodies can be present in low concentrations. And they can produce falsely high results or falsely low results. They can interfere with one or more manufacturers' immunoassay system, but not necessarily in all assays. They may interfere in a number of immunoassays for different analytes, and the inclusion of one or more interference blocking agents in the manufacturer's immunoassay reagents may be insufficient to overcome the interferences. Now, how are we going to get a clue to the possibility of an interference in our test reports? One is when the results are discordant. For example, let's take the thyroid hormone reports. When the FT3, FT4 reports do not have the relationship with that of the TSH, that is, they are usually, they have a reciprocal relationship. When FT3, FT4 is high, TSH is low, or if this is euthyroid, and that should also be euthyroid. But of course, there are few conditions where all the three are on the higher side, like thyroid resistance, or all the three are on the lower side because of a thyroid euthyroid 6 syndrome. So these are all exceptions. But whenever this relationship is disturbed, we always should think of a possibility of an interference. And some values, like for example, TSH again, they are fairly constant in an individual. So when you compare the previous results with the present result, and if there is a huge difference between the two, then definitely there is a possibility of an interference. Then with the clinical settings, if the lab is lucky to have an access to the clinical history of the patient, and if it is able to communicate with the clinicians and elicit the history from the patient, then it is easy for us to get a clue towards the possibility of an interference being present in the sample that could lead to a false report. So again, in the clinical history, we can elicit a history for recent immunization, transfusions, autoimmune diseases, any history of monoclonal therapies, or contact with pets. Now, why should we be bothered about these interferences? Because it's very difficult to detect as well as to investigate. So the main reason is because the misinterpretation of patients' results by the laboratory could lead to...
We are running out of time. Time is up. Time.
Okay. Wrong course of treatment being given by the physician. So how do we investigate these possible interferences? One is that you always suspect for the presence of interferences, and this can be made possible by creating awareness for the presence of interferences. The simplest way is to do a serial dilutions just to see whether the linearity or a parallelism is exhibited, and a repeat analysis on another instrument from a different manufacturer, because between the manufacturers, the antibodies used are different, and the target epitopes for these antibodies are all different. So it's always better to repeat the test using another manufacturer assay. Then use of a polyethylene glycol precipitation whenever an autoantibody is suspected. Autoantibody are nothing but the antibodies to body's own components. For example, the classic example is macroprolactin, wherein the prolactin complexes with its own IgG antibody, forming a large immune complex which remains in the body and gets detected in prolactin assays, leading to false high reports of prolactin, raising a suspicion for prolactinomas. And then you can do another simple thing is to check using another matrix. If you think there is a spurious elevation of beta HCG and there is no evidence for pregnancy, we can just take the urine sample and test it for HCG, which is going to tell us what the real status is. And then use of heterophil blocking tubes. This is again debatable because it's not always 100% used for completely blocking the heterophilic antibodies when present in the sample, because when we have a result, we can still say that we cannot rule out the presence of interference even after using a HBT tubes. Then the investigations which I enumerated earlier are all possible in a non-specialized laboratories because they are all simple steps. But in a specialized laboratory, we can have some special investigations which will definitely drive us towards the presence of a interferent, and those are extraction using organic solvents, gel filtration column chromatography, and measurement using LCMS mass spec. And how do you prevent such consequences? Informing the clinical and the laboratory staff of ever-present possibility of unexpected interferences, laboratories to put process in place to detect and test for these suspected interferences, and ensuring that a brief clinical detail is available for all the laboratorians. And above all, facilitating the excellent communication between laboratory and clinical staff is the key to minimizing the risk of these clinical mismanagement arising because of an unsuspected interferences. So I have just two case studies. A 2-year-old boy was brought by his mother complaining of poor growth and just 2 minutes, poor growth and poor weight gain. And the investigation showed us TSH to be very, very low with normal FT3 and FT4 levels. This this test was repeated once again after 3 months because it was not correlating clinically, and further investigations were all done to rule out a central hypothyroidism. And again, the sample came to the laboratory, and again we reported a value of TSH 0.01. Then the clinician spoke to me and said, this is not correlating clinically. So the first step was I repeated it in another platform. And this was the result. With the center, the value was 0.01. With the other two platforms, the TSH value was very much normal. So this very low value of the TSH in center raised a suspicion that this could be a method-specific interferent. That is, possibly the molecule is not getting detected by the immunoassay that we were using. So then I asked for a gene analysis of TSH gene, and it turned out to be a mutation in the TSH beta gene affecting the immunoreactivity, but functionally it was normal. So again, this is a perfect example of using a highly specific monoclonal antibody in the assays. Another case study was on a young female who came to us. She's with a history of hypothyroidism for a master health checkup, and HS troponin I was done as part of risk stratification for future cardiac events, and twice she had repeated, and it was found to be 461 once and 211 the second time. So here again there was a huge difference in the value, and but anyhow a thorough cardiac assessment was done. Because there was hypothyroidism, I had asked for anti-thyroid antibodies and then rheumatoid factor. There was a marginal elevation of anti-thyroid antibodies, but the rheumatoid factor was negative. Again here, the test result was discordant with the clinical picture, so it was pointing towards a method interference. So a dilution check was done. Again, there was not exact linearity that was exhibited, so I went ahead with testing it another platform, and I found this, that in 2 other platforms, the reports were very, very normal. So I suspected a macrotroponin I, and which proved to be the case after doing the PEG precipitation. A post-PEG precipitation, it was analyzed in the same Abbott Architect platform, and we got a value of 5. So the diagnosis was macrotroponin I interfering with the assay. So the take-home message is every immunoassay is prone to interferences. Divergence with the previous results or discrepancy with any other biochemical parameter or clinical setting is paramount to suspecting an assay interference. The correct reporting of interference is the responsibility of the clinical laboratory, and no single test is sufficient to identify an interferent. Thank you.
Thank you so much, Dr. Dhanalakshmi. It was an excellent talk. The case studies were excellent. Something unusual. Any questions for Madam?
I don't think so. So may I request Dr.
Sir. Hi, thank you ma'am for a very good lecture. I'd like to ask, have you seen hook effect in your clinical practice? Because it's kept on mentioning on all the kit inserts, but have you seen it, and have you like how to suspect it? Yeah.
We are more, whenever we think of this hook effect, we think only in terms of prolactin, because that is where the clinicians most of the time interact with us, saying that can there be a hook effect that you are not able to detect a high value of prolactin. But I have not seen any hook value for prolactin, because our assay, the hook value cutoff is almost 60,000. Because each kit defines the cutoff limit for the hook effect to occur. So our kit has a cutoff limit of 60,000, but I have encountered this effect in the free light chain testing in turbidometry. When we use these turbidometric assays in our regular chemistry analyzers, it fails to detect the high concentration of the samples, nor we are able to appreciate this hook effect, because we are unable to see the reaction curve. So that's where I have encountered one hook effect.
May I request Dr. Ujjwala to hand over the certificate to Dr. Dhanalakshmi? Now, may I request Dr. Riju Matthew to come on stage to deliver his talk?
Welcome, Dr. Riju. Dr. Riju is a professor at Church Medical College, Tiruvilla, and Vice Dean in BAAHS. Dr. Riju is Westgard Certified Quality Manager and Six Sigma POI of NABH Full Accreditation, certificate holder in law in hospital administration by CMAI with distinction, diploma holder in end user computing, first CPQIL in Kerala by CAHO, ranked in top 20 professionals for laboratory excellence program by Stein Westgard. Assessor as per ISO 15189-2012 by ACCLMP. Auditor, trainer for ISO 15189-2022, NABL 112, NABH assessor as per essential standards for clinical laboratories and having 25+ years of laboratory experience and extensive teaching experience, approved KUHS research guide. Welcome, sir. We are here to listen to you.
Thank you, ma'am. I'll start with a question. I especially thank the organizers and the team Mindray for arranging such a beautiful, wonderful conference. I thank for this opportunity to present this small, little, potentially threatening topic in front of you. I also ask your pardon if I make a mistake because I'm a small child in front of the knowledge of artificial intelligence. I can give you a wrong information because the information might have already changed from the time I have updated it, where my updation ended few hours back. Okay. Artificial intelligence has a role everywhere. Now you are sure about it. How many of you are using ChatGPT? If you could raise your hand, it will be good. And may I know, are you using for diagnosis as well? Did you ever try that with your laboratory values? Okay. And we need to thank the person, Mr. Altman, who is now not there in OpenAI. He moved from there to Microsoft, who actually made a user interface, UI, for the artificial intelligence which I was hearing from my early childhood and 10th years. The concept of artificial intelligence came from the fact that we learned everything from nature. So one of the most complex things that we couldn't understand, or we are trying to understand until now, is the neuron function itself. So people tried how this can be replicated, and here you have the answer. You are lucky, or our generation is lucky to see the outcome. Probably before we die, we'll see many, many, many wonderful things. This is the concept that is created, or the model that is created, which is the background of what we see in ChatGPT, where the knowledge is artificially created from the information that is available, especially in the form of internet now. It is not only from internet, it is also from digitalized content. When I was in the 11th standard, or about 40 years back, this was an article that came in 1983 on evaluating AI system for medical diagnosis. Was maybe a fun at that time because people doesn't know what is artificial intelligence at that time. Only the techies. If you see the journal, it is an AI magazine. So only the techies know what it is. They could only explain. But I'm sure that the AI concept at that time was not evolutionized as what we know now. In 1987, expert system for clinical diagnosis are worth the effort, the question raised again in the same journal. Why? Because there was a concern whether this will be able to give the outcome as expected. By 1990, application of artificial intelligence were used for identifying small clinical problems like diagnosis of jaundice. Later, by 2006, computer-aided diagnosis based on medical image processing was used. Probably this could have been the beginning of the probabilities or the possibilities the AI can have has raised. Now it's very well known that the AI is capable of better reading the X-rays or any other images, including the pathology slides. Why? Because the vision of the human eye has limitation, while the transformation of image into different type of computer or you say computational method is possible, including Fourier analysis. High performance medicine, the convergence of human and artificial intelligence was came as a review article in 2019. I'm sure by 2019, the biggest respected journal in the industry, Nature Medicine, could identify this and place it, which was the beginning of artificial intelligence being widely used and probably certified by FDA like approving agency. I remember that by 2019, there were few algorithms or protocols were certified by FDA. Where all AI can be used? Everywhere. It used some technical terms like causal modeling, strategic reasoning, explanation of things, knowledge acquisition and verification, validation and evaluation of AIM systems, anatomic reasoning, temporal reasoning. It goes on like this. Later, by the 2019, the American Association of Clinical Chemistry also started publishing related AI-related matter, where people were confused with the terminologies. What is AI? What is machine learning? What are models? What is learning? What is supervised? And what is semi-supervised and computer vision like? Computer vision is the vision or the vision capability by computer or any camera as you see anywhere else. Definitely will change or might change already the capability that we have as a laboratory personnel. There are models like causal models which are mathematical models, where we were told in the beginning that if you don't study the maths, somehow we took the biology by chance, but we didn't want to study the maths. But if we would have studied both maths and biology, this would have been easy for us. The mathematical model of biological things or other things that made the causal models possible. Those who doesn't like the statistics, regression models and R values will not be able to understand this concept also, where relationship between the components you use in a artificial intelligence are regression based, so that the relations can be easily identified. Then the question raises that why you want AI? The why has many explanation. I think I have updated the PPT which is not here. I'll explain why. Why has many answers now? Why? Because now in laboratory, from the beginning, that means from the pre-analytical area to the post-analytical area, the computer vision or the artificial intelligence will play a big role, probably that we will see by our eyes within few years. I have been lucky enough to try and do on FDA certification for one of the equipment by Sigtipple, which is a Bangalore-based company, who was doing slide-based PBF certification, that means peripheral blood smear reporting, which they could market it very soon in the market, and this equipment is currently available in the market. They were also trying to see whether they are able to do urine slide evaluation as well as the bone marrow slide evaluation also on the same analyzer. They were successful in keeping urine testing in the same analyzer, but I am not sure about the bone marrow because later we couldn't have a tie-up. So that equipment is currently FDA certified and is available in the market. When you stain a slide and give it to the equipment, it will scan the complete slide and classify all the cells according to the differential morphology and count it and give you a PBF report with the diagnosis. But it doesn't give a diagnosis as such, will allow the pathologist to take a better decision. So it's in the pre-analytical. I was wondering and imagining about a pre-analytical system where you need not keep a barcode. Instead, you just keep a camera there. It will be able to read the sample and identify the sample. There are systems like that that is currently working in US, where biggest supply chain like Amazon is not using barcode-based identification tool. They are using AI-based identification tool with 100% accuracy. So that is going to be something very big, that the pre-analytical thing will be completely controlled by the artificial intelligence, where all the sample transport, storage, retrieval, everything will be easily done by AI in future. And that is, we are going to see this. In post-analytical, whether the results are appropriate. When we give interpretation to a particular test, it is a fixed interpretation now being given, because your footer is the same. But in future, what is going to happen is AI will be able to give an interpretation that is according to that particular person based on the other tests being done also. So the interpretation and the post-analytical evaluation of the result is going to be totally changing, and that also has been applied in many places. The question is, will it replace the expert like you and me? Don't get afraid. If you get afraid and if you are apart from that, you will not be going forward, because you have to learn this and be with this, so that this is the era that is people were afraid of, or people were not expecting. You have to be with this particular thing, that you need to learn what is the principle, where all you can apply this and be with this system, so that your learning process as well as your work will be simplified. Best example is my work has been reduced to 1/10. I have started taking more challenging work, because I could be supported by the artificial intelligence, including for programming. It was initially started used for the radiology and pathology. Now you can see it is being used everywhere. Pathology, I'm not going into the details. In hematology, I have an example I already told you for calorimetry to digital imaging and AI. I am sure that there are organizations or the vendors who are already applying this. Instead of calorimetric assay, they started using image analysis. With the pixel concentration, they are able to give a better result than the calorimetric result. So this is going to replace probably our wet chemistry into dry chemistry or spot-based chemistry. And this image analysis is not the advantage of such image analysis, that you need very little sample, as well as the complexity of the equipment can be reduced much, so that so that the point of care equipments can be available. I have done a trial for one of the AI machine that was capable of doing chemistry as well as the hematology in a single machine. That was the shape of a CPU. So this is already there in the market. Okay, AI can be helpful in reporting as well as releasing the results also, because releasing, as you know, need many information that when we try and search may not be available in the computer, or the AI will be able to get this information, because it can handle a large number of data. As I told already, FD has approved many such equipments as well as protocols, and now you have a very huge list of AI-supported equipments, including for diagnosis as well as for laboratory. Now AI-based chatbots are available. One example is the ChatGPT, but ChatGPT is not specific for medical. There are medical-based AI algorithm available that is readily available to Google, and there are many other companies are also coming up with such algorithms. Remember that when digital pathology was initially introduced, it was a 1 crore or 2 crore project. Now it is going to be very cheap, so that you can use digital pathology and make your work more easy. That came in 2019. I just told I have updated the slides. It was not there. This I have seen from one of the biggest threat that we have. If you are not a person who is knowing in detail and understanding the concepts very clearly. This will be the outcome. I just want to warn. This is the first ever suicide of a robot happened in the world. You can go back and read this. First ever suicide of a robot happened. This was a cleaning robot in US. This came in newspaper in Mirror in 13th November 2013. And this is the second one reported. It was a cleaning robot in the open air. Didn't want to do its work. Just went. Thank you.
Thank you, Doctor, for taking us from chemistry to the world of AI. I don't know what we are going to see next.
Yes.
Thank you, sir.
Any questions for Dr. Riju? So when it's AI, I would like to make everyone aware that Mindray is already into hematology AI, and we have we have invented our very own digital morphology system, and it's already working at 2 places in India. So AI and hematology, we are proud of it.
When I met the corporate head office at China about 6 years back, I asked them, do you have a plan? I heard that they already came up with a plan. That's very good. Happy to hear that.
Thank you, sir. And we have a TLA system, Total Lab Automation System, which also is based on AI. So
Dr.
we are
Vinita was
already
with
on the
me.
path.
Remember, ma'am? Okay.
So we are already on the path, and coming years we'll see more. So thank you, Dr. Riju. And may I request Dr. Ujjwala Maheshwari to please present the certificate to Dr. Riju Matthews.
Histopathology is there. Yeah, okay.
May I request Mr. Dharampal Sharma?
Now they are trying to find out all the cancers.
May I request Mr. Dharampal Sharma to please hand over the certificate to Dr. Ujjwala Maheshwari, please? Thank you so much, Dr. Maheshwari. Thank you, Mr. Dharampal Sharma. So, may I invite Dr. Afshan Rafi, and may I invite Dr. Vinita Kothari to chair her session, please? May I request the next 3 speakers to please abide by the time? Thank you for understanding.
It's my pleasure to introduce Dr. Afshan. She is an NABL assessor for clinical biochemistry, and she has worked as assistant professor in CHEMS. She is also a consultant biochemist in Fernandez Hospital. And presently, she is working as consultant biochemist and HOD at Global Hospital in Hyderabad. Dr. Afshan.
Thank you, ma'am. Good evening, all. I think it is almost the last sessions going on and all are feeling drowsy. Hope my slides won't make you sleep more. So first of all, I thank Mindray CAHO for giving me this platform. And also a special thanks to Shama and Mr. Fareed, who have been constantly in touch with me and coordinating about this. So thank you.
serum ferritin. I'm going to talk about serum ferritin, which is more of inflammatory marker or anemia marker. So since I think I can say before 2020, we have seen that ferritin, as per my experience, I have seen ferritin only as an anemia marker. Like in my hospital, all the clinicians used to write serum ferritin mostly only for anemia. But since 2020, the picture of ferritin has completely changed. So it has become one of the inflammatory markers, and COVID-19 has taught us that ferritin has become a hero in CLIA technologies, like apart from vitamin D, vitamin B12, and ferritin. So since I was told to talk about something related to CLIA, so I picked up serum ferritin. So I'll be talking a brief introduction about ferritin. As we all know about it, still a brief introduction. And the functions related mainly to anemia and inflammation, diagnostic uses, deficiency, and excess. So ferritin, as we all know, is a globular protein complex consisting of 24 protein subunits, forming a halo nanocage with multiple. So as this picture of my first slide shows, this is the ferritin. Ferritin is a universal intracellular protein that stores the iron. So as we all know that ferritin is used mainly a anemia marker for iron deficiency. And it also releases it in a controlled fashion in both prokaryotes and eukaryotes, thus keeping iron in a soluble and non-toxic form. That is, it is converting the ferrous to the ferric form. In human, it acts as a buffer against iron deficiency and iron overload. So one of the properties is that it maintains the iron hemostasis. Serum ferritin is also an indirect marker of total amount of the iron stored, and hence it is a diagnostic test for iron deficiency anemia. Aggregated ferritin transforms into toxic forms of iron called the hemosiderin. So there are many functions of ferritin. So I'll be talking mainly about the iron storage, the peroxidase activity, the immune response, and the stress response. Apart from that, ferritin also maintains the hemostasis, iron hemostasis. Also, it it controls the hyperferritinemic syndrome, and it is helpful in inflammatory diseases. It gives a protection against the free radical by converting the toxic iron into a soluble iron. Obesity also it controls. It also controls the infectious diseases, autoimmune diseases, and mainly it has become one of the COVID markers. After this COVID-19 has come up, it has become a marker for inflammation. And also it was also an acute phase reactant. It also protects us from malignancy. Coming to the iron storage, the ferritin is present in every cell type, and it serves to store the iron in non-toxic form, to deposit it in a safe form, and to transport it to the areas where it is required. And the free iron is toxic to the cell, so that's why it converts it into a soluble form. And also if there is a higher amount of the iron is there, the ferritin is related to the hemosiderin. Next comes the peroxidase activity, where the ferritin controls the penton reaction. That is the conversion of the ferrous into the ferric form. And thereby it prevents the release of the hydroxyl radicals. Next comes the immune response, where the ferritin concentration increases drastically in the presence of an infection or cancer. So the same thing I'll be dealing about few case studies where the ferritin levels were increased in the inflammation. And this is mainly because of the endotoxins which are present, which regulate this concentration of the ferritin in the case of the infection and cancer. This was the scenario where in COVID-19 I had taken from one journal, and this is where the SARS-CoV-2, when it attacks the body, it causes the cytokine storm, and this cytokine storm causes the release of the ferritin, causing hyperferritinemia. This hyperferritinemia in turn causes this excess ferritin to bind to this
nuclear receptor co-activator 4, and this causes the autophagosomes to release the iron from the ferritin. So these autophagosomes, they release the iron by the degradation of the ferritin, and thereby the intracellular iron increases. This in turn causes the increase in the reactive oxygen species, nitrogen and sulfur species, and ferroptosis, in turn causing the tissue damage. So this was the scenario which was we have seen in the case of the COVID-19, and how this hyperferritinemia was seen in individuals who were suffering from COVID infection. And in the case of the stress also, the ferritin levels have been seen to increase, such as anoxia, which implies it to be an acute phase protein. So now coming to the diagnostic uses, as we all know, it is a immunoassay parameter which can be used by ELISA or CLIA or CLIA methods. It can be tested, and we use the nanograms per nanograms per milliliter and micrograms per liter, where the conversion unit is only 1. And the serum ferritin levels, we have seen that they have mainly they used to do for the iron deficiency anemia. So since the ferritin levels measured usually have a direct correlation with the total amount of iron stored in the body, so therefore whenever there is iron deficiency anemia, there was a decrease in the ferritin levels also. But sometimes the ferritin levels may be artificially high, mainly because of the anemia due to the chronic disease. So in this condition, we have to rule out the inflammation, and then only we can judge that it is the ferritin is high because of the inflammation and not because of the anemia. So coming to the studies which have been done for as an showing the iron ferritin as an inflammatory marker. So there was a study done in 2021, and this was after the COVID. They have shown that the cytokine storm syndrome in patients with COVID-19 is mediated by pro-inflammatory cytokines, resulting in acute lung injury and multi-organ failure. Elevation in serum ferritin and D-dimer is observed in COVID-19 patients. This is another study which was done in 2020, and this shows that an inflammatory player keeping iron at the core of pathogen host interactions. And they have shown that during the infection, both host and the microbe need the access to the iron and avoided toxicity. So alteration in serum and cellular iron have been reported as important markers of pathology. In this regard, ferritin, first discovered as an iron storage protein, has emerged as a biomarker, not only in iron-related disorders, but also in inflammatory diseases, or diseases in which inflammation has a central role, such as cancer, neurodegeneration, or infection. And even there was one study done in 2021, and they have proved that ferritin measured at admission may serve as an independent factor for predicting in hospital mortality in patients with COVID-19 ICU. So it was even shown that it is not only a diagnostic but also a prognostic marker also. Coming to the deficiency, these were the studies which were done in, one was done in 2015, which showed that ferritin level below 30 nanograms per ml indicates iron deficiency, while a level below 10 nanograms indicates the iron deficiency anemia. And WHO has stated that iron deficiency in children below 5 years is below 12 nanograms, whereas it is below 15 nanograms per ml in children above 5 and over. And also this low serum iron is more specific in lab finding for iron deficiency anemia. And however, it is less sensitive if it is increased in the blood by infection or any other type of chronic inflammation. So this is one study which we have seen in the second wave of COVID in my hospital, where one case had come, a 66-year-old male came to hospital with history of fever, SOB since 1 day, and dry cough with history of travel. And the vitals were normal on examination, and SpO2 was 94% at room temperature, and HRCT showed cohorts 5 with 80% lung involvement. So in this patient, we noted that it was COVID was tested and it was positive. D-dimer was like it was low, and when the patient was admitted, it was low. And by the time of around 5 days, it became high. NT-proBNP was normal. LDH also was low at the time of admission, and later on it increased. But the ferritin was 111, and later on it reduced to 53. In this patient, actually iron study was not done. It was a comorbid case. It was brought in ICU. So that's why I could collect the data from the CBP, and I noted that the hemoglobin was 10.9 at the time of admission, and later on it decreased to 9.5. And peripheral spheres showed hypochromic microcytes. So the reason for ferritin to low in this case could be the iron deficiency anemia, which was it was not much looked upon it, since the patient was COVID-19, and so on that based, the treatment was given for infection only. Now, excess of the iron. If the ferritin is high, there is iron in excess, and or else there is acute inflammatory reaction in which ferritin is mobilized without iron excess. For example, ferritin may be high in infection without signaling body iron overload. So ferritin is also used as a marker for overload disorders like hemochromatosis and hemocelluloses, apart from the inflammation.
Dr. Afsan, please finish it in 2 minutes.
Ferritin is also an acute phase protein, and it's often elevated in the course of disease. And in the case of to rule out anemia and infection, we can also do the CRP levels to check whether the ferritin high is because of inflammation or because of anemia. So these are the 4 case studies which I have seen. One is a 65-year-old female. She came with a history of fever, cough since 6 days, and SOB of grade 2. Vitals were normal. SpO2 was 94. And CORAD 5 was there in HRCT and more than 30% lung involvement. So in this patient, D-dimer was done, NT-proBNP and LDH, they were all normal, and the ferritin was only 682, and the patient was COVID positive. So in this patient, I could say that ferritin was mainly the inflammatory marker with which we can rule out there is a viral infection, but all the other parameters were normal. Another case study which was in a 71-year-old male. In this patient also, there was CORAD 5 with more than 40% lung injury, and D-dimer, NT-proBNP, LDH were all normal. Only the ferritin was more than 1200 nanograms per ml, and patient was COVID positive. In this scenario also, it was the same. On the day of admission, ferritin was high, and then by the 5th day, it reduced to 572. And we did IL-6 also, it was normal on the day of admission. D-dimer, NT-proBNP, LDH were all normal. So we can say that the ferritin is more of the inflammatory marker now presently, because people have become more conscious with their health, and iron deficiency has become less now. So now whatever cases are coming to our hospital, mostly with the viral pneumonia, we are using mostly ferritin as a inflammatory marker. This is another scenario where again LDH was little high, but ferritin was 1121.9 nanograms per ml, with more than 30% of the lung involvement. So with this, I can conclude that the low levels of ferritin can be used as an anemia marker, and high levels can be used as an inflammatory marker. Thank you. These are my references.
Very nice presentation, Dr. Afsan. Any questions?
No questions. May I request Dr. Vinita to hand over the certificate to Dr. Afsan? Thank you, Dr. Afshan. Thank you, Dr. Vinita Kothari. So may I call upon stage the next speaker, Dr. Kalpesh Gandhi, please.
Our next speaker is Dr. Kalpesh Gandhi. He is an associated fellow in industrial health, and he has the experience of running standalone lab, hospital lab, small chain of lab, and research-based lab, practically all types of lab. He, since 2009, he established a central clinical path lab at Zydus Research Center that caters to basic research, preclinical studies, and clinical trials also. His lab is NABL accredited since 2009 and CAP accredited since 2011. Dr. Kalpesh Gandhi, please.
Or what we can do is we can flip the dated. Uh, uh, so my topic is HSCRP and homocysteine. We are dealing with this since probably 1990 or more that, because the first standard was developed for, the first certified reference material where the CRP assigned is probably in 1993, if I am not mistaking the year. It is invented in both probably invented in third or fourth decade of this 20th century. So what I will do is I will start with the take home message and then go with this, because it is only a theory. Because that's okay. फ्लिप कर दो, कल रख लो अदरवाइज चलेगा। हाँ ठीक है। So I am coming down from the stage and the next speaker will come. Working. Okay.
May I request, there's a technical issue, so
That's okay.
may I request Dr. Deepti Eka to come over stage, please? Extremely sorry for this technical glitch.
Dr. Dipti Eka, she is a lab director and co-founder in Rapidex Lab Delhi. She recently completed a certificate course on autoimmune and allergy from Harvard Medical School. She has a total of 15 years of experience. She has also worked in Leprosy Mission Trust, Yashoda Hospital, Hyderabad, and many other. Her area of expertise is clinical biochemistry, immunoassay, PCR, allergy, inflammation, autoimmunity, and so many. She has multiple papers published, and she was felicitated by state government of Haryana for exemplary service during COVID-19 pandemic. Dr. Dipti, please.
So thank you, Chairperson, and thank you, Mindray and CAHO, for giving this opportunity. It is a pleasure to be here. So the topic on which that I'll be speaking on is the different methodologies and analytical interferences for HbA1c. Now, diabetes is a major non-communicable disease in India, and it contributes enormously to the mortality, morbidity, as well as the health cost. And seeing the diabetes burden on rise since 1919, it has been leaping at a very fast rate. There has been many undiagnosed diabetes cases, which is estimated to be approximately to 57 million, which makes HbA1c as the most prescribed test to diagnose and monitor treatment for diabetic individuals. Now, seeing the time constraint, I'll skip this slide and I'll move to directly to HbA1c. Now, what is HbA1c? HbA1c is also called the glycated hemoglobin, where the glucose, it binds irreversibly and non-enzymatically to the N-terminal valine residue of the beta chain of hemoglobin A. Now, this reflects the glucose concentration during the previous 8 to 12 weeks. Now, this HbA1c concentration, it is directly proportional to the lifespan of the RBCs and the glucose concentration, which makes HbA1c directly related to the risk for diabetic complications, making it an essential component to diagnose and manage the diabetes mellitus. Now, the HbA1c detection methods are based upon charge differences and the structural differences. Now, ion exchange, high performance liquid chromatography, and capillary electrophoresis are based upon the charge differences, and the enzymatic methods, immunoassays, and boronate affinity chromatography are based upon these structural differences. Now, we'll have a look at HPLC first. Now, the ion exchange HPLC method, it uses buffers of increasing ionic strength to separate the hemoglobin HbA1c from the other species, Hb species, by differences in charge. So the concentration of hemoglobin in each fraction is quantified by calculating the area under the peak. Now, in this case, all the chromatographs have to be reviewed to identify the physiological forms of hemoglobin. The main advantage of this method is that it allows a presumptive identification of Hb variants. However, changes or mutations in the hemoglobin molecule affecting the charge will alter the HbA1c results if these variants co-elude with HbA or HbA1c. Now, borosilicate affinity chromatography is also similar. The difference is only that the column is filled with boron, and this separates only the HbA1c and not the other forms of other fractions of hemoglobin. Now, the next method is immunoassay. In this, the interaction is between the HbA1c, which is the antigen, and the HbA1c specific antibodies, which bind to this antigen by recognizing the N-terminal glycosylated amino acids. So this interaction, it leads to the formation of immunocomplexes, which lead to turbidity, which is then measured photometrically using a turbidometer or nephrometer. Now, in parallel to this, the total hemoglobin concentration is measured biochromatically in the pre-incubation phase. Now, the enzymatic method. In this, the percentage of HbA1c in the sample is directly measured, and it does not require any additional measurement of total hemoglobin. The samples are lysed and react with agents to eliminate signal interfering substances. The lysed whole blood then undergoes protease digestion, which releases amino acids. So these glycated valine that are released in this process, they act as substrate for this specific recombinant fructosyl valine oxidase enzyme. This enzyme then specifically cleaves the N-terminal valine, producing hydrogen peroxide. The final reaction is then catalyzed by the horseradish peroxidase, and it involves the oxidation of a chromogen, that is the leukodihydrogen peroxidase. And the resulted colorimetric signal is then read as 670 nanometers. The last method that we are going to talk about is the capillary electrophoresis. In this, the hemoglobin fractions are separated in silica capillaries by the electrophoretic mobility and electroosmotic flow at a high voltage in an alkaline buffer. Now, the separation of the hemoglobin species occurs in decreasing order of their charge to mass ratio, with the positively charged hemoglobins eluting first. Now, if there are two or more molecules that have the same charge, then the system will further resolve these by size, and the larger molecules will elute first. So overall, the positively charged molecules are eluted first, followed by the neutral species, and then finally by the negatively charged hemoglobin species. Now, this is a comparison of the advantages and the disadvantages of all these methods. So if you see enzymatic, immunoassay, and the boronate affinity, these have no interference with the variance, and the throughput is also good. But this serves also like a disadvantage for these methods, in which we do not get any indication of the variance, which serves as an advantage for the ion exchange, HPLC, and capillary electrophoresis, in which it detects any undiagnosed hemoglobinopathy if it is present in the patient. Now let's move on to the factors that interfere with the HbA1c results. Now the most common is the Hb variance. Now apart from this, there are other factors like Hb derivatives, chronic renal failure, iron deficiency, anemia, B12 deficiency, or splenectomy that will lead to falsely increased HbA1c. HbF, vitamin C and E deficiency, hemolytic anemia, chronic liver disease, splenomegaly will lead to falsely decreased HbA1c. We will further on see briefly about these interfering factors separately. Now first, I will be talking about the Hb variants. Now, the most common Hb variants that are present worldwide is the HbS, E, C, and D. Now, presence of any of these 4 variants, it can lead to change in the charge and the mass of the hemoglobin molecule. Therefore, the detection methods, which are based upon the physical properties like the ion exchange chromatography or capillary electrophoresis, these are vulnerable to interference. Now, boronate affinity, it does not get affected because as it separates only the glycated portion and not the other fractions. So this is not affected by the variance. Whereas if we talk about immunoassay, there is no interference by the HbE or the HbD variant. But because the HbS and the HbC variants are very close to the N-terminus of the beta chain, there might be chances that some of the immunoassays are affected by presence of these variants. Now next we'll talk about the elevated fetal hemoglobin. Now elevated fetal hemoglobin mostly affects the HPLC method, immunoassay, and the boroaffinity chromatography. Now in HPLC, if you see that the HPF and the A1 peaks are eluted adjacently, so very high level of HPF may overlap and it might distort the shape of A1C leading to falsely high values. Now, if we talk about boronate affinity assay, if we see the HPF, it does not have the beta chain, it has the gamma chain. So, in this case, the glycation, it occurs on the lysine residue of the N-terminus of the alpha chain, which makes the glycation slower, affecting this BAC assay. Now, immunoassay is based upon the interaction between the antigen and the antibodies. So these HbA1c antibodies are not able to bind with the glycated portion of the fetal hemoglobin. Therefore, when the total hemoglobin is measured in this, it will also include the HbF, which will eventually lead to reduction in the measured HbA1c. Now, next is the very high levels of triglycerides, hypertriglyceridemia, or severe hyperbilirubinemia. It might affect the HbA1c measurement, specifically with the two assays, that is the immunoassay and the enzymatic assay, leading to falsely low HbA1c concentration. Now, next we move to the clinical factors that affect the interpretation of HbA1c results. Now, first is the shortened erythrocyte survival. So, any clinical condition that increases the erythrocyte turnover, like for example, hemolytic anemia or a recovery from acute blood loss or the recent transfusion. Now, in this, there is the erythrocyte pool is enriched with younger cells. This will lead to falsely lowering of the HbA1c, and this is regardless of the assay method that is used. Now, next is the increased erythrocyte survival. Now, in conditions where there is splenectomy or hypersplenism, now this will increase. So, or any other clinical condition that increases the mean erythrocyte age. In this case, the hemoglobin molecules, they get exposed to blood glucose for a very longer time, leading to falsely elevation of the HbA1c test. Now, next is the iron deficiency anemia. So similarly in this, when the red cell production is decreased, it will increase the age of circulating RBCs and will falsely elevate the HbA1c levels.
Dr. Ikka, 2 minutes, please wind up.
Next, we move to the chronic renal failure. Now, in this chronic renal failure, the patient exhibit, in this there is both overestimation as well as the underestimation of HbA1c. For like in metabolic, when we see in CKD patient, there is metabolic acidosis or carboxylate hemoglobin. This will lead to increase in the HbA1c level, falsely elevation. Whereas when there is these patients go on dialysis, it will shorten the RBC lifespan and decrease the A1C levels. Now, similarly with the chronic liver disease, it may lead to falsely low level of HbA1c. Then there is another interference by the drugs, that is the long-term aspirin usage. Now, long-term aspirin usage, it induces acetylation of hemoglobin. Now, it changes its surface charge. Then the acetylated product that is formed, it co-migrates with the A1C, thus leading to falsely elevation of the A1C value. Other than this, long-term or very high dosage of ingestion of vitamin C and E, it will block the glycation, leading to lower HbA1c levels. Now, just to wind up, now there are various methods and there are various interferences. Now, laboratories, we need to be aware of all these interferences as well as the methods available and ensure that our HbA1c testing is matching the clinical perception. Now, even for cases that have possible interference, particularly the method or in which the RBC lifespan or the RBCs are being affected. Now in this, the HbA1c level, they need to be determined using different instruments with different analytical principle. And whenever the interpretation of HbA1c is negatively affected by erythrocyte lifespan or the glycation rate, non-HB test like glycated albumin and serum fructosamine should be used to assess the long-term blood glucose levels. These are the references, and thank you for your patient listening.
Thank you for such an excellent presentation. Any questions?
So may I request Dr. Vinita Kothari to please hand over the certificate to Dr. Dipti Ekka. May I please request the last speaker for the day, Dr. Kalpesh Gandhi, and sorry for that technical glitch, Dr. Kalpesh. May I request Dr. Dilip Ghosh Dastidar to chair the session for Dr. Kalpesh Gandhi.
Let me go with the. You will use this, sir? No, this is.
Good evening, ladies and gentlemen. It's my great pleasure to introduce Dr. Kalpesh Gandhi for the topic significance of prognostic cardiac markers, HSCRP and homocysteine. He is Associate Fellow of Industrial Health in 2010, experience in running standalone laboratory. And he is also an annual assessor since 2009 and CAP accredited since 2011.
Thank you, Dr. Ghosh. So this is the take-home message. First, I flipped these slides. So CRP is essentially the same as what we are analyzing, but with the high sensitive, which increases the sensitivity of that particular analysis. But there is a lot of buzz is around since 2000, I suppose 17, 18, CRP monomer will go to that. It is not the same as the HSCRP or CRP. The analytical method which we use for CRP must have the traceability of CRM 470, which was first assigned way back in 1990s, probably 93, if I am not mistaking the year. And then again reassigned by NIST. First assignment was done in collaboration with IFCC, European Commission and US FDA along with the CAP. So we are using a lot of terminology since morning, that is prognostic marker, monitoring marker, predictive marker, risk marker. Right now CRP has established as prognostic marker, susceptibility risk marker and predictive marker in association with the other biomarkers for cardiovascular disease. We will go to the categorization of the biomarker in subsequent slide, where FDA has clear-cut given the subcategories or categories of the biomarkers and how you should, to harmonize the usage of this biomarker, how you should use the terminology of that biomarker. And the last thing is that there are ample of possibilities that the CRP or MCRP specifically can be used as response biomarker, which is pharmacodynamic marker. So there are a lot of research are going on drug development in terms of drug development. Let's go with the first slide. This is actually the last slide of CRP. So let's go with the definition of biomarker. So biomarker is this definition is essentially given from for US FDA whenever somebody has applied a particular parameter to has the biomarker capability. It should have a defined characteristic that is objectively measured as an indicator of normal biological process, pathologic process, response to exposure or intervention that includes the environment as well as the therapeutic intervention also. So essentially the biomarker is to aid the understanding of one or more the prediction of disease, its causes, diagnosis and response to the intervention. So these are the 7 categories which was given by the US FDA. So all the essentially biomarker could be anything, not only the serum or blood biomarker. Radiology, radio imaging is also a biomarker. Blood pressure is again a biomarker. So it could be a histopathological, radiological, molecular, all. The response biomarker is again divided into 2 categories. That is the one is the pharmacodynamic biomarker and the surrogate endpoint biomarker. So what is the diagnostic biomarker? That used to detect to confirm presence of a disease or a condition of interest or to identify individual with a subtype of the disease. The classical example of this diagnostic biomarker is glucose, which ADA has given the cutoff value, HbA1c again this. So that is the diagnostic biomarker. Monitoring biomarker is a biomarker which measures repeatedly for assessing the status of the disease or medical condition for evidence of exposure or a medical product or a environment agent. So PT-INR used when patient has having this warfarin effect. That is classical example, age-old example of the monitoring biomarker. The response biomarker is a biomarker used to show the biological response potentially beneficial or harmful has occurred in an individual who has been exposed to medical product or an environment agent. That is again, as I told you, divided by into 2, PD and the surrogate endpoint biomarker. When INR used as a pharmacodynamic biomarker, when it is used for the prevention of thrombosis event in a patient with the warfarin treatment, that is the PD marker. It's a definition, I'll skip it. And surrogate endpoint is the endpoint biomarker which used as a substitute for the direct measure or direct evidence of how patient feels, functions and survives. Classical example is HbA1c reduction in case of the complications associated with the diabetes mellitus, used as the surrogate endpoint biomarker. The predictive biomarker is used to identify individuals who are more likely than similar individuals with the biomarker to experience a favorable or unfavorable effect from exposure to a medical product or environment agent. So CTFR mutations, that is the example of a predictive biomarker in evaluation of the treatment of cystic fibrosis, where some of the treatment which has been given to a selective patient. Many companies are working with the companion diagnostic as they are offering, probably Roche is the company which is offering the molecule along with the testing. So that could be the The prognostic biomarker which we are discussing is the biomarker used to identify likelihood of clinical event, disease recurrence or progression in patient who have the disease or medical condition of interest. So CRP is a prognostic biomarker which is identify patient with the unstable angina or history of acute MI with a greater likelihood of recurrent of coronary artery disease or event. Gleason score is another example of the prognostic biomarker in case of prostatic carcinoma. Safety biomarker is a biomarker that is measured for the after the exposure or of medical product or environment agent to indicate the likelihood of presence or extent of the toxicity as an adverse effect. When we see see the nephrotoxicity or the liver toxicity, creatinine or ALT, AST are the examples of the safety biomarkers. Susceptibility or risk biomarker is a biomarker that indicates the potential for developing of a disease or medical condition in individual who does not currently have clinical apparent disease or medical condition. So CRP or HSCRP used as a susceptibility or risk biomarker to identify adult patients with a greater likelihood of incidence of coronary heart disease. So this is actually the data of WHO, which is from 2000 to 2020. And here, if you can see that the percentage of causes of specific death in control total death, which is gradually decreasing throughout the years. This is taken from the fact sheet of the WHO, which it is available on the WHO website. So we, the pharma companies are understanding, or we, the scientists, are understanding more and more the mechanisms or pathophysiology of the this atherosclerosis and vascular disease, where they are using the development of drug, and because of that only, the reduction of percentage of death due to cardiovascular disease is decreasing over the period of years. So this is few of the biomarkers of cardiac disease, and this is the few of the biomarkers of the atherosclerosis. As per estimation, almost 200 biomarkers are right now with the few of the one or another pharma company has given for the drug development that is relevant with the cardiovascular or atherosclerosis biomarker. It is actually FDA data. I am going to skip the mechanism of atherosclerosis, where the vascular endothelial cell and all this stuff. This is again few of the biomarker with the mechanism of atherosclerosis. CRP again, skipping this thing. The one point which I wanted to highlight is the baseline CRP concentration in part is genetically regulated. That is very well established in 2004. So when we interpret this as a single independent cardiovascular risk marker, it is very important to see that what would be the baseline of that particular patient. So this is the structure of CRP. So it is a pentamer structure where each individual is connected with one another, and CRP has two binding sites. One is the phosphocholine binding site, which connect with the FC fraction of the antibodies, and another is the calcium binding site, which activates the complement pathway. In that way, it activates the inflammatory response. I'm skipping this pathophysiology of how CRP adipose tissue. This is something which probably I wanted to highlight, which probably not available somewhere everywhere. The change in the shape of the extracellular matrix, which is again associated with the CRP monomer, mCRP. So these are the papers. It's a meta-analysis which was done. This is probably the monomeric CRP. People are working on that. Here they have given the hypothesis of that. Here they have given the confirmation. Since probably 2012, there is a antibodies are available to develop the ELISA kits. So this is again how the CRP, pentamer CRP, which converted or degraded into the monomer CRP, and it causes this kind of effects in the bloodstream and as well as in the tissue. So these are few of the data which were present with the analysis of MCRP, mostly ELISA, but one assay which you can see that the cytometric bead assay. So someone in 2022 has developed flow cytometric analysis of MCRP also. So why this MCRP has lot of buzz? We'll see in the next slide, but this is again the, as I told you that this is for the inflammation and with the high sensitivity. This is the cardiovascular marker, essentially the same reagents, but the high sensitivity kits. Traceability should be again CRM 70 whenever you are using. These are few of the data where you can see that the CRP is directly associated with the relative risk and the probability of cardiac free survival as your CRP is less than 1 is increases. Here there is a meta-analysis which was done. You can see that HSCRP and homocysteine. This was done way back in 2000. So we are dealing with actually 70, 80 years old parameters right now. So you can see that these are the parameters which are directly associated with the CRP cardiac event. This is the few of the meta-analysis with the number of patients where you can find that CRP is associated with acute coronary event in patients with unstable angina. Post-MI CRP correlates with infarct size. CRP predicts coronary event in patient with unstable angina. So these are in symptomatic patient, and in asymptomatic patient also CRP or HSCRP is directly correlated with the cardiac event or peripheral arterial disease also. This paper indicates that it is an independent predictor of peripheral arterial disease. Now this is something which was published in 2014. Dr. Paul Ridker has done a lot of work for CRP pathways, multiple metabolism in atherosclerosis, etc. So these are few of the mechanisms where upstream target for the biomarkers and these are the downstream targets for the biomarkers, where these are the pathways where pharma companies are working to block this, and enhance this. In that way, the molecule has been developed. So if at all you get the approval for that particular molecule, so that becomes the pharmacodynamic marker of for that particular molecular for CVD, that is MCRP or CRP. Again, this is one of the paper where the study has been done, the trial has been done for graft rejection in the renal transplant patient. So if you can see that they have tried to block this and this, because pentamer CRP is converted into this attenuated, or you can say the PCRP, activated PCRP, and then it is disintegrate into the MCRP. So they are trying to block these two pathways. Further detail of this, again, time's up. So anyway, so we are coming back from the first slide to last slide. The CRP is same as HSCRP, but it is not same as MCRP because the antibodies are different, diagnosis is different. Analytical method must have traceability of CRM 470. It has already been established as prognostic susceptibility risk marker or predictive marker in association with other markers, and future possibility of PD marker. I'll skip that and we'll skip the homocysteine. So, I mean, basically I am associated with the basic research and we are working on this few of the pathways. I can show this on this molecule. So on this pathway. So this is something which probably I thought I should tell the things which are not available in the kit inserts. I think the clap is for the completion of the session or gala dinner for the gala dinner.
Any questions? Because it was a very good presentation.
Sir.
Previously, CRP and HSCRP were been doing it different platforms. You needed nephrometry or something to do HSCRP. Now
Correct.
both are available on the same platform,
Correct.
same reagents, just
Correct.
the dilution factor and serum volume being adjusted in the
Correct.
probe. Can we shouldn't be do away with the names and use single terminology to avoid confusion?
So if you can see all the paper, na, so they are using the single terminology only.
Okay.
So it is for the Mindray people.
Yeah, yeah.
To get the incentive on HS. So the costing would be different for CRP and HSCRP. So if you can anyways get the high sensitivity reagent for CRP, you can very well use it. Because what is happening that the HSCRP has the cutoff is as you say that 1.5 to less than 1.5 3, and CRP has as an inflammatory marker it is 6.
If you use HSCRP kit for CRP values, a high value would you would result in doing multiple dilutions to arrive at a higher
No,
value.
ma'am, the thing is that when you have a CRP kit, na, so what you need to do, you need to increase the sensitivity of CRP kit.
Yeah.
And the lowest limit of detection, you need to go beyond that.
Correct.
So if you have that kit, it is not the matter of the how high it is.
Exactly. So when you are interested on the lower portions,
Correct.
then you
So
use
in case
this
of
high
HSCRP,
sensitivity,
yes.
but when you increase the sensitivity of a kit, there is a compromise on the linear, the high, the upper end of detection.
Why?
Yes, HSCRP has a limit only up to 12 or 12.5, beyond which once again you have to dilute and keep repeating the assay to
No, but
arrive at a
see,
final
if
value.
you have anyways more than 10 HSCRP, na, you can use CRP kit.
That is what I'm saying. So you need to have two kits.
So, any more questions? Or we conclude for the day? So we conclude for the day and come back. Yeah. So we conclude for the day and we come back in an hour's time, and you know the theme and all. Okay. So may I request Dr. Dilip Ghosh Dastidar to please hand over the certificate to Dr. Kalpesh Gandhi, please. Dada ko bula, dada. Dr. Dilip Ghosh. Thank you, Dr. Dilip Ghosh Dastidar, and thank you, Dr. Kalpesh Gandhi. So may I request Mr. Sudeep Mukherjee to please hand over the certificate to Dr. Dilip Ghosh Dastidar as the chairperson. Thank you, sir. Thank you, Mr. Sudeep Mukherjee. Thank you, Dr. Dilip Ghosh Dastidar. So disperse, crowd. So everyone is happy. No, ma'am. I will never be happy. We will start next day. So we want to see everyone dressed in
For the online attendees, please note tomorrow's session start at 9 a.m. So please join using the same link and tomorrow's session will start at 9 a.m. sharp. Thank you. These are the sessions of day 2. So we hope you join us live tomorrow morning at 9 a.m. using the same link. Thank you so much. If you have any doubt, please visit www.mindray.com/in for more details about this event tomorrow.